Firstly, I'd really like to take the opportunity to welcome you all to our second iXCells Rare Disease Awareness Workshop. February is the Rare Disease Awareness Month. This year, we are extremely honored to have a constellation of invited speakers from patient advocates, leading researchers, and biotech entrepreneurs to share their experiences and insights about developing personalized medicine. So we are excited to have you joining us to learn about how we can join forces to tackle these debilitating diseases and advance personalized medicine through a very collaborative ecosystem. Rare disease is becoming a global public health crisis. Based on a recent report by Global Genes, there are about 11,000 rare diseases identified, and the number is going up every day. 20 million people in the United States and 350 million people globally is affected by rare diseases. And 50% of these patients are children, and 3 out of 10 cannot reach to see their 5 years old birthday. Majority of these rare diseases are genetic. They are often caused by a single DNA mutation in a related genes. And because the disease mechanism is usually quite clear, it is relatively easy to find a strategy to fix the problem caused by the mutated gene compared to some other common diseases. However, only 5% of the rare diseases have the FDA-approved treatment. The rare disease patients are generally abandoned by the major drug makers, not because it is more difficult to find a treatment, but because it is hard to make it profitable due to the small patient populations. In the recent years, a group of patient families, they refused to accept the fact that their children will die because there's no pharmaceutical companies willing to help them. Instead, they decided to develop their own drug through their connections and raise funds to support research. And this is so-called patient-focused or patient-driven personalized therapy. There are many challenges in front of us. Science and technology, clinic and regulatory, and finance and business. So we don't know too much about most of the rare diseases because we have poor understanding of the natural history. And usually it is very difficult for the diagnosis, especially if the mutation is happening in the non-coding region. Also, we are lack of animal and cell models. That's another challenge for technical. In terms of clinical and regulatory, because the small patient population, the rare disease is facing a very unique regulatory landscape. And also another challenge is lacking the appropriate endpoints. Even for a patient with the same mutation, the gene, same gene mutation, based on the mutation site, the endpoint and their outcome and symptoms vary quite a bit. So this is making a lot of challenges for FDA to approve the drug, to evaluate the benefit and risk factors. And finally, the financial and business side is extremely costly to develop the therapies. Not to mention, this has to be supported by the patient families. And that's why it is very critical to have the government and insurance company reimbursement to be involved in this part to move things forward. And most importantly is that as a business, as an entrepreneur here, we've been always thinking about how to make a sustainable business model to make the profitability to attract more capitals to the field to really scale and benefit more patients. But however, I talked to many patient families. I remember it's Julia telling me that, we know all these challenges, we know it's difficult, but it is a crime if we don't do anything. So as a doer, not a dreamer, I think if we don't do anything, if we don't make any mistake, there will never be a solution there. So our company, iXCells, has been working with more than 12 patient foundations to explore multiple modalities, ranging from antisense oligo or ASOs, small molecule repurposing, and gene editing, especially base editing. And we are honored and proud to be one of the trusted research partners by many patient advocates. So today, we have three major sections. So the first section is Fighting Like a Mother. We invite three patient parents to share their stories and insights. These are all the pioneer patient parents. Some of them already developed the drug. Some of them still have several pipeline ongoing. We have Julia joining us remotely, and Amber and Michelle is here with us. And after that, we have 25 minutes break, and feel free to grab something to eat. Yeah, after Julia's talk, we can have 5 minutes maybe Q&A section because she has to leave a little bit earlier. And afternoon section, we have four superstars in the research, stem cell research areas. So the topic is unlocking hope. So we're going to talk about iPSC-derived CNS model and a little bit touch about the oligonucleotide or DNA therapy for the rare diseases. And last section is empowering progress. We invited four industry leaders. They are going to share their experience and especially through the interaction with the rare patients to share their insights about the personalized N-of-1 solutions. Because the timeline is short, I want to move to the first section, Fighting Like a Mother. As I said, we have three patient parents. The first one, I think a lot of people in the audience is very familiar with her name, Julia Vattaro. She has another more well-known title, which is Mila's mom. She founded Mila's Miracle Foundation when her daughter Mila was diagnosed with a deadly rare disease called Batten disease. Mila received the world's first personalized medicine together with the physician scientist Timothy Yu from Boston Children's Hospital, who will also give a talk later today. And later on, Julia also founded N=1 Collaborative and Everyone Medicine with the mission to transform the medicine to benefit more rare disease family and children. Without further ado, Julia, so do you want me to play video or?
Yeah, I just 1 minute. Yeah, I would just say first of all, thank you, Nanwei, and everyone at iXCells. And I'm sorry that I can't be there. I know some of you that are there, and I would have liked to be in person. But a lot of you have heard Mila's story, I think. But I do think that it's helpful to always show a very short video of my daughter, just 3 minutes, and then tell you a little bit of Mila's story, even if you've heard it before, because I think it sets the tone for how I'm thinking about individualized medicines in regards to like changing access to rare disease treatments. So with that, I think it would be great if you don't mind playing the 3-minute video to start with. Thank you.
Sunshine, you make me happy when skies are gray.
So our daughter's name is Mila Virginia Makovets. She was born on November 5th, 2010. When I think about Mila as an infant, I think about smiles and laughter and health and...
Half an hour after she was born, she was strong enough to hold on to my finger and I could almost pick her up off the bed.
When Mila was a toddler, she was a climber, she was a hiker, she was a runner.
How old are you, Mila? Here's our sugar high daughter.
Slide, slide, slide. Woohoo!
Very physical, always loved to ride a strider.
Nice, Bug.
Play in the snow, go sledding.
Coming!
We noticed that Mila was getting stuck on her words. She would say, "Mommy, Mommy, I have to, Mommy, I have to," and not be able to continue. And then suddenly, around 3 years old, we started noticing that her feet were slightly unturned and she was walking strangely. And then at 4 years old, we noticed that she pulled books in close. And then around 5 years old, Mila's feet started moving really quickly in this strange pit-pattering way, and then she would fall back on her butt. And every time she stood up, she would fall back and stand up and fall back. You know, we took her around to different doctors who never could really kind of figure out what was going on with her. And then time passes, and then in just 1 month, everything can change. She could no longer see anything. What's brought her so much happiness is playing with her toys and or looking at her books and watching TV and playing with her brother, and suddenly it looked like she could no longer play with anything. It's so hard to see such an outgoing little girl not be able to play with other kids and not be able to play with toys and not be able to watch Elmo anymore. I started, let's say, Mila's kind of story began and her life began, our life began in a way that was very typical, which I know is not always the case in rare disease. But I think her life resonated with many people because she was so typical and she didn't have any red flags. I was never in the doctor's office. She was actually very advanced verbally and physically. We live in Colorado, as some of you have probably heard, and Mila was skiing, you know, at 2 years old. She was going up to the top of the mountain and skiing down. She was in a little rock climbing class when she was like 2.5. She was very into singing and memorizing all the words to Puff the Magic Dragon. And, you know, I think she was like 3 years old or 2.5. She had like declared that her entire preschool make Puff the Magic Dragon like the, you know, annual theme song for the whole school. She was very, very outgoing. And, you know, at 3.5, 4 years old, she started having problems. I carried around a tiny little piece of paper that I wish I still had right now, because I remember it said neurological symptoms with a question mark on it. And the little list got longer and longer. It started with the fact that she was like covered in bruises all the time, and she hadn't been before. It started, you know, changing and becoming what's like question mark eyes. She got glasses. That didn't help. She started becoming really clumsy and falling. She started stepping on her toys. She was pulling all her toys and books into her face. And this list grew and grew between the age of like 3.5, 4 until 6. And I remember when I was thinking about talking today, I remembered something I hadn't thought about in a long time. When she was about 5 years old, I was exhausted because I had taken her to like 100 different appointments around the country, doctors and therapists. And I was so tired, and I had a newborn and, you know, 1.5, I guess a 1-year-old at that point. And she was climbing into the car and she wouldn't get in. And I just got so frustrated and was like, Mila, get in the car. It's so easy. And I remember her looking up at me and saying, like, Mommy, it's not easy. And I was thinking, like, how is this not easy? She's been like jumping into the car for the last, 3 years. And now she's saying it's hard to climb into her car seat. So all of this kind of culminated at 6 years old with a visit for me. I just put her in the car and brought her to the ER at Children's Hospital, Colorado. And that I didn't go home that night. I stayed there for a whole week. And after a week of tests and eventually genetic testing, which was not the first thing that was done at the time, in 2016, I came back with a diagnosis, which at the time, I will be honest, was a huge relief, because I know many rare disease parents understand this, is like, I just had been told I was crazy by so many doctors and kind of poo-pooed so many times and been like, oh, she's fine, she'll grow out of this. And so for me to have an answer meant that maybe I could find a solution. But it took me like, 5 minutes Googling CLN7 Batten disease, which was a rare type of an already rare fatal condition, that it was like the worst diagnosis ever. I had doctor after doctor after doctor that I would find online and email tell me, I'm so sorry, that's the worst disease I've ever come across in the last 30 years. And it didn't take me long to realize that like no child had ever lived, and that I had a few years with Mila at most, and that she was going to lose all her abilities in the meantime. So like many of you and many parents out there, I just did whatever I could, and I read everything, and I reached out to every doctor and scientist, and I talked to a lot of parents, and the parents had lost their children and they were still fighting, and they had foundations. And that was a very pivotal moment in my life, where I realized that this might not be in time for Mila. In fact, there was little to no hope at all at that moment. I was told there was no hope. that if I was going to do this, it had to be for more than Mila. And these other parents were really, really inspiring. And so I started a foundation. I started working towards a gene therapy because little to nothing was known about Mila's gene, where there were like 25 known cases, I think, in the world at the time. And along the way, I was also fighting another battle, which was that she had an autosomal recessive disease needing 2 mutations, and labs could only find 1. And I kept pushing Mila's poor, geneticist, that I kept pushing and pushing, and he was very wonderful and helped me ask more and more labs, and they couldn't find it. So I finally reached out via Facebook in a plea to get speedy access to whole genome sequencing through one of the very few labs at the time that did it. And that ended up not getting me to where I thought it was, but it got me somewhere even better. And it got me to Dr. Timothy Yu, who will be speaking later today, as Nianwei said, who works at Boston Children's Hospital. And he Mila's entire story took a turn at that point. And over the next year, he not only found the missing mutation, but it was at a time when Spinraza, which was an ASO for spinal muscular atrophy, had just been approved that same, I think, 4 weeks before. And so it was on everyone's mind. And all the neurologists were thinking of this as a pretty serious game changer in neurology. And lucky for us, that was on people's mind. And Mila started checking kind of all the boxes. Her disease, her mutation seemed amenable to a similar approach, but they couldn't find anyone else in the world that had the same mutation as Mila. So as you can imagine, a lot of people said, this is crazy. How can you make a medicine for one person? Luckily, Tim Yu, we wouldn't be here today talking about this if it weren't for he and his team that said, why not? And, kept pushing through every open door and thinking maybe they would find a closed door, but they didn't. And within 1 year, a lot happened, of which I'm not a scientist, so I can't tell you, but I was involved in all of those steps along the way. And it was a Herculean effort involving hundreds of people across the world, academics, industry, regulators, that did an incredible job, led by Tim Yu and his team, to turn an idea for a Spinraza-like drug for one person into a drug called Milacin, which was named after Mila. And we moved to Boston. And honestly, we're at the 6-year mark as of a few weeks ago, which is kind of crazy. And until that day when Mila was receiving Milacin, I really never had a moment to actually stop and breathe and think. And, I'd been told a year before that no child had ever lived from this disease, that I was told by NIH researchers that we were 10 years away minimum from being able to come up with any treatment at all. And then here I was sitting in, Boston Children's Hospital, and my daughter was receiving a medicine that was being injected to her brain, and it was targeted, highly, highly targeted to like her mutation, like the most underlying genetic cause of her disease. And I remember thinking, I couldn't even believe it happened. It's as if I didn't look up for an entire year to realize what was happening. In Mila's first year, I will say I was shocked because, Mila did so well. She was spiraling downward leading up to this. She was at a very progressed state. She had lost her vision. She had lost all her words. She had a G tube to receive her, her food, and all her nutrition and her water. She wasn't laughing and smiling as much, and she was really kind of crumpling when we were trying to walk with me holding her from behind. She was really kind of just not being able to lift her feet well and not sitting up straight, and therefore not swallowing well. And everything was getting really difficult very quickly. And in that first year, it was pretty unbelievable. Like, I remember seeing reports on Mila, and I couldn't believe I was looking at my own child's report. That she was like, it stopped, it was for an entire year. Her seizures from 30 a day went down to nothing, sometimes 1 or 2 tiny little movements, but not, arms thrashing and legs thrashing for 2 minutes straight, in the 30 of them. And she started eating by mouth again, and it was pureed foods, and it wasn't perfect, but it was better for sure. And she was sitting up straight. And so that meant that I could hold her from behind, and she could take steps up the stairs, and she was just laughing, at the same. She loved laughing at the times when in the books and the songs when someone would sneeze or cry, and she loved crying baby. She thought that was hilarious. And she was doing that more, like noticeably more. So it was incredible. And then the second year, it was very unclear to me whether or not Mila was kind of stable or slowly declining. And then the third year was when COVID hit, and we lost a lot of support from people that were kind of helping us as well. And her disease was progressing independent of COVID. I could see that slow decline. It was significantly better than where she should have been. She was on 0.5 ml of one seizure medicine instead of 9 or whatever, a lot of children with Batten disease have. So it was significantly helping, but I could tell that it wasn't in time. And that was a very, very excruciatingly painful year for me. And 3 years ago, which is so hard for me because it's 3 years this weekend, Mila, I just always like to say that her very big, beautiful spirit, left her little tiny body. And I don't know how any parent does this. I don't know how anyone continues living after having to wake up every single day without your child. But the reason I'm here today and why I'm still here and why I'm still in this is because, I feel like I carry Mila on my back. I used to carry her around everywhere on my back, and I feel like she's still on my back in some ways. And it reminds me all the time, I feel her, that I'm not alone in this, and that so many other people are going through this. And in fact, millions are going through this. And if you look at global gene statistics, which Nianwei pointed to earlier, if you do the math and look at all those numbers and percentages, it comes to 60 million children will die before the age of 5 from a rare disease. And we can tear apart statistics all we want and say that they're not good. I don't really actually care if it's 60 or 40 or 20 or 10 million. It's still a massive number of children. And that doesn't even include Mila, who died at 10 years old. It doesn't include people that have, children that have lifelong debilitating diseases. It doesn't include adults. And so as I appreciate, Nianwei, you using a phrase that I use all the time, which is, this is a global health crisis. There is no other way to put this. This is massive. And, it keeps me up at night. I will say that we are in a very unique time right now. This was not true when Mila was diagnosed, but the reason we're in a unique time is because technology, the curve that we were supposed to see over those 10 years when I was told we were 10 years away from doing anything for Mila, has gone significantly uphill faster than we ever imagined. And what does that mean? It means that we have the technology today to find the Milas if we want at birth or in utero. And we have the technology to design medicines to treat them. Not all of them right now, but more and more and more, kind of by the day, by the month, by the year. And we Millicent wasn't in time for Mila, but it did show what's possible. It showed that, we have the technology to find her and to treat her, even if it wasn't, unique to one person. And so it kind of points to, I think Mila's story points to a future of, instead of kind of the current system where you have one drug for hundreds of thousands of people, you have hundreds of thousands of drugs for 1 or 6 or 20 or 40 people. That is the direction that we're headed. The problem is that despite having this technology, it still depends on luck in order to access a treatment. And it's very, very simple in my mind. It's either you're lucky enough, and I hate to use the word lucky, but it's true. You're either lucky enough to be part of a large disease group that is commercially viable, so it's interesting to a company and they're going to develop a medicine for it, or numerous companies are, or you're lucky enough to be, a child of a parent that's highly motivated, that's working with a highly motivated physician and is going to raise millions of dollars and get hundreds of people and rally them and push to, have a one-off treatment. And so in my mind, I always picture kind of like the names of 60 million children on the left side. And then these technologies, which are developing and maturing, and a lot of them are becoming more programmable, meaning like the whole process stays the same. It's just the design of the medicine that's different. And you have these children, dying children in these technologies, and the only way to draw a line between them, so access to them, is whether or not you're part of a clinical trial that turns into eventually an approved drug that's for a commercially viable large population, at least 2,000, if not more and above, or this one-off sort of Herculean effort that's like a research or compassionate use IND. But in the middle, there's nothing. And that's where, 99% of these people are falling. And, over the years, I've been very fortunate to be part of thousands of conversations with the academics and the companies, and the regulators and the payers and the governments and all this, around this discussion, lots of really good conversations. But the problem is that here we are 6 years later, and accessing a treatment is not any easier. It's actually in many cases longer, harder, and more expensive than it was for Mila 6 years ago. So we're doing something wrong. And I think what I've kind of in the last year and a half realized is that, and this is kind of a little bold to say in front of lots of like scientists, probably and researchers, but that are in this crowd as well, is science isn't the limiting factor. It doesn't mean we don't have to work on it. And thank goodness especially for iXCells, who's really a critical piece of this, is that has to keep moving forward. But it isn't what's stopping the access. What's stopping the access is that we don't have the infrastructure and the processes that connect the patients to these technologies. And by that, what I mean is regulatory oversight, where it makes sense to have it and where it doesn't. Reimbursement, as Jeanne had pointed out earlier, both of these points. And I think the kind of like underlying foundation of these topics is the risk kind of benefit or the risk tolerance, especially when it comes to dying children with rare disease. So starting with regulatory, the way I see this is that right now, regulators have a system which they've kind of inherited over the years that is it's ill-equipped for this new paradigm. Science is changing, regulation, the pathway is not changing with how science is changing. And this paradigm is so different, it doesn't fit into the current system right now. And currently, regulators see every single step A to Z for every molecule. And the way I see this is regulators have a mandate. Their mandate is to protect public health, and protecting us from thalidomide and other scary drugs makes perfect sense. Everyone agrees with that. No one is going to disagree with that. However, there's another way to interpret protecting public health. And I think it's ensuring access to the technologies that exist today, independent of how many people could benefit from a medicine. And so if that's the case, of which I have found a lot of alignment on that in the UK, where their regulator, the MHRA, is very innovative and does think that that is their mandate. In fact, they think that if we continue doing things the way we're doing things now, regulatory pathwise, we are impeding access to technologies that exist today. There were no technologies before, but now there are. And the system is not allowing us to access them. And so then the question is how do we change this? There's lots of regulatory guidances and there's lots of attempts to try to fix this, but they're relatively small sort of renovations of the house. Whereas I don't think that we can renovate this house. I think we can keep the house. We don't need to knock it down, although sometimes I want to, but I think that it does its job, maybe for large kind of cohorts of people, but we do need to kind of build a house next door. And that house is going to look quite different, actually. And so what it means is that, from A to Z, which of those steps should a regulator be having oversight over? Which of them are not needed? Where are there flexibilities in other parts of the FDA that we can pull from or they can pull from to justify that we have accredited centers, so that we're working, as long as you're working with an accredited center and as long as you're working kind of within these guidelines, you can continue at scale, kind of getting patients to treatments as quickly as possible, submitting data, but not stopping, for example. There's a lot of small areas that could make a very big difference. Moving away from animal models, that's great to say that. I know it's very easy to say that, but there are actually mechanisms in place to be able to do that. And so I think we have to drastically change where there is oversight and where the regulator should be stepping back, the FDA should be stepping back. With reimbursement, I think this is, and Jeanne, I'm glad you brought this up. This is really important. Like right now, this is individualized medicines is an academic endeavor. And this all falls on the heads of academics, and thank goodness that they're doing what they're doing. And there are 500 people in the N of 1 collaborative right now raising their hands because they either want to or are doing this. So there's a huge amount of interest, and they're working with parents who are selling, lemonade and cookies to raise millions of dollars. This is all we have right now, but it's not scalable and it's not sustainable. And so for me, reimbursement is fundamental here, and having a viable business model is fundamental, because we need companies to be able to work with the academics together, like a full collaboration where each of them do what they do best. And physicians are finding patients and they're vetting them, and they're helping design, the trials, and they are treating them, and they're monitoring them, and they're collecting the data and sharing it. That's critical. Companies are coming in with the money, because this is all great, but if you don't have the money, then no one's going to be treated. And we can't rely on parents to do this. And so they bring the money, they bring the drug development know-how and experience, so we should be moving to that model. And the only way to do that is to drastically drive down the regulatory process and allow for reimbursement, which is not allowed right now. This is an academic research pursuit. In the UK, like I said earlier, I found a lot of alignment there. We are conducting a pilot there. And that pilot is specifically trying to kind of test the premise over the course of years, over the course of many patients and different diseases, different ASOs to start with, and trying to see how over the course of that pilot can we shift this regulatory path. So every few patients, how can we learn from that and change the process as we go? So by the end of a pilot of a number of years, we have a new regulatory path that's kind of has proportionate regulation, as they call it. And that we are also kind of figuring out what reimbursement financial model could work over the course of that, so that by the end we have a model that allows for reimbursement and gives incentives for more companies to be in this space, with the hope that there's a bustling ecosystem in a few years of many companies. I started one of them, Everyone Medicines, that's trying to create a business model where there is not one. It's impossible to have a business model today, but the hope is that we can set an example that will allow others, and we can help shift the regulatory and reimbursement. My hope is that there's this bustling ecosystem where there are many companies, many academics working to bring these individualized medicines, starting with ASOs, but moving on to other RNA therapeutics and gene editing, et cetera, that are all kind of programmable, and bringing them at scale. Because I think the biggest problem right now is at scale. We are doing this. There are more people doing this, but it is so hard and it is so slow. And unless we make these big changes, it's not going to happen at scale. The last kind of thought I want to share, and then I'm happy to have, if anyone has any questions, I'm happy to answer any, is that, like I mentioned a minute ago, the base of this sort of house, if you want to call it, if we're trying to build another house for this new paradigm of individualized medicines, it is kind of the risk benefit or the risk tolerance. And it consistently blows my mind that I have friends who have lost their children, unfortunately, to cancer, and little children. And the risk tolerance that's already been established in end-stage cancer in children is there. But for some reason, we're sitting at the table and we're all getting around the table and saying, okay, let's sit down and start from scratch. What are we willing to do for a child like Mila that has 100% fatal disease? This does not mean, by the way, that children that don't have fatal diseases should not be considered here, but it's like the tip of the spear. It's where we're beginning. And children that have, like Mila had end-stage genetic disease. I don't know why we're not calling it that. That's what it is. She had end-stage genetic disease, and yet a girl her exact same age that has end-stage cancer can undergo not just chemotherapy, which already obliterates your system, but that's more well established, a very highly experimental cancer treatment like CAR T or others, where the risks are very high. At most, you're hoping in those cases to maybe buy that child a number of months, maybe a year. And society has already accepted the risk for those children and the willingness to spend a ton of money in hopes that it maybe gives that child a little bit of a better quality of life and a little tiny bit longer life. Sometimes can cure, but kind of rarely. And so we need to be able to use that same risk tolerance when we are talking about starting with, ASOs, and that will move over time. In the sense, you can stop an ASO, you can ramp it up, you can ramp it down. There are adverse effects. We know a lot of, we've seen trends of what they are. We are much better at monitoring for them. We can do MRIs, we can look for hydrocephalus, like we can do all of these things, and we can stop these, by the way. So unlike gene therapy and gene editing, we can stop these. So we've also had ASOs have been around for like 3 or 4 decades in animals and more recently in humans. So it blows my mind that we are on the other side of the Grand Canyon from children that have end stage cancer, and what risk we're willing to take as a society for them, and the regulators and the physicians and others are willing to take. So I think we need to, as parents, and those parents in the room, is really, really align on how we're talking about this, because it's up to us. It's us who the companies, the physicians, the regulators, everyone look to. And those of us that have children with fatal diseases really need to kind of stand up and kind of draw that connection between those two. So all of this to say, this is the most devastating world ever. I wake up every day feeling overwhelmed by how unbelievably devastating and how little is being done in some ways. I also wake up feeling extremely hopeful that a lot has been done, and that I kind of now feel like I can see the path forward. And there are few like the UK that are really pushing to make that drastic change. And there are companies in this space, of which there are a handful only, that I think are really, really trying to do this for the right reasons. And I think, Nian Wei from the top down has set just an incredible culture at iXCells for really being one of those companies that's, trying to survive and has to have a business model and also do the right thing. So I really appreciate that, Nian Wei, that you've brought these people together to talk about this, because you're the right person, and iXCells is the right place to be having this discussion. And so as for my title, we can talk about scaling, but really what it means is how do we get from million to millions? And I think we can, but we're going to have to kind of all do it together. Otherwise, I'm going to be here giving the same talk in, 10 years. So thanks for listening. And I'm happy to answer any questions if anyone has any.
Questions from the audience?
Maybe I have one. Can you say more about the pilot that we referred to in the UK, and what the pilot is about?
Julia, can you hear?
It's very hard for me to hear that. Could you repeat it, Nian Wei? Or
Sorry, I didn't hear. Yeah, can you? Sorry. It's only for speaker.
Hi, Julia. Thanks for sharing your story with us and making the right points going forward. My question was, can you give bit more details about the pilot in the UK you referred to? What exactly is that about?
I can give you a little bit, yeah. So at a high level, the idea was I went over there 1.5 years ago, knowing that their former prime minister's child had died of a rare disease, and that since then the country has really hung their hat on genomics. They have a very unique situation in the UK that some of you might know, where they have, one system, the NHS, where all their clinical and now genomic data, as of recently with Genomics England, which is a company owned fully by the UK government, is sequencing 100,000 genomes, and very recently has just started doing the newborns as well. And so the idea is they have all the clinical and genomic data in one place. They have a country that already really is proud of genomics and is kind of trying to spearhead it. They also just have the academic network of academic centers that have a lot of rare disease centers that are really highly motivated to do this. And most importantly, they have an extremely innovative MHRA in terms of the head of the MHRA. So meeting with each of these people, bringing them together, Department of Health as well, and working with them over the last 1.5 years culminated in a pilot, which is a very, very small scale pilot with a very specific framework around ASOs and children with fatal diseases to start with, and got really great buy-in from the government, including just a few months ago, all the way up from the Minister of Health, kind of approving this as something to move forward with, and they seem really proud of, which is very exciting. I would say I can't give a ton more detail except for to say why I think this happened and why it's different, is that we can have thousands of conversations about how to do this. And we can have lots of intellectual conversations. In fact, there have been thousands of conversations about how to do this. But the premise behind this pilot was, isn't the best way to do this by drawing a circle around something very small scale to prove a model first, and then be able to kind of unroll it, roll it out if it were to work, and roll it out at a national level. Do this with a small kind of representative academic, representative sort of companies, the Department of Health, Genomics England, the MHRA, et cetera, reimbursement as well, all in one small pilot to say, can we treat children, especially the ones that are dying and with ASOs, because it's the most mature modality, and can we use our experience to change the system over the course of the pilot, as opposed to just saying, we need to change the entire country and this is how we're going to do it, because we don't know. The MHRA doesn't know how to do it. I don't know how to do this. The physicians don't know, but we do know the premises is that the current way of doing things is far off of where we need to be. And changing regulatory and changing reimbursement are the two things that we can learn from over the course of the pilot. So that was the purpose. In a way, I like to think of it as like a small little road crew that's trying to like pave a road or a highway, with the goal that if we are successful, we open the road, and then the road is accessible for anyone, any academic, any company, anyone that wants to be on it, as opposed to trying to take on a national level massive change, which in the US would be, I can't even fathom, but even in the UK would be hard to do, to bring all the clinicians on board, set expectations for all the families, change the regulatory process for the entire MHRA. This kind of small pilot allows, with the right premise, the right people decided to sit at the table and say, we don't know the answers, but we do know where we'd like to go, which is over here. Let's do this by treating, learning, and sharing. So that's really critical, is that it's with iterative learnings with every single treatment is going back into this group to make sure that the next patient is done a little bit differently. So I don't know if that's helpful. But that's the overview.
Thank you, Julia. Thank you very much.
Thank
wish you the best of the day.
Thank you so much. Appreciate it.
So, because we're running a little bit behind, so our next speaker is one of my favorite patient advocates, Amber Freed. Amber is the founder and CEO of SLC6A1 Connect. Beyond her role as a successful leader, Ms. Freed is a devoted mother to adorable twins, Riley and Maxwell. On the journey with Maxwell's diagnosis of SLC6A1, a rare neurological disease, become a turning point in Amber's life. With unwavering determination, she left her career in equity research analysis on the day of Maxwell's diagnosis, committing herself to the mission of finding a cure.
I'm so happy to be here today, and I'm so honored to be here for such a special occasion. And I got to see the ribbon and the big pair of scissors. My name is Amber Freed. I'm a former equity analyst, but much more important, I'm Maxwell's mom. I commit 100% of my time to developing treatment for rare disease and founded the nonprofit SLC6A1. I would like to say I became involved in these efforts from pure altruism, but that's not the case. My son was struck by genetic lightning. Prior to Maxwell, I lived in blissful ignorance that this was a thing in this world, and that there were people working on cures for diseases for everybody, and found that's just not the case. So I wanted to share a video of my family so you can meet Maxwell and Riley yourself. The only thing I ever wanted in life was to be a mother. And like a lot of people, it was harder than we thought it would be. With a lot of prayer, 2 years of IVF, we finally received the news that we were having not one, but 2 precious little babies. And it was the happiest moment of my life. Where are we going, Mark?
We're going to the hospital.
I went into labor early, and we were so nervous. And the moment I heard Maxwell's cry changed me as a person. The day we received Maxwell's diagnosis, I went back to work. I told them all something really bad has happened and I have to leave right now. And I didn't even grab my earbuds. I just walked out. And I didn't know what I was gonna do or how I was going to do it, but I knew I was going to do it. I knew I was going to help Maxwell and every other child with this disease. Maxwell was diagnosed with a rare disease that was so rare it didn't even have a name. It's known as SLC6A1, which is the genetic location. At the time of the diagnosis, we were told he was one of 50 in the world and that nothing could ever be done. And just go home and give him the best life you can. Between the ages of 3 and 4, these kids develop a debilitating form of epilepsy, and then they regress because there aren't any drugs that can treat the epilepsy well. So his neurological system will essentially be on fire with no way to help him. I will fight and advocate for him to the death, and if something can be done, I'm going to do it. So I just looked right back at the doctors and I said, if this were your son today, what would you do? And they said, start calling scientists. Every scientist said, have you thought about gene replacement therapy? It looks like this disease can actually not only be treated, but cured. There was one scientist in Texas that we identified as our man. He's extremely busy, nearly impossible to get his attention. So I started sending him Uber snacks and snapshots of Maxwell. I found out he was speaking at the National Institute of Health at a conference. I took a red eye and I just showed up. And there he was sitting in the front row. And I walked right up to the front row and I just sat down next to him and was trying to play it cool. And he turned to me and he said, hi, Amber. And I said, hi, Steve. Can we go out to dinner tonight? And after a 4-hour dinner, we had a plan and we were gonna cure this disease together. The cure is actually created now. We're going to come close to 500,000 within the next couple of weeks. We've raised all of that money through $20 donations on GoFundMe. Gene therapy is being done in thousands of diseases like Maxwell's. So we know it's going to work. We need to raise the money and complete the science for his particular gene. The more money we raise, the faster we can go. We can do some steps in parallel that we're doing chronologically right now. The treatment itself is extremely simplistic. You receive a 2-hour spinal tap where the new gene is introduced into the body through a virus that doesn't make humans sick. The virus goes up through your spinal fluid to your brain, and it's a smart virus. So it attacks the bad copies of the DNA and stacks good working copies of the DNA on top. And viruses permanently alter your DNA. So it's a 2-hour treatment that's once and done for the rest of your life. Someday, this disease will be on a newborn screening panel, so children will be diagnosed while they are still in the hospital, and parents will receive the devastating news that their child has a rare neurological disease, but they'll be treated before they leave the hospital, and they will never become symptomatic. There will never be another Maxwell Fried. I'm coming before you today as a humble mother, asking for your help to help save Maxwell from his rare disease. I really don't have the words of gratitude for you, other than to say thank you so much for helping my baby. So you can see that they were absolute best friends from the beginning, and they continue to be best friends. If I had not had twins and two babies right next to each other, I may not have noticed the things that were different about Maxwell as quickly as I had. And I would go to the doctor, and doctors would say, Amber, you are a type A paranoid mother. And I'd say, yes, obviously, but why is Maxwell floppy? Why does he have these strange movements that I now know are called stereotypies? Why doesn't he play with toys? Why does it feel like he's trapped in his body? And doctors would say, well, boys are slower than girls, and don't compare twins. And over time, I sensed the change in their tone. And I was already 5 steps ahead of those doctors, getting Maxwell into the right doctors at Children's. And after genetic testing, my worst fears were confirmed. And I can just say to everybody sitting in this room, as a function of living, we've all survived our most dark, desperate moment in life. And I thought I had too. I mean, I was hoping not to have any more. But having a little baby in your arms diagnosed with something that nobody can help, that is just the most sickening feeling when doctors say nothing can or will ever be done, and you're not even going to find a doctor that knows very much about this disease. Your best bet is to begin talking to social services. I haven't really seen too many families where both parents are able to work. Start thinking about things you can do for Riley. I remember that moment so clearly, and I wanted nothing more than to crawl under a rock and just disappear and die. And there are many days that I still do. But I'm a mom, and we wake up the next day with the same amount of energy that we had the day before to help our kids. And this wasn't about me then, and it's not about me now. It's about Maxwell, and it's about every other kid with rare disease. And so I made the decision at that moment to fight like a mother. I always like to use the tagline that I'm fighting like the third monkey on the loading deck to Noah's Ark, and it just started to rain. Like, we're going to knock some monkeys off of the boat. We're going to change the way the system has historically been done. And as you saw from the video, that was my call to action. And I set off to create Maxwell's Miracle entirely on my own. I started SLC 6A1 mostly as a backward nonprofit. We know where we need to go. We need translational solutions. We need to target the genetic cause of what is causing this monogenic haploinsufficient loss of function. We have the tools today at our fingertips. How do we get there? So I started calling scientists with the ultimate goal of putting this nonprofit out of business. I would love to be unemployed by this very terrible paying job and depressing, because if I put myself out of business, kids are cured. There is impact. Like let's knock one off and move on to more things. And so we have, in the day-to-day, it doesn't feel like that much has happened, but when you take a step back and look at the course of a year or 2 years or now 5 years, you can really see what's been done. And I understand that we're just the tip of the iceberg. SLC6A1 controls half of the body's most important inhibitory neurotransmitter, GABA. The transporter is named GAT1. Most people don't know the gene SLC6A1, but GAT1 does resonate with them. We're the 10th most prevalent cause of autism, the 6th most prevalent cause of epilepsy, and we're a large contributor to many psychiatric conditions. So you can imagine the type of impact you could have by making discoveries in this gene and what other breakthroughs that could lead to. These children with SLC6A1 may be able to live into young adulthood, but I'm not sure many of us would want that. Like, as adults with functioning minds, would you really want to live like these kids do? It's a terrible quality of life. There's no place for them in society, and as parents, like, we hope that our children outlive us, but what if you're terrified of dying as a parent? What if you can't die because there's not going to be anybody to take care of your child? And that's what I see with SLC6A1. The way we've identified our oldest patients, which are not old, is that, like the kids, they're severely intellectually handicapped, they have refractory epilepsy, they suffer from mental illness, and they have a movement disorder. And they are fertile, so they have kids. And when they have kids, a lot of times they are part of the justice system in some form, and their kids are taken away immediately, and they're put in foster care, and then they're able to go to some type of center of excellence where the children receive good health care and a lot of times genetic testing because something is just not right. And then they become part of SLC6A1. We have families of 4 or 5 kids, all with the same disorder. And as a society, we are juggling to provide medical care for these families. Kids are separated from their parents. Parents can't take care of themselves. Here is a solution to so many problems that we're all dealing with. And this cycle right now could make me vomit, but it's also such a big opportunity for change. And so for SLC, I like to think that we are a catalyst for change. The old paradigm of drug development is being turned upside down right now because parents are impatient. Parents don't have time for the way things have been historically done. We really need answers, not in our lifetime, in the next 5 years. We've created a mantra of saying, I'm going to create my own miracle. I am captain of my own ship. And I had no idea what I was getting myself into when I set off on this journey, but essentially, parents are tasked with landing on the moon, get an honorary PhD, become an MD, support yourself, support your family, and take care of super sick, loved ones, all while raising the most ridiculous sums of money in $10 increments, creating GoFundMes, and bearing your darkest heartache on social media. It's really kind of I don't know, like a sick joke of like nobody likes Facebook, but here, go on Facebook and start crying, talking about the most terrible flaws of society and terrible things that have happened to your family. So I do like to touch on our common core beliefs. And my dream for SLC6A1 in rare disease is that parents are given this terrible diagnosis. Like your child was just diagnosed with a disease too rare to have a name. It sounds like alphabet soup. But when that diagnosis is delivered by a clinician, they also come to that family with like a cheesecake factory menu of options. And they educate the families. Maybe small molecules are the right thing, or antisense, or gene replacement. And they educate those families. And the families can make the best decision for themselves. So I view SLC6A1 as the glue that binds scientists creating the options, doctors implementing those options, and meanwhile, educating families so that we can empower them to make the right decisions. We bring together the patients, scientists, and industry partners to create what will someday be a much different future for our children than what we have experienced for ourselves. And some of our accomplishments in the past couple years have been a 3-year natural history study that we own, a publicly available biorepository, 2 registries, building infrastructure from the ground up, get an ICD-10 code, work with your state on newborn screening initiatives, doing everything entirely by yourself. Like, go, absolutely engineer mice of your kids in China. What? Okay, like I don't even know what that means, but we did it. Now I know what it means. And I think it's really important to brag about iXCell for a moment, because every day we are juggling all of these insane expectations and things we know nothing about. You just want one thing in your life to be easy. And for us, that has been iXCell. We've been able to streamline everything we do in cell systems because we have a reliable, consistent partner, and we trust those test results every single time. So we're finally at a stage where we're moving things out of academia and saying, design whatever it is you have and send it to iXCells, and it's fine if it's blindfolded, whatever. But that's where we keep our cells. And so everything can go to one central location and we can get things done on a timeline that would make sense to the Mayans instead of science talk. I refer my friends all the time, of course, to help everybody, but to make their lives easier, just one thing to relieve stress. And I can say that it's relieved my anxiety like much better than Zoloft did. We started with the little boy you see there, but we've grown into what you see below. And constantly new families are contacting me, like all of the time. New organizations are popping up and we're finding better ways to do things. SLC6A1 is a transporter. I have my friend Monica Elnikave here. She's a GABA receptor. We've paired together. She's working with iXCells to begin things. And we are making insane progress, but of course it's never as quickly as we want it to. Even with that, we wake up, we can take more punches than Mike Tyson in a day and wake up with the same resolve the next day to keep working because it's our kids. And this is, as Neon Way said, it's a crime to turn your back. So in closing, if you would like to follow me or connect, that is my LinkedIn, and I update regularly. I'm going to do a better job of updating more frequently. And thank you so much for having me here today.
Thank you so much, Amber. Your words means a lot to us. Again, we're trying to rush a little bit here. So next, our speaker is Michelle Jeanette. Michelle has a very beautiful family located in San Diego. Last week, she invited me and our coworkers to her family. She has 4 lovely boys, and the youngest one, Tristan, was diagnosed with KCNH1 related disorders. We were very impressed and shocked to see how the rare disease can affect a family from so many different aspects. Michelle is committed to driving the research and improving the treatment options. She believes in making a meaningful impact on the lives of children and families. Without further ado.
Well, I have to admit, I was a little intimidated to have to speak right after those 2 legends. I don't know what I can possibly add that they haven't already done times 10, but I'm incredibly honored to be here. And I hope I can share, Julia and Amber gave a lot of big picture points. They're a lot further along in the process. So I hope I can kind of provide a bit of a case example of what the early stages of what this patient-led drug development can kind of look like. So when my son Tristan was diagnosed in 2021, and we were in that stage of complete shock and grief and knew nothing about rare disease or genetic treatments, someone sent me an article about Amber and how she had raised millions of dollars to cure her kid's disease. And we figured, well, if Amber can do it, we can do it. Why not? And so that's what started this whole process of us diving into this world of rare disease and drug development and trying to move mountains to help Tristan and the other kids with KCNH1 diseases. As it turns out, I cannot really quite compete with Amber's ability or Julia's to fundraise, but we do our best. So I wanted to share our video, although I hope it's gonna play a little better.
This is when we met Tristan for the first time.
Tristan was born November 26, 2019, the youngest of our 4 boys. From the minute I saw him, I thought something looked a little off about his eyes, but none of the doctors or nurses mentioned anything or seemed concerned, so I didn't think too much more of it. When he was about 4 months old, we started noticing that he wasn't reaching for things or bearing weight on his legs like his brothers had. So we got him into PT and OT and figured he would catch up, as many children do. He's not that far behind physically, so we were completely unprepared for the severity of the diagnosis. The one that hit me hardest was the fact that he would probably never speak. I don't know why, but in all of my wildest, worst case scenarios, it never even crossed my mind that my little boy wouldn't be able to talk to me. It took a long time for it to start to sink in. That first month, every single day, it was a fresh gut punch. I would wake up in the morning, fine for a second, and then suddenly it would all come rushing back in and it would literally take my breath away. And then about a month after the diagnosis, he had his first seizure.
When I pulled up to the house, I actually heard the sirens coming down the street as I came in. I'll never forget when I walked in to the house and saw Colt at the top of the stairs, and the look on his face said, Daddy, something's wrong with Tristy. I rushed up the stairs, and of course, fearing the worst, and didn't know what I was going to find, and I come in, Michelle's over Tristan, he's on the ground, and she's trying to calm him down, stroking his head and singing to him, and trying to get him to start breathing. And I mean, Michelle was a rock star that day, and the paramedics and the firefighters, they followed me into the house afterwards, and they got him on oxygen, and put him in the ambulance and rushed him to the hospital with Michelle. It's just, that's when it all hit me. This is real. Tristan has this life-shattering disorder, and our family's lives will never be the same.
I've had to almost completely give up my private practice because of all the therapies and doctor appointments and exercises. He's in therapy 5 days a week, hours a day, but the progress is painfully slow. As he's gotten older, it's become more and more clear how impaired his little brain is. He works so hard to learn things, but even when something finally clicks, and he's been doing it consistently for weeks, it can suddenly vanish. And if you ask him to try to say or do something that he used to know, it totally breaks my heart. He's looking, his eyes are darting all over your face, his mouth is moving, he's breathing hard.
I mean, he's trying so hard, he just can't get his body to cooperate.
Tristan is the sweetest, most loving little person. He's always toddling over to lay his head in our laps or grab us as we're walking by. When he's alert and engaged, he's such a happy, affectionate little love bug. But a lot of the time, he's just off in his own world, kind of checked out, zoned out, like at Christmas. All the other kids were so excited and tearing into presents, and he's just sitting there, staring off into space. I just want so desperately for him to be able to participate in things with our family and enjoy things with the rest of us.
Thinking about the future is pretty overwhelming. I mean, these kids can't ever be left alone. They can't feed themselves. They can't ever even be potty trained.
And it just gets harder as they get older. I mean, just imagine having a Kevin-sized toddler for the rest of your life. It becomes very difficult to do anything as a family. We see with other families, they try to do something as simple as going to a game, and they end up spending the whole time trying to keep their full-grown kid from grabbing other people's food or climbing over the seats. It would be one thing if these kids were happy and healthy overall, but it doesn't seem like most of them are. Many will go months where they only sleep a few hours a night. Lots of the kids are on anxiety meds for agitation, and some will go through periods where they have hours-long crying spells every day. And of course, it can be impossible to figure out the problem because they can't tell you if they're in pain or what's going on. After spending every spare minute for almost a year researching KCNH1 and contacting scientists all over the world, we've learned that there's a clear path forward for KCNH1. It's a very good candidate for gene expression therapy. In other similar disorders that have been treated with this therapy, kids have gone from wheelchair-bound to walking, nonverbal to verbal, multiple seizures a day to seizure-free.
The technology exists, but the funding doesn't. As far as traditional drug development is concerned, KCNH1 is too rare to care. That's why it's up to us to make this happen. We need to raise $1 million to cure KCNH1.
There we go. All right. Thank you, Kevin. Anyway, so exactly a year ago, finally, after a long time, we finally had our very first ever KCNH1 patient-derived iPSC line. And KCNH1 is an ultra-rare disease. There's now maybe 100 patients diagnosed globally and only a handful of researchers studying it around the world. But as soon as we had those cells completed, we started shipping them off to anyone and everyone who would take them. So I have a little schematic here just to share the far-reaching impact of just one asset, the very first asset our foundation developed. So we developed the patient-derived iPSCs along with sibling control from Tristan's next oldest brother. We sent them to, let me get my little, there we go. Hello? Oh, there we go. Can you see it?
Yep.
Oh, okay. Well, anyways, we sent them to, okay, I guess it doesn't show up on my slideshow. Anyways, we sent them to iXCells, we sent them to Al George's lab at Northwestern, and we sent them to our KCNH1 expert at the Max Planck Institute in Germany, who's been studying the channel for 30 years. And in order to avoid the headaches and delays of trying to import into the EU again, that researcher was able to share them to another research team in Italy that is studying the ciliary effects of KCNH1 mutations.
Sure.
Okay.
Maybe. Yes.
No, nothing's showing up.
Oh, it shows up from the, oh, it
Oh,
does show up. It does show up. There you
Yay. I need an angle.
Maybe you can stand.
Oh, I have to hold it down. Okay, there we go. So anyways, our researcher here at the Max Planck Institute shared them to Italy, as well as to another team of channelopathy specialists in Germany at Tübingen. And they were able to develop the first KCNH1 neural networks and able to see a phenotype. And then meanwhile, Al George generated an isogenic control for us, which obviously is a big step up on the sibling control. And he sent them back to us here at iXCells for us to generate our neurons and our brain organoids. And after connecting through our foundation, he also generously shared that isogenic control to the team at Tübingen. And they were planning to have to do an isogenic control. That obviously saved them a lot of time and expense, and they are now on the verge of being able to start drug screening in that model system. And then finally, just a couple weeks ago, we got a request from a lab in Australia that focuses on animal toxin therapeutics. Pretty cool. If you have a chance, look up, there's an article. They apparently have like 500 venomous animals in the lab, and it's pretty crazy how they like manually milk the venoms from snakes, etc. Anyways, they have a couple of spider toxins that potently inhibit the KCNH1 potassium channel. And so we're really excited to see how those studies will go. So just a little snapshot of the work that our foundation has done in 2 years. We generated the first ever patient-derived KCNH1 iPSCs, shared them across soon to be 3 continents, 3 countries, 5 research groups in less than a year, resulting in the creation. So there's our iPSCs, resulting in the creation of the first KCNH1 neurons, neural networks, and brain organoids, disease model systems. We also spearheaded the development of a KCNH1 knock-in mouse model that will be made available to the research community. We've also funded, in terms of drug development, treatment development, we have funded a repurposed drug screen and designed, manufactured, and screened a small batch of ASOs. And both of those projects yielded promising hits that have been shared to numerous partners and will now be validated in the CNS model systems. We also plan to launch a base editing project here at iXCells in the next few months, and we're really excited about that modality. After connecting with our organization and learning of our progress on disease models, we now have 4 research teams that are pursuing large grants for KCNH1 studies, which is amazing because those are KCNH1 research dollars that I don't have to fundraise. And we now have also 2 industry partners who have launched exploratory KCNH1 drug development programs. So in the span of 2 years, from having 0 disease model systems other than simple transfected cell lines and having 0 efforts underway to develop treatments for KCNH1, we now have multiple in vitro systems at multiple labs around the world and multiple treatment modalities that are being actively investigated. Unfortunately, at home, things have not been going so well for Tristan. As I had mentioned in the video, if you could hear it, there are a lot of the KCNH1 kids will go through phases where they're just really distressed. They spend hours a day crying. And unfortunately, that started happening for Tristan within the last year. And it just is completely heartbreaking for us to see him suffering and have no idea how to help him. And you know, it's really all encompassing when he's in one of these phases. We have to either just sit with him and try to comfort him or try to carry on and get things done, but we really have not learned how to do that. It just basically wrecks us to see him like that. And we know, you know, if he could even so much as point to a body part that's in pain or nod or shake his head to answer questions, it would be completely life-changing. You know, just the ability to communicate even a little bit would completely change his and our family's life. We have been working intensively for years now to help Tristan be able to communicate with an iPad communication device. And this was this past summer when, for the first time ever, he independently, unprompted, was able to ask for something. He asked for some water. And it showed us that he actually does get the concept that he can use this to express himself to us. But like so many of his other communication skills, he showed it a couple times and then he lost it. And so we're just left, you know, imagining how frustrating it must be for him. We know he understands so much more than he can convey, than he can express, and how frustrating it must be for him to not even be able to express his most basic needs, to be basically trapped in his body. Then this fall, we had a really terrifying seizure relapse. He had been on an investigational repurposed drug and seizure-free for 18 months. All of his seizures have been triggered by illness. That's why you'll probably see me wearing my mask, because we all try to protect him. But he got a stomach bug, and each of his seizure episodes is just more and more intense and dangerous. So he had 9 seizures over the course of 3 days, 4 days in the hospital, and a just really, really rough time recovering. It took weeks and weeks for him to get back to himself. And, you know, so it just leaves us wondering, is it a dose issue? Is there some kind of compensatory mechanism kicking in? We just know so little about these complex CNS disorders, and we're hoping that these model systems can help us learn a lot more. These are pictures from our vacation this past summer. Tristan unfortunately spent the vast majority of the trip cooped up in the living room of the Airbnb. And of course, one of us always cooped up with him because he would just get so upset whenever we tried to take him out. And then the last day, we took him to a different beach for whatever reason, he suddenly loved it. And I can't even describe what that meant for him to be able to actually enjoy something, for our whole family to be able to enjoy an activity together. That's one of the few times that's ever happened in his life. And we just, we know if Tristan was able to share more with us about his needs, what he likes, how we can help him, that we would get a lot more days like this with big smiles, with joy, with him being able to really participate in life more. And of course, we all know this is not just about Tristan, right? Tristan represents millions of kids and adults with rare diseases all around the world. And we know that if we are successful, these targeted genetic treatments are so cutting edge, and there are so few of them in existence yet. We know that if we are successful in developing something that helps Tristan or Maxwell or any one kid or any one gene, that is a significant advance for the whole field. And I mean, I'm sure you guys all know Mila's story, that she was the trailblazer for personalized ASOs. And I know Julia's frustrated that we haven't made more progress, but her story has inspired so many people and had such a wide-ranging impact. And so I want to thank you all for being here, for dedicating your time and your talent to the rare disease community and helping us make a difference for our kids and all the other rare patients out there.
Thank you, Michelle. It's always very inspiring to see a lot of parents from totally different backgrounds. They're starting to, in a very short time frame, they become a PhD scientists. So it's a very, very powerful story from all these 3 moms. They are all my heroes.
So our first section in the afternoon is Unlocking Hope. We invited 4 researchers. All of them are they're thought leaders in their field, their own field. So we are going to focus on iPSC-Derived CNS model and ASO development for rare diseases. And our first speaker in this section is Jeanne Loring. Jeanne is a professor emeritus at Scripps Research Institute, founder and founding CSO at Aspen Neuroscience, board of director of Summit for Stem Cell Foundation. She is the author of more than 250 publications, inventor of pluri-test. Jeanne developed one of the first NIH-funded human embryonic stem cell courses. So she has another well-known title as the godmother of stem cells. Many young scientists, including our employees at iXCells, are familiar with her book, Human Stem Cell Manual and Laboratory Guide, which is considered the Bible for stem cell culture. So her work is still continuing to inspire a lot of young scientists and entrepreneurs here.
Yeah, thank you very much. There's a new edition of this in the works, just in case you're wondering, and I may be asking some of you to write chapters for it because I just got the first of the year reminder that I have an obligation to finish this book. So, okay, so I'm going to tell you 2 stories today. I'm going to tell you a story about how we went from stem cells to a therapy for Parkinson's disease, which is going to start, I hope, within the first quarter of this year. And I'm also going to tell you a very short story about other work I'm doing right now, which involves sending neural organoids derived from iPS cells to the International Space Station to try to find out what the effects of microgravity to perturb them in a different way. Whatever reason, it's a lot of fun. Okay, so I have disclosures. I founded Aspen, and I said there's a clinical trial. I am scientific advisor for a number of companies, in fact, more than this now. Novaglia, Conception, Symbiosis, and CademaStem, and I'm also an ethics advisor for Merck in Germany. And I've done a lot of bioinformatics, and I do machine learning, but I'm really interested in AI. And my main use of AI right now, ChatGPT, is to ask it to write haikus. So this is my haiku to begin. Unlocking life's code, 25 years of promise, stem cells light the path. And it's been 25 years. I've worked on these cells for 25 years. As you know, pluripotent stem cells are unique. They aren't like other stem cells that you find in your body. They exist only in cell culture. It's important to note that they're the only non-cancer cells that don't senesce, that continue to divide in culture. And they're also able to differentiate in any cell type in the body. And as you know, there are two sources. One was the first human embryonic stem cells were reported in 1998. The first mouse embryonic stem cells were reported in 1981. So we're really standing on the shoulders of giants here. This has been going on for a very long time. We just happen to be working on humans. There are a number of pluripotent stem cell therapies that are now in clinical development, including age-related macular degeneration, Parkinson's disease, epilepsy, lipodystrophies. These are using different cell types that are derived from iPS cells. And in most cases, it's a cell replacement therapy, but not always. There is a company here in San Diego that's working on hair loss using epidermal cells from iPS cells. iPS cells are being used for cancers by Fate and Shoreline here in San Diego. Inflammation is being treated with mesenchymal stem cells, which are the cells that you would find in fat, for example, of us right now. But in this case, they're making them from pluripotent stem cells, from iPS cells. And that, as you know, has all the advantages of being able to get unlimited quantities of cells and to be able to characterize them really well before you use them in the clinic. Heart disorders have been really challenging. Blue Rock, HeartWorks, and StemCardia. StemCardia is a new company started by a group at University of Washington, Chuck Murray, and also type 1 diabetes. And there are a couple of companies working on this. There's actually a lot more than I have listed here, Vertex and Sana. These are kind of the easy targets right now. And so there are multiple groups working on each one of these. What did I do with my thing? Here it is, I got it. No rescue necessary yet. So Parkinson's disease is caused by the loss of a particular type of neuron in a particular part of the brain. The part of the brain is called the substantia nigra, and the symptoms don't arise until about half of those cells have died. It's progressive, so those cells will continue to die and the disease will get worse. There's a drug therapy that allows people to have a normal life for a fairly long period of time, but it doesn't help the loss of neurons. So there is no therapy that prevents the continued degeneration of dopamine neurons in this disease. But Parkinson's disease is an especially good and early idea as a candidate for cell replacement therapy. And this goes back for another 30 to 40 years. There were a group of people who were treating Parkinson's disease patients with fetal tissue. This is before there were any embryonic stem cells or iPS cells. So they would take the precursors from an embryo, very small embryo, 6 weeks or so old, 6 to 10 weeks, and they take this little tiny piece out and then implant it into the brain in the part of the brain that receives the dopamine neuron input. And that was really important because that's a pretty large target to do a transplant, whereas to try to put the cells right back where they originated would require a smaller target. It is a smaller target, but also would require them to then grow processes back up to the striatum. So they put them directly into the striatum so they could connect really quickly. Now, this didn't work very often. It worked very seldom, but this is where hope comes from. Once in a while, it worked beautifully. There were a few patients who never had to take drugs again and were completely cured of the disease. So our goal was to make this more common, to make it possible to be consistent in treating the disease and reversing the symptoms. I wanted to point out Kurt Fried here, who wrote a book called Healing the Brain. It's his own personal story about the fetal cell transplants. It's a really interesting book. So in around 2017, there was a meeting in Kyoto. It was a cooperative decision for multiple groups who are working on cell therapies for Parkinson's disease to all get together and share their methods. And we published a paper about it. So this is really important. Having colleagues who are willing to share information with you over and over and over again is the only way that you can make one of these things work. None of us can do this on our own. The 4 groups are still the 4 groups that are starting clinical trials now. There was the group in UK and Sweden, Roger Barker and Malin Parmar. They were using embryonic stem cells, and similarly, a group in New York, this is Blue Rock, is using embryonic stem cells. Jun Takahashi in Kyoto is using iPS cells, but they're not matched to the patient. So all 3 of those require immunosuppression for at least a year. So this is being treated like a kidney transplant or an organ transplant. You have to be completely immunosuppressed. And we don't know whether you can actually ever come off immunosuppression because there's no information, there's no history of doing this in humans. So when we started doing this project in about 2012, this is the autologous iPS cell project, we decided to take advantage of the unique benefits of iPS cells, and that is personalized therapy. We can make those cells from any person. So all we had to do was to turn those into dopamine neurons and then get them approved for a clinical trial. And that took us a bit longer. This is a story that you could read in the Wall Street Journal. This is about our efforts, and it gives a really good overview of how iPS cells are generated and how they're used to treat Parkinson's disease. This really is about the company that I started called Aspen Neuroscience. So why would we use autologous patient-derived cells instead of a single line? It is harder in some ways. It's easier in other ways. So I think it really evens out in the long term. If you're using allogeneic, which is unmatched, that means that you do have to immunosuppress the patients, no matter what the therapy is. Manufacturing is in the form of scale up, and so you make hundreds or billions of cells in a big bioreactor. The problem starts coming when you start worrying about something that happens over and over again, iPS cell lines, and that's genomic instability. When cells divide, remember I told you, they're just like cancer cells, they can proliferate forever. Well, they're just like cancer cells. Every time they divide, they acquire mutations. So that's something we always have to keep in mind. It's sort of the dark side of pluripotent stem cells. The last one is redosing, and there's a positive aspect of that. If the cells for some reason don't survive in the first transplant and we are given permission to do so, then we could transplant the cells again because there'd be no immune response, no buildup of immune response. So I want to talk a little bit about the perils because this isn't mentioned very often. And this is from a paper that we published quite a long time ago. 2011. And in this paper, we took 186 pluripotent stem cell samples from all over the world. This was more of a task of me showing up at conferences and asking people for their cell lines. That turns out, I think other people have used that sort of idea. You show up, you ask the researchers directly for stuff, and they'll give it to you. So that's how we gathered them all. And then we analyzed them by SNP genotyping, which is the same methods used for 23andMe and Ancestry.com. It gives you an overview of the entire genome. So we found that there were a lot of copy number variations. You can see just in this diagram on the right side of the slide that everything that's red shows an example of a duplication. Sometimes the duplication is an entire chromosome. And what I'm trying to say is that if you don't look, you won't know, because none of these people who sent me their cells knew that they were abnormal. They all forgave me because they were co-authors on the paper, but a lot of them had to throw those cell lines away. But at least we knew that they were bad and they didn't invest any more time in them. So the reason why they acquire mutations is they have a what I call evolution in an incubator, and it really is evolution. A cell, one cell in a group of cells will have an error. That error gives it a selective advantage, just like selection, evolution. And then as those cells expand, that cell will proliferate. It'll be more resistant to stress. For whatever reason, it becomes dominant. So the whole culture has now evolved into a mutant culture, and those mutants are often associated with cancers. One of the ones that we see a lot is a lack or a mutation in P53. And we have found in 2017, in fact, P53 mutations were discovered in a whole bunch of embryonic stem cell banks. These banks were, in some cases, actually clinical grade cells. So you have a clinical grade bank, you've invested, I don't know, $1 million in it. You discover there's a P53 mutation in that bank, and you have to throw it away. Unless you can figure out some reason that the FDA will allow you to use cells that have a P53 mutation, because they are cancerous by definition. So what we did when we decided to do the autologous therapy is develop a lot of genomic methods in order to quality control the cells, because we had to make the cells each time from each individual. And in order to do that, we needed a lot of help in qualifying those cells to make sure that we got the same product at the end. So essentially what we did was this little loop here, you see. We start with somatic cells. We start with fibroblasts. That's more traditional than anything else. They're fine, but you can use other cell types. The somatic cells are analyzed by whole genome sequencing, and then again by SNP genotyping to look for structural changes. Then they're turned into iPS cells. We use Sendai virus with the Yamanaka factors because the FDA thinks that's just fine, but it also works really well. And then at this stage, we have, instead of using like a panel of antibodies or some other kind of single marker analysis, we decided that we would look at the entire genome of our cells that were our iPS cells and our cells that were going to be used for transplants. So this is done by RNA sequencing. We developed two tools. One is called PluriTest, and that's widely used all over the world for making sure your cells are pluripotent when you start with that. But we also developed another tool called NeuroTest, which is to make sure that the cells that we are going to transplant to patients are at the ideal stage. And this requires gene expression profiling and bioinformatics, but essentially it's not very hard, because what you need is a whole bunch of examples of things that work, examples of things that don't work, and then you can have your computer solve that equation and be able to predict what sort of profile works and which one doesn't work. So the whole genome sequencing is done actually at three times during this process. This is essentially what we presented to the FDA. So this is what Aspen is doing, whole genome sequencing 3 times, because it's really important not to have any dangerous mutations in these cells. And then the RNA sequencing is done twice. Oops, sorry. Okay, now this should show you. Let me see if I have to poke this. Yeah, here you go. You're going to come and rescue me? You're going to do it. You do it. Okay, so this is the assay that we use to show that the cells are efficacious. It's a rat model. What we do with these rats is to get rid of the dopamine neurons on one side of the brain. That results in them rotating. When you give them amphetamine, they'll rotate in the direction of that lesion. And so you can measure the number of rotations. That's how you quantify it. And, it isn't exactly Parkinson's disease, but it is really, it is definitely the same neurons. So this is the work that we used to present to the FDA. We showed that the rats that were lesioned and then treated with a transplant of human iPS cell-derived dopamine neuron precursors were recovered completely over time. So you can see that in that those two colored lines, the blue and the green, that they return to normal over a period of about 24 weeks. Not fast, but the reason it's not faster is because they actually have to make synapses. These are young neurons. They have to make synapses in the brain in order to function. Oh, there's another one moving, and now another one moved. There you go. See, there's the transplanted one, not circling. Okay, so I'm going to show you briefly how our rationale for predicting the fate of cells. These cells can make everything, right? So how do we, when we differentiate them, how can we be sure that they are going to make the cell type that we need? Especially that is difficult when the cell type that you need is not going to be developed in culture, but is going to differentiate in the brain. We can't transplant fully mature neurons, they die. And it turns out, I'm going to show you in a moment, that we have to make sure that the cells are at exactly the right stage. And I'll show you how we did that. This is an example from pluritests, and this has become a very popular tool for demonstrating that your cells are pluripotent. It's popular because it's free, at least it was free, I think it's still free. It's free, and it takes no work except for an RNA sequencing of your sample. You upload it, it comes back down, it tells you whether it passed or didn't pass. You don't have to know anything other than that, and that's why it was really popular. Second reason it was so popular is that journals accepted this as evidence of pluripotency. So we use the same model essentially to create neurotest. In this case, we're trying to predict that cells that are at a neural precursor stage are going to become dopamine neurons. And we use exactly the same strategy. You just have to generate lots and lots of cultures and have some of them work and some of them not work. So that's what we have done. And we measured either the in vivo recovery of these cells, like I showed you earlier, or we showed that these cells become fully mature dopamine neurons. And that requires showing that they will release dopamine just like a neuron is supposed to in culture. They also fire a very typical bursting behavior if you do electrophysiology on them. So they're very easily identifiable once they're mature. But we needed to get a gene expression signature that told us that they would get like that, right? Okay, so this is the publication on the primary work that we did that outlined the entire thing that we submitted to the FDA. And that was our work to determine exactly when in culture, and when I say when, I'm talking about gene expression signature, not days. Gene expression signature changes over time, so where's the sweet spot in that? And we also were interested in discovering why some cultures worked and some didn't in order to narrow it down. So what we discovered was that if we grew our cells, in this case, for 18 days, under differentiation conditions, they would get recovery in the rats. If we grew them for another week, 25 days, which actually was the time point that was originally used in the rat studies long ago, they didn't work. However, they engrafted in both cases. So what's going on here? Well, you can see here on the left that there are effective cells and there are ineffective cells. In both cases, you see that the brown is tyrosine hydroxylase. That indicates where the dopamine neurons are. And you can see that there are dopamine neurons in both of those transplants, but only in the upper one were they effective. And the reason for that is that they grew out, they had outgrowth. They sent out neurites. They connected with the host cells. And you can see when we did the gene expression analysis, we discovered that a lot of the genes that are suppressed by a particular transcriptional repressor called REST were all lower in the cells that worked and higher in the cells that didn't work. In other words, there was a switch, and we had to be before that switch, because then a whole plethora of genes associated with neuron differentiation and maturation couldn't be on or the cells wouldn't work. So even harder. We don't even, we can't just look at them and say they're going to be neurons. They don't look anything like neurons. Okay, so that study, and you can look just at the bottom of this, was approved by the FDA on August 8th last year. Aspen Neuroscience, the company that I started, got approval for a phase 1. It's actually a phase 1/2A. And the other three people that I talked about who were all in Kyoto are also all doing their clinical trials. One of them is Blue Rock, Sloan Kettering and Blue Rock. And they got FDA approval before the rest of us. And they have finished their phase 1 trial with 12 Parkinson's disease patients after 1 year. They reported on the 1-year results, which are not terrible, but not very impressive. That's the point at which they get rid of immunosuppression. So we're all looking forward to the future to see whether those patients get better or they get worse, because that's going to be a really important thing to know. So the other ones are Kyoto. Kyoto has taken a very long time, and they've been very quiet about telling us what's going on. I did learn from the PI that the cells are surviving in the patients, but they are not giving us any information about how they're doing. The Lund University, that's the Swedish and UK group, is now partnering with Novo Nordisk in a phase 1, which they started last November. So they're slightly ahead of us, but not by much, and we're still the only ones who are using autologous cells. So we don't have to worry about that immunosuppression, like when you take it off and when you put it on. I want to acknowledge this because this is really important. This actually evolved very much like some of the stories you've already heard about rare diseases. I couldn't get funding to save my life in 2011 to do this. I mean, it was people just said, I mean, they actually kept saying it was impossible until like August 7th of last year. And when we got FDA. So the way it started was a group of Parkinson's disease patients and their advocates climbed Mount Kilimanjaro, which is not a small mountain. It's 19,340 feet. And they had gotten a promise, if they did this, that they could raise funds to start development of this autologous therapy in my lab at Scripps Research. And then they did some other things. They went to Everest Base Camp. They did other hikes. And all in all, the Summit for Stem Cell Foundation, which was the group that does this, raised $3 million for me over this period of time. When we got the first work done, we started getting grants, which helped a lot. And then in 2018, I decided to start Aspen Neuroscience, and we have raised about $220 million to date. And that may sound like a lot of money. Anybody think that's a lot of money? It's a lot of money. It'll get through the phase 1 easily, and presumably get to the stage where the company can raise more money for phase 2. So we also got an FDA fast track designation, which allows more interaction with the FDA. These are the people I wanted to give credit to. When I started the lab, I actually left Scripps and I took my entire lab, all the people in my lab, all the equipment in my lab, all my grants, and I started Aspen. So I actually moved out from academia into biotech completely with the lab intact. And that turned out to be the best possible idea because I had all these people at this company, the startup company, who had experience on this very project. And I also got credibility because we had grants that I could transfer over there. I also got to move all the equipment, which was extremely cool because it cost $1 million. And so we just essentially, it took us 1 day to go from being an academic lab to being a biotech company. All we did was keep going. So that was really important. And then we've also had support from CERM, from NIH, from the Silverstein Foundation, and from Aspen Neuroscience. So here's my haiku for the future of stem cell therapy for Parkinson's disease. Neurons bloom anew. FDA's blessing ignites hope. Parkinson's may fade. That's what ChatGPT thinks. Now I'm going to tell you the short story of what I've been doing over about the last 5 years, but now it's getting more intense. And this is to study the effect of microgravity on neurons. Now you can imagine if you are an astronaut, they can take blood samples, they can measure your muscle strength, they can look at your bones and see how strong they are, but they cannot get into your brain. So in order to find out what happens to neurons on a space station and presumably farther out in space, we were asked to start this project, which has turned out to be absolutely delightful. We've now had 5 missions, and it is funded through the National Stem Cell Foundation, which is the blue logo there. It's a nonprofit, and New York Stem Cell Foundation is our collaborators. Space Tango and Axiom Space have helped us get the cells up into space. So here's the haiku. Cosmic voyage starts, stem cells defy gravity, life blooms in space, space's embrace. Okay, so cells can make everything. So I just wanted to do a spin on this particular cartoon, which is very popular. You can be anything when you want to be when you grow up, and then I have the cell saying, I want to be an astronaut. So our project was to understand the effect of microgravity on the brain and focusing on neurodegenerative disease. This is the way we designed it. So there are a lot of different kinds of neurons in the brain. I think you know that. Only certain ones seem to be affected by certain diseases. So we chose, just because this is where our expertise was, just to study Parkinson's disease and multiple sclerosis, different parts of the brain. So for multiple sclerosis, we made iPS cells from people who had multiple sclerosis and a control. And we generated cortical neurons from them. For Parkinson's disease, we did exactly the same procedure that we used to transplant to patients. We had a Parkinson's disease patient and we generated dopamine neurons. And this is where the really cool thing came about. We wanted to reproduce a part of the brain. In order to have the brain be have the organoid be sort of brain-like, it needed to have a key component of the brain, which is the brain's immune system, because neuroinflammation underlies a lot of neurodevelopmental and neurodegenerative diseases. So we made 2 separate populations. We made the neurons and we made microglia. We had to do that because microglia come from an entirely different part of the body during embryonic development. They come from a structure called the yolk sac, and they migrate into the developing brain. The neurons develop in the brain, so we couldn't get them both in the same thing. So then we mixed them together to make organoids. And then the secret to our success was that we decided, since we were going to send them up into space, that we should we wanted to design a sealed system. So what we did was, and everybody, this is another thing that nobody thought would work. This is really fun when stuff works. We took each organoid, put it into a cryovial, 1 ml cryovial. Those are sturdy little tubes. We put 1 ml of culture medium in it, sealed it up, set it up space for a month. And I'll show you what happened when we came back. So it takes a long time to get to space. I just want you to appreciate this is not a lab experiment. We prepare the samples and then we have to ship them to the Kennedy Space Center. They get loaded into a device that is approved for putting on the space shuttle. And then there's a launch, and then it docks. All this takes time. And then it gets installed on the ISS, and then a month later, it splashes down and it goes back to the lab and we analyze them. Okay. We've gotten pretty good at this over time. But this is the most important result, I think. First of all, that even though the cells were in those little vials, they came back not only alive, but they came back even healthier than our controls that were on the ground. And one of the things that we discovered from gene expression profiling is that the cells are more mature when they're in space than they are on the ground. Now, go ahead and hypothesize. We're testing my hypothesis right now, so I'm not going to tell you what it is until I find out whether I'm right. But there was less cell division and there was more maturity, which is, a really interesting and very clear-cut result. It didn't matter which cells they were. Both types of neurons do the same thing. So I want to acknowledge the help of these people. This is the group from we call our space team. One of the advantages of this, there are two advantages I'll mention to you. One of them you can see in the lower right, and that is I get to watch the rockets take off from the same place the astronauts' families get to watch. And then the other is that I get to send my own cells up into space. So if you see in the middle there, those little organoids, which you can barely see, those are from my own iPS cells. So we made dopamine neurons from my cells. I'm the control, which is, kind of a cool thing to do and worth 25 years of work on iPS cells. This is from our latest flight, which is an Axiom mission. And this was a private astronaut mission. There were international group. They're going to come down on Sunday. They've come away from, they've undocked from the space station, and now they're doing this orbit, which brings them lower and lower to Earth. And then they'll splash down on Sunday morning. Okay, I'm going to close with my stem cell haiku for the future of stem cells. Stem cells hold the key to healing and longevity. A bright future lies ahead. Thank you.
Our next speaker, everybody is very familiar, Timothy Yu. Many of you already know him, and Julia also mentioned about his contribution in the N-of-1 community. For those of you who don't know, Tim is a physician scientist at Boston Children's Hospital, associate professor at Harvard Medical School. He led a groundbreaking research in autism gene discovery, neonatal care genome sequencing, and therapeutic approaches for pediatric neurogenic disorders. Together with Julia, he developed the world's first individualized drug, Milasen, and co-founded N=1 Collaborative. Our company has been working with Dr. Yu for several N-of-1 projects since 2021. I am always inspired by his passion and dedication to the field. And without further ado, he's for sure, he's my favorite rare disease warrior. So Tim.
Thank you so much, Jeanne Wei, and it's a pleasure to join you virtually here today, especially after such a warm introduction. Thank you for all that you've done for the rare disease community too. Great. Yeah, again, thank you so much for having me. And I'm going to follow that wonderful talk with a few with an overview of work that we've been working on in this new era when you have really remarkable tools available to us for understanding the basis of neurologic disease and now intervening in it. And in particular, the title of my talk is going to be Progress, Challenges, and Solutions in Personalized Therapy. Now, I suspect that with Julia and Amber having preceded me, that some of these concepts I'll describe are not new. But what I hope to do is provide a little bit of an overview of a new area in therapeutic development, which is the development of therapies that might apply to as few as single individuals. And I hope to convince you through this high-level overview why this is important and necessary and not niche, as well as highlighting how far we've come, but what many challenges remain. So to set this up, of course, one has to acknowledge the current landscape of unmet needs in genetic disease, which is that even despite remarkable success defining genetic causes of conditions that afflict us, there are thousands of them. And these thousands of conditions are fragmenting the disease population into very, very small camps, such that most of these individual conditions are incredibly rare, with well less than 1,000 individuals ever identified with the condition. Often for the diseases that are my domain of expertise for neurologic diseases, these are some of the most recently discovered conditions. We're not even close to 1,000 individuals. Maybe these disease populations might number in the tens or dozens or at most hundreds. And when you then layer onto that the number of these conditions that have therapies, 500 FDA-approved therapies that might be applicable in some way, even symptomatic, if not targeted for these conditions, we've got a long way to go. Of course, this is 2024, and we are able now to envision a future when these new genetically targeted therapies that are currently on the scene are able to fulfill their promise. And it's our excitement about these technologies, many of which are still in their early phases, that leads to us the work that I'm going to be describing. But these genetically targeted therapies, gene therapies, mRNA therapies, antisense oligonucleotides, they all share in common the fact that they act via the human genetic code, either at the genomic level, the DNA level, or the transcriptome level, the RNA level. And they can be incredibly precise in which genes they target and what mutations they target, even what base they target. But that precision and that power comes at a price. These are powerful technologies, but because they are working with such specificity, they often, not always, but often require mutation-specific action. Some examples of what I mean. Well, here are three of the most exciting examples of therapeutic revolutions in the last 5 years that we've been privileged to see. We've been privileged to see an antisense oligonucleotide developed with great success for infants with spinal muscular atrophy. And this drug, Spinraza or nusinersen, it happens to work as well as it does because it can be given to thousands of individuals because it targets an evolutionarily acquired mutation that all humans have in the SMN2 mutation, in the SMN2 gene. Other famous success stories in this space include exon skipping strategies for Duchenne's muscular dystrophy as one of the first examples of a precision genetic therapy. These are precise genetically targeted drugs that allow one to skip over mutations if you're lucky enough to have them in one of these particular exons in the Duchenne's gene. Or the most recent FDA-approved success of CRISPR editing strategies for sickle cell disease. They rely on the fact that sickle cell disease is caused by recurrent mutations that are common in the human population, allowing one targeted drug to be used for many, many individuals. The point, however, that we can see beyond these examples is that these opportunities, unfortunately, are the exception, not the rule. And by highlighting that fact, we have to confront the fact that, well, what do you do with these technologies if the mutation target for which that technology may be perfectly suited is only in a small number of individuals, not everybody? What if it's in a vanishingly small number of individuals or even just one? How do you get it developed? How do you regulate it? How do you tell if it works? That's been the problem that's been captivating us the last 5 years. And we began working in the space really because of the story of Mila, a patient who we were lucky to encounter in 2017. And Julia has already talked about her story. And our experience working to first diagnose Mila and then develop a drug for her has really led us to continue to try that strategy for more and more individuals. The theme that I'm describing, of course, is of programmable medicines, the idea that you can use these genetically targeted features of these drugs to flexibly design from some parent template, for instance, the SMA drug nusinersen, additional children drugs that from parent to child that use that same general strategy, even though they might not be exactly the same as their parents. And this type of work now has a place to live in the regulatory framework with the release of the first FDA draft guidances for this type of individualized approach. These are all released now 3 years ago. In January 2021 was the first, and then over the next 12 months, 3 more guidances. And for your reference, multiple examples of this using this approach have now been published from ourselves, from colleagues at the Columbia Medical Center and the University of Massachusetts. And I'm not going to go into detail, but I may refer to a few of these examples. Here are examples of 4 trials that our group has run in the last 5 years, targeting individualized therapies for CLN7 Batten disease, for ataxia telangiectasia, for KCNT1 epilepsy, and an inherited form of blindness, FLVCR1 retinitis pigmentosa. Much of this work has been done actually heavily reliant on IPS technology, as in the talk here before preceding me. And actually, a significant amount of these efforts have been done with the help of Nyanwei, you and your team. This space has really grown. And the number of partners that have now moved in to work on this type of therapy is really something to see. Between the FDA and their engagement and their blessing of the space by creation of these guidelines, to the Oligonucleotide Therapeutic Society that was critical in supporting these early efforts and produced one of the first white papers describing the prospects as well as challenges for the space. And then other groups like NIH beginning to lean in, and many academic and nonprofit colleagues in the green box. Many institutions are now working to support the development of these drugs that are for orphan conditions. I'll in particular call out, you'll hear from the N of 1 Collaborative later, from Winston Yan, the president of the N of 1C and N of 1 Collaborative, a group that Julia and I founded. I'll also call out in particular your local neighbor, the N-Lorem Foundation, that was a nonprofit started by the founder of Ionis, that has been really hard at work developing drugs for dozens of more additional indications, and has currently, I think, 3 of these in clinical trials right now, including this one where the investigator happens to be my boss, Wendy Chung, the new physician-in-chief at Boston Children's Hospital. So I'm going to reflect a bit on the themes of opportunities and challenges in the space. And at a high level, let's talk about the opportunities first. The opportunities for this type of new technology, or actually it's the approach more than the technology. The first and foremost one, of course, is to help patients and families. We're talking these individualized genetic technologies, by being orthogonal to standard traditional approaches that may be, say, gene targeting, these actually may provide an orthogonal approach towards addressing needs of a community that might actually allow folks who can't be served under the current gene-targeted model to be served perhaps under a mutation or individualized targeting model. And the opportunity to try to bring some hope to families of patients, especially with these conditions, which can be very severe, progressive, and fatal, is number one. A critical aspect of these opportunities that is of, I would say, equal but different importance is to learn about these children's conditions. That by definition, since we're targeting, these efforts are targeting conditions that are orphaned, meaning that they're too rare to have received significant scientific or clinical attention. These are generally occurring for diseases in which there has been no prior therapeutic attempt, certainly no successful one. And so these efforts allow us a really unique way to conduct, in a way, pilot experiments or to generate pilot experiences that tell us about whether a disease is even treatable, and how high the ceiling may be, or how high or how low the ceiling may be if a therapeutic intervention were to be applied. And they could also offer really critical information about what types of responses might be achievable. And third, in order for these opportunities to be fully realized to the maximum extent, each of these opportunities represents an opportunity to improve how we do this, how we deliver this type of care. I say that carefully because at some point there's a distinction between research and treatment and care that I'll discuss at some length later on in the challenges section. So if these are the driving opportunities, I have a longer list of challenges that I'd like to discuss. Let's start with this one. The first challenge, the first category of challenges that I like to contemplate are represented by this picture, meaning to represent uncertainties. And I'm going to try to explain this very simply. We're not always sure what we need to do. What do I mean by that? We're not always sure of the genetic mechanism or the genetic strategy which is most appropriate, because by definition, these are conditions for which not a whole lot of attention is perhaps have been paid to in the past. Do we have to rescue splicing, as in the Mealison case? Do we have to boost expression of a gene, as in other cases that I could cite? Do we have to knock down both copies of the gene because perhaps the gene is creating an overactive, overexuberant gene, and reduction of function is going to be a benefit? Or do we have to knock down the mutant copy, right? To illustrate this a little bit more concretely, I'll give an example of a project that we've been working on the last few years. There's a condition that's caused by mutations in a gene known as MAPK8IP3. That's a mouthful. In the literature, it's also known as GIP3 from the prior biochemical and cell biological literature. This is a protein that's been studied for some 10 years, but its disease relevance was only discovered relatively recently, a few years ago, when it was found that heterozygous mutations in this gene were described in individuals who have developmental brain abnormalities. They have malformations of how the brain develops with complicated names like perisylvian polymicrogyria, cerebral atrophy, hypoplasia of the corpus callosum. So these are abnormalities in brain development, and there's also a neurodegenerative component. How this manifests in kids is with weakness and spasticity of the legs, with neurodevelopmental delay and intellectual disability, and sometimes seizures. And in the initial descriptions of this clinical syndrome, there were de novo variants reported in 13 unrelated individuals. 6 of these were missense changes, single amino acid changes. 3 of them were truncating variants, frame shift or nonsense mutations. Interestingly, 3 of the missense variants were recurrent, meaning that they had spontaneously arisen in unrelated individuals with the exact same amino acid change. And for human geneticists, perhaps not obvious to non-geneticists, but for human geneticists, this type of spectrum of mutation pattern is actually a very confusing spectrum. Typically, if you see recurrent mutations occurring where by chance any mutation could arise in this gene, but patients with this condition seem to have the same ones reappearing independently through random mutations in different pedigrees. That suggests that there may be something special about the mutation, that perhaps it's a gain of function mutation that has a specific impact on how the gene works. And that might suggest that this gene needs to be knocked down, either with both copies or at least the mutant copy. On the other hand, the finding that there are mutations that truncate the gene, that are nonsense mutations that simply create non-functional protein, that has the opposite implication. It suggests that it's lack of this gene's function which is critical for the disease. And that would argue that you have to increase the function of this gene to boost its expression or to replace it entirely. And this is just one example of many that I could cite that illustrates that when we take on these projects, that there's really basic biology that has to be figured out first, and such that we bear risk that has to do with the biology of the gene and the disease itself, which may not be fully nailed down. So that's an example of of an uncertainty where we're not always sure what we need to do. Another example of uncertainty is we're not always sure when we need to do it. And by this, I'm talking about interventional window. Does the intervention have to occur before birth? Does it have to occur during infancy? Can you intervene usefully in childhood, in adolescence, or later? And these are questions that often when we go into these exercises, we don't know. Perhaps there's some animal model data, but often there's not. And even if there is, it may not translate well. A really somber example of this, unfortunately, is in fact our very first case, Julia's daughter. As you heard from her, we developed an individualized medicine for her untreatable form of CLN7 Batten disease. We met her at age 6, developed the treatment. She was 6.5. We developed the treatment by around age 7.5 and launched it when she was had just turned 8. And we saw improvements in quality of life and in seizure burden. But eventually, after 3 years, she passed away. The treatment, which was working, failed to arrest the ongoing neurodegeneration in her brain. And that process, which had been preceding the initiation of treatment for some 3 years, slowed a little bit, but it didn't stop. And so it reduced her quality of life such that her family had to make very difficult decisions about her goals of care. Now, this taught us that despite this treatment working, and I should say that the bottom right panel shows that in the really generous gift of autopsy tissue that the family bequeathed upon us at the end of her life, we were able to see that the drug was working. This illustrates the point that when one goes into these efforts, one has to ask hard questions about whether or not the treatment window is still open. And the answer may vary for different diseases hugely, for neurodegenerative diseases, for neurodevelopmental diseases, even within those categories. We don't always know. Another category of challenge that we face, that's this denoted by the scale, the concept of risk-benefit. Here, all cases by definition, because when we are contemplating these types of interventions, they involve very serious conditions. All cases are, of course, urgent, but not every case is urgent in the same way. Here's an example. So here's an example provided by ataxia-telangiectasia. Ataxia-telangiectasia is a cerebellar neurodegenerative condition. It's a disease of progressive incoordination caused by progressive atrophy of the coordination center of the brain, at the base of the brain, the cerebellum. And the natural history of AT is such that severe symptoms don't manifest until age 5. However, we know that patients at birth already have pathology. They already have abnormalities in the way that their cerebellum has developed to a certain extent, and that those abnormalities get worse over time, even though clinical symptoms aren't immediately evident. And so this is a condition where, on one hand, there's a very, very serious consequence of the degeneration that occurs beginning at age 5. These children become wheelchair bound by age 10, and it's typically fatal by young adulthood. But there is a period in early life where those disease processes that cause these symptoms are underway and ramping up, but the patient does not obviously look that sick. And in these instances, early intervention actually from animal data and other experience with other neurodegenerative conditions may be critical. And in fact, the top left slide shows an index case of ataxia-telangiectasia that we've talked about, where we were able to intervene shortly after birth. here's another example of this question of timing, of this question of severity. Here is a child who, with a condition called FLVCR1 associated blindness, and you've likely not heard of this, but this is a child who in infancy was born with a congenital insensitivity to pain. She had multiple injuries to her digits because she couldn't feel the harm that she was doing herself by banging on them or chewing on them or other incidental activities. She also had progressive vision loss beginning at age 2, beginning with loss of night vision and then increasingly restricted tunnel vision. She was diagnosed with a condition called FLVCR1 blindness, and the pain insensitivity in this condition is fixed and irreversible, but the vision loss is progressive and impairing her quality of life. And considering that this is not a fatal condition, but then considering what the impact of this on this child is, in addition to her having sensory problems, she also has difficulty understanding where her joints are in space because of those same sensory neuropathies. And the combination of her deficits plus her vision changes really meant that she relied on her vision a lot for navigating the world. And even though it's not a fatal condition like ataxia-telangiectasia or like Batten disease, this required a different risk-benefit calculus to think about whether or not the ASO treatment might be offered to slow this vision loss. Not only are the severity of her symptoms critically important to consider on an individual basis, but even the risks of treatment are different for different purposes. In this case, treatment of the eye would have required intravitreal injection, or did require intravitreal injection. And there's some risk of cataract here, but it's a very different profile of risks compared to, say, intrathecal treatment for brain diseases. These sorts of considerations were discussed and with the family, with ethicists, with the FDA, and a trial was launched in August 2023. But the take-home lesson is simply that we need to consider risk and reward on an individualized basis, and we need to get used to that. It's hard to come up with one-size-fits-all rules. All right, what's this slide meant to represent? So here we have a path in the woods, a fork in the road, that leads on one side to dark and foreboding woods, and the other side to flowers and light. And that's meant to denote this concept of endpoints. And at what point in a trial are we able to draw conclusions that tell us which path we're on? When do we conclude that a quote-unquote trial is complete? Not necessarily that the treatment is complete, but that the experimental part of the trial is complete. When can we consider it complete because we believe now that it's working? Or when do we need to call it complete because we've decided sadly that it's not working? And these are obviously questions that are traditional clinical trial questions, but they're honed to a incredibly sharp and difficult point for small n trials. The types of signals and the type of work that is done to establish efficacy in larger populations simply don't work if you're talking about very small populations. And here I'll again draw upon the ataxia-telangiectasia example. This is again a condition that's marked by progressive motor incoordination and atrophy of the cerebellum. And so in this case, we chose to use serial MRI imaging as a way to look for hopefully stability of the cerebellum over time, to see whether or not we had been able to forestall shrinkage of the cerebellum over time. As an aside, it looks like it is working in that respect. But then other measures like incoordination are actually notoriously difficult to quantify. As a clinician and parents who know the AT population, you can look at a child and say, they're doing better than this child over here. But to actually put that into numbers is not a trivial thing. In this case, we had to deploy individualized and longitudinally deployable biometrics, biosensors on the wrist and ankle that could measure rates of motor movement, that could give us objective and rigorous data points that we could use to convince us and hopefully others of success. All right, last couple of challenges. So this is about the misnomer of individualized medicines. And the misnomer of which I'm probably primarily guilty for having used this phrase in the beginning is that these are not just Ns of ones, but many of these therapies that are developed under this N of one paradigm will eventually be found to be applied to at least a few individuals, and occasionally many individuals. Now, again, here I'll rely on the example of ataxia-telangiectasia, this mutation-specific treatment for initially a single individual with AT. She was the only one in the world that we knew of with her particular mutation. But we later found additional children, first a second child in Europe 2 years later, and then more recently, 5 additional children in Turkey. And so what do we do when N of one becomes N of many? There are practical considerations and logistical, regulatory, and ethical considerations. At a practical level, like it's difficult to run trials on an individual basis, and then to run an individual trial in another country with another investigator, and then another trial with a different site. At a regulatory level, each of these countries may involve different structures for conducting and overseeing the trial. In some cases, like in Germany, the EMA, the European Medical Agency, is involved. In other cases, in some cases like Turkey, the Turkish regulators are involved. In some cases like Germany, they actually are not, and local hospital ethics boards oversee these. So these are considerations that are at the logistical level. Ethical considerations include if one now is expanding from an N of one to an N of many, are there additional requirements for manufacturing? Are there additional standards that ought to be met for safety testing? And additional burdens on the threshold of proof required before embarking on the larger population. I'll just point out that this is an issue that's cropped up in our splice modulating ASO trials, but it's especially an issue for knockdown ASOs, where it's pretty frequent that a single ASO might be given to larger numbers of individuals. In the J-Suffusin trial, that began with 1 individual with FUS ALS, but then that expanded to 11 and now is a full-fledged trial. And of course, that's a good thing to expand this to more and more individuals, but having a playbook for how to navigate this is critical. A really interesting space where this question is going to come up more and more often is for allele-specific ASOs that target a specific segment of the population that has a particular genetic marker that is the tag that ASO binds to. The KIF1A example that I alluded to earlier is an example of that, where an initial child, Susanna, being treated has offered hope, but also an opportunity, but also logistical requirements to expand this to other children with KIF1A, of which perhaps 5 to 10% might eventually be eligible for that type of, that same drug. My last challenge that I'll close with is illustrated in this slide. And this is the giving tree, of course, except with 2 protagonists, one on the left, one on the right, with different heights of ladders and different abilities to reach fruit, the low-hanging fruit. And that's to evoke this concept of equity, reimbursement, and scaling. How are these patients and these opportunities being identified? This is not a trivial thing to get a genetic diagnosis these days. And then to get a genetic diagnosis, to know the mutation, and then to know who to reach out to declare potential interest and eligibility requires having well-educated families with access to the medical technology and the clinicians who are savvy enough to connect to these research efforts. Who pays for these efforts? Is there sufficient research funding? Is there sufficient philanthropic funding? Are there commercial ways to support this type of work? Generally, the thought is no, that no one has an idea for how this could be sustained at a business level, which of course is necessary if this is going to scale. And here I don't have solutions. I just point to activity in the space to consider these questions. Editorials considering both regulatory and ethical considerations. A lot of think tanks, including our own and the N of 1 Collaborative, thinking about different working models under which this type of work could continue to advance. And on the diagnosis side, I will say that one tide to raise and lift many boats will be if there are ways to institute more logical, earlier, and more equitable genetic diagnosis. And here I just like to point out the newborn genome program that the UK recently launched to sequence 400,000 newborns to hopefully pave the way towards a way where it becomes much easier to identify these types of cases earlier and systematically, not just with who can manage to arrange to have genetic testing. Thank you so much. I hope this overview was interesting. And my thanks to the many folks in the community who are working hard on these problems, and to my collaborators, more direct collaborators and funders shown on this slide.
Thank you so much, Jeanne. Your talk is always inspiring, and yeah, as we all know, there's hundreds of challenges in front of us. Until we really dive into the detail of moving our needles, we never have a solution, right? So really appreciate all your dedication here. Our next speaker, Dr. Don Cleveland from UCSD. I guess I probably don't have to introduce him. He has a lot of contributions across a lot of different disease areas. Many of you probably already know him already, but a couple of weeks ago, I'm trying to get the bio from Dr. Cleveland's assistant, and I received a PDF file. Oh my God, 61 pages. I really don't know how to summarize his contribution, but I'm going to try. He is the elected member of National Academy of Science, American Academy of Arts and Sciences, National Academy of Medicine, American Academy of Microbiology. And Dr. Cleveland is the 2018 Breakthrough Prize recipient in Life Sciences for his pioneering research in neurodegenerative diseases. His achievement includes the groundbreaking work on ALS and Huntington's disease, innovative gene silencing therapies, AAV-mediated treatment, and pioneering the development of synthetic CRISPR RNA. So our company, iXCells, has been collaborating with his lab to develop ALS cell models using iPSC-Derived motor neurons since 2017. So I'm going to stop here, and Dr. Cleveland.
For the people in the room, for those in the room, the bottom of the slides are a little cut off, but the people online, they're perfect. So I was trying to solve that, but I failed. Okay, so yeah, I'd like to describe today. We're a little out of order, but I'd like to describe the birth of the use of these designer DNA drugs, these antisense oligonucleotides, in neurodegenerative disease. And our strategy or our impetus began from the recognition from human genetics that of the major diseases of the aging nervous system, Alzheimer's, Parkinson's, ALS, Huntington's, that in almost every case, the gene, where there are genes that are known to be causative or contributory, they're expressed broadly throughout the cells of the nervous system, not uniquely within individual classes of neurons whose dysfunction gives rise to the phenotype. And even more so was the discovery by two genetic teams that if you inherit 3 copies of one gene, the initial founding example was from Andy Singleton and his colleagues at NIH, that if you inherit 3 perfectly normal copies of the gene encoding alpha-synuclein, you will develop Parkinson's disease. So too much of a good thing. And that was followed by the discovery that 3 copies, no mutations, 3 copies of an unmutated amyloid precursor protein gene, you will develop Alzheimer's disease. So we could say broadly that, or at least in those examples, and we know that alpha-synuclein misaccumulates in deposits throughout almost all Parkinson patients, and a piece of amyloid precursor protein accumulates in Alzheimer's disease. So we thought, oh, the therapy, why don't you just turn down the production? Since at 150% the level of a normal protein is enough to drive almost certain disease, actually early onset disease. So let's just turn it down. And so we thought, could we develop designer DNA drugs that could do that? And the strategy for the scientific ones, the strategy, every young person, if I look out in the audience, every young person would say, oh, let's use an siRNA. But no, we use the older methodology, a single-stranded modified DNA oligonucleotide, which if you can get it inside the target cell, it can then pair with the target. Let's see. It can pair with the intermediate copied from a gene, an RNA, and then make an RNA-DNA hybrid for which we have, which kind of it looks like a viral intermediate to one enzyme that actually sits in the nucleus of all of our cells, RNase H, which thinks that it's a viral intermediate and that enzyme destroys the RNA, but not the oligonucleotide, the DNA. And so, oh, it's catalytic. It now, a single molecule could go find another RNA, another RNA, another RNA. And the problem, and in Timothy Yu's talk, he mentioned part of this, and the problem is that actually, how do you get the drugs delivered? And if you deliver them any way known today in the periphery, zero gets into the nervous system. Okay, so we're going to put it directly into the fluid that bathes the brain and the spinal cord. And there, if you do that, today it seems like everybody says, well, just use an ASO. But at the time, this is 2004, at the time, everybody knew this couldn't work. These drugs are 15 times too big. They're very heavily negatively charged. Every textbook said these can't work and be delivered broadly in the nervous system. And thank you for the introduction. Yes, I did win this big prize, the breakthrough prize. And the breakthrough was we tried it anyway. Okay, it turns out the cells of the nervous system didn't read the textbooks. And now, now we do realize big W, and I should have, it was on slide one. I'm sorry, I failed to point that out. I am a consultant for Ionis Pharmaceuticals. And we started with Ionis in 2004. And I own not one share of Ionis stock. Okay. Now, we now know and have demonstrated that the team has demonstrated that we can deliver these drugs when put into the cerebral spinal fluid, the fluid that bathes the brain and the spinal cord, that you can deliver them very broadly. Perfectly, almost perfectly in rodents. Oh, but you don't care that much. But in non-human primates, you can do it very broadly, maybe not to the deepest structures in the brain. And so there will definitely be some limitations. And we're anticipating there are going to be improvements in that delivery approach. But today, what can you do? All right, so, and the disease we started with is amyotrophic lateral sclerosis, or ALS. The salient, and I know it's not a neurology audience, but the salient feature is the premature degeneration and death of the neurons who hook up to the muscles and trigger their contraction. And typically a disease, I used to say it was a late onset disease, but now I realize it's an early midlife disease, since I'm over that age. Anyway, and 10% of the cases are clearly inherited from a mutation. And the founding mutation, it was here in this paper in 1993, discovery of mutations in a gene called superoxide dismutase that were causative. They're causative of about 10% of the inherited cases of ALS. And here, this is the paper in Nature, and here's the News and Views article for the scientists. The News and Views article in the same issue, which declared that these mutations, the gene encodes an enzyme, and it was the proposal was that it was loss of function, that it reduced function because it had mutations in it. And yeah, ALS frequently called Lou Gehrig's disease. And so we tested that. we made mice. We tested one of the mutations and found it to be absolutely have zero activity as a dismutase. So we thought, okay. Then we expressed that in a mouse, but the mouse has 2 endogenous alleles. And then we removed one of the endogenous alleles or both. So we have mice that have an inactive mutant gene and normal dismutase activity, half or 0. And you don't have to pay attention to which color is which because they're identical. There was no change in disease course in animals, fatal paralytic disease from lowering enzymatic activity. And that, after a contentious period, led to what is now, I think, a universally held view that these mutations provoke disease through the acquisition of a mutant property of the damaged protein, not through loss of activity. And then, so we thought, okay, so let's try to use an antisense oligonucleotide. I hope we can start calling designer DNA drug. Little snippets of DNA with short pieces, relatively easy to synthesize, and put them into the cerebrospinal fluid and ask, can you lower that, can you target degradation of the RNA encoding the mutant gene? And the answer is, well, here's the level. If you mock treat them with just saline, oh, look, we can lower it. Not only did lower where we infuse the drug, but we could lower it throughout the entirety of the nervous system of a rodent. And then this is a rodent that has a hyper-aggressive disease because it expresses a lot of this mutant gene product. And so we dose them at disease onset. Not before, but at disease onset. And this is the founding use of an ASO in a therapeutic strategy for one of these human neurodegenerative diseases. And we saw that here, if you look for how long do the animals live after onset, and the answer is, well, it's hyper aggressive. I said that. They only live about 25 days. But when we dosed them, we doubled survival after onset. And so we thought we have been able to achieve it, even in really hyper aggressive form of disease. And just last year, the human trial went through the initial safety trial, went to phase 2 trial, to phase 3 trial, and the outcome of the phase 3 trial was announced last year. And here you go. Here, the data from the phase 3 trial were here published released in 2022, and we saw that here's in the green bar are the treated patients, and the dashed blue line are the placebo patients from that trial. And the trial was just too short, so it was a 6-month trial, and the trial missed the initial clinical benchmark. But then, in the open label extension, where almost 100% of the patients continued and were continued to be dosed, look what happened. This is a muscle strength measure. There were a bunch of these clinical measures, and disease course flattened. And so far, it hasn't quite been reported yet, but at Washington University in St. Louis, where they have a big practice, in their practice of the patients in the open label extension, 1 year after starting dosing, 25% of their patients are stronger. This is just so we have benefit, and indeed FDA approved it in April last year. Okay, so and Jeanne Loring introduced Parkinson's at the beginning of this session. Parkinson's disease is, there is a genetic component of Parkinson's, and the strongest genetic component are mutations in a gene called LRRK2. It's an enzyme, it's a kinase, and the mutations are kind of unusual because they activate the kinase, not inactivate it. And and the world's most prominent at-risk individual is Sergey Brin. This is quite public knowledge. His mother is a homozygous for that mutation, and he's heterozygous, so he's likely to develop Parkinson's disease because he has this mutation. And this is me getting the breakthrough prize presented by Sergey Brin. And indeed, we have, working with the Ionos team has designed an ASO to turn down the LRRK2 gene, and that now is in trial. So I wished I'd known that when he handed me the prize, but I didn't know he had the mutation at that time. Yeah. Okay, so it's, all right, so, and now I mentioned before that the discovery that AAA, 3 copies of a normal alpha-synuclein gene is sufficient to be causative of Parkinson's disease. So just turn it down. And that could then, in addition to the kinds of cell therapy that Dr. Loring talked about, just turn it down. It could be good. And indeed, that is now ASO trial, not quite in Parkinson's disease, but in a slightly related disease called multiple system atrophy, which is caused by mutations in alpha-synuclein. That's now in trial. That'll be the safety trial for a Parkinson effort, and we just can't wait for the outcome of that trial. Okay. And then the biggest of the neurogenetic diseases, Alzheimer's disease, there are 2 major pathologic events in the neurons at risk in that disease. One is the accumulation outside those neurons of a piece of the amyloid precursor protein called A-beta. It accumulates outside, and it has been the target of almost all pharmaceutical company efforts, especially for, I mean, who could not have seen that there were a flurry of efforts using monoclonal antibodies to try to clear extracellular A-beta. But there's also a protein that accumulates inside the affected neurons, and the name of that protein is tau. And tau was discovered, this is now, we're at the 46th anniversary, discovered by a young graduate student. And for the scientific community, yes, we actually did show that it was a natively unfolded protein. It's one of the first of the proteins to be demonstrated to have the properties of a natively unfolded protein. We just didn't coin that phrase. Okay, so, but in the Alzheimer's community, I think there's a broad consensus that the best predictor of cognitive decline is not the accumulation of A-beta containing plaques. And if we had well, this is a pretty young audience I'm looking at here, but if you had a somewhat older audience, there would be individuals who have plaques who are cognitively normal, but there are almost no one who has tau tangles who is cognitively normal. All right, so you could say, and I just want to show you the outcome of trying to target tau, just turn the tau gene down. And so the phase 1 trial was published, you can see, last April. And as you might expect, you can, in a dose-dependent manner, as you go to higher doses, you can lower the level of tau. Well, measured by spilled into this fluid, spilled because of neuronal damage. And some think it total tau or a variant of tau that's been modified, phosphorylated tau. You'd lower both of them. So that's good. But the best is what came last October. So there is an imaging agent, PET imaging for the scientists, the PET imaging that measures some aspects, some kinds of this aggregated tau. And here are 2 patients from that trial. It's an Ionis Biogen trial. And you can see at the entry to the trial, there's the scan to look for this tau deposits. And look, this patient has tau deposits, both sides. The second individual has tau deposits, and they were in the placebo arm of the trial. So 6 months later, 6 months later, look, the tau deposits on this side, they're bigger. So I think I animated it. Yeah, and the tau deposits on this side of the second patient, they're bigger. These are called mild cognitive impairment, the earliest phase of Alzheimer's disease. That intentionally, because you want to be able to intervene before too much damage has occurred. And then those patients, those 2 individuals, plus 12 others, were converted into the drug arm of the trial. And 1 year after being in the drug arm, the tau deposits are strikingly diminished. And that was true in all but one of the 14 examples that were published in this paper in just last October. So I think it's this is not evidence this is going to be effective, but it's really about as for the imaging, it's about as positive an outcome as one could imagine. And so, okay, so I'll just one more example, then I'd like to summarize where we are as a field. And back to ALS, the discovery of Virginia Lee and her colleagues, Virginia Lee, John Trojanowski at University of Pennsylvania, and their colleagues, they discovered here, they discovered in 2006, but I took this image from the 2008 paper, and that cells in the brain that with individuals who have a dementia, it's called frontal temporal dementia, and those individuals, many of those individuals also develop ALS, that you look at the affected neurons and you see one protein should be intranuclear. Here, this brown stain. But in affected ones, look, there's clearing from the nucleus, and it's a huge aggregate of it in what is the cytoplasm. And the name of the protein is TDP-43. It's an RNA binding protein. And we now recognize, I think a consensus view is of almost 100%. Almost 100% of ALS has this loss of this protein from the nucleus and its aggregation to the cytoplasm. And about half of the second most frequent dementia, frontal temporal dementia. Actually, it's very broadly true in neurodegenerative disease. All the big diseases, there is loss of TDP-43, at least in some of the neurons. And we demonstrated that actually, so if you lose this RNA binding protein, what happens to, it works on, it was thought at that time to function in the maturation of many different RNAs. And we had, along with the next speaker, Jeanne Loring, shown in 2011, that 1,500 genes, RNAs from 1,500 genes change in abundance or in splicing pattern when you lower TDP-43. We missed the most important one because we did that in mice. We did it in real mice in the real mouse nervous system, but if you do it in human neurons, the gene most affected is one encoding the protein called Stathmin-2. And it's abundant, and as we later showed, it's a gene product that is required in the examples that have been tested for recovery from an initial, neuronal recovery from an initial injury. Okay, so here it is, and we know molecularly what happens. We know that the TDP-43 binds here in the pre-mRNA from the Stathmin-2 gene, and it gives rise to a cryptic splice and use of a cryptic splice site and a cryptic polyadenylation site. And you can see here at the protein level, here's TDP-43. We've suppressed it to about 25% of its original level, and Stathmin protein just disappears. And we know, and again, we demonstrated again for the neurodegenerative disease scientists, you'll realize that within the last 2 weeks, there have been 5 papers that have discovered that there is cryptic polyadenylation when you lower TDP-43, but we showed that in 2019. And here you can really see that if you do. I know that the technical part, the typical RNA-seq experiment, we see exon 5, 4, 3, 2 with a 3' bias at typical. But in ALS, after you suppress TDP-43, man, you get that cryptic splice and polyadenylation site very efficiently. And it's really because TDP-43 binds right here, right adjacent to the cryptic splice site and the cryptic polyadenylation site. But even more so in ALS patients, if you recover the surviving neurons at autopsy from patients who died from this disease, and you ask, did they have a Stathmin suppressed? Well, if you do that, if you recover neurons from individuals who died but not from neurologic disease, there you see exon 5, exon 4, exon 3, just as you'd seen before. But in ALS patients, oh no, almost complete loss of the exons coding for the actual protein. It's almost complete null in those patients, and that was true in using autopsy samples provided by my colleague John Ravitz at UCSD, and it was true in 13 out of 13, all 13 patients that we tested, all of which are sporadic ALS patients with no known genetic component. Okay, so but does the loss of Stathman, does it really matter? And so what we did, and the we is her, for those in the room, is Yoni Lopez-Raskin right there. So what we did was to transiently, in a normal aging adult animal, transiently suppress the synthesis of Stathman, or chronically, beginning at 1 year of age. And when you did that, if you look at the light blue stuff is really good. Here you can measure the innervation of a muscle, and that's what you see here. See the modeled appearance. It's innervated, and if it's oh, I see. Sorry, you're right. I'm looking. Thank you for that. I'm looking at a different screen, so it wasn't on my screen. Okay, there you go. So yeah, so here in the light blue is good. Those are 100% innervated muscles. But after 8 weeks of suppressing Stathman, oh, look, you get the bad stuff. You get partially innervated or fully denervated muscles. And if you do it chronically, it gets worse. So in fact, the animals developed the early stages of ALS-like disease from simply suppressing Stathman. So we argue on that basis that it's a pretty good target. It's not good, it won't be perfect, but we should not let the perfect be the enemy of the good. So, and now, how are we going to fix it? And so we're going to fix it using the spinal muscular atrophy example, the first approved use of an ASO in the human nervous system. And for spinal muscular atrophy, all the kids, it's a fatal disease, typically in the first year or two. Typically in the first year of life, the kids can't develop muscle strength. They can't lift their head. Even SMA clinicians, you can bring them to tears very easily because they've seen so many disappointing outcomes. And here, all those patients have both copies of the SMN1 gene are inactivated. But we, almost alone among the primates, have a second copy, the SMN2 gene. But it has a 1 nucleotide mistake in it, and it's almost completely misspliced to make a non-functional RNA that can't encode the protein. If it did, if it were spliced correctly, it makes the perfect protein. All right, so the strategy was to use a modified antisense oligonucleotide, which would bind to the pre-mRNA precursor, but not be recognized by RNase-H, and not be cleaved, but convince the splicing machinery to make the thing, to process it correctly. And here's what happens. In the original samples where the SMN1 gene is inactivated, and you look at what comes from the SMN2 gene, it's mostly missing exon 7, completely non-functional. But if you add one of these ASOs, you can now make it almost completely correctly spliced and restore synthesis of the protein. And that's the basis for the success, the initial success in spinal muscular atrophy, and to the breakthrough prize in 2019 for Frank Bennett, the chief scientific officer of Ionis, and Adrian Craner at Cold Spring Harbor in New York, who an academic corporate partnership to develop that. And it's changing course of disease. This is actually the first example that a human neurodegenerative disease can be treated. So, okay, so, and we did nothing, my team did nothing on that except activate Ionis and demonstrate that this could actually work. We'll come back to one more comment. So, okay, so what do we want to do now? And the answer is, we want to be able to determine by taking a group of antisense oligonucleotides that are scaling across here and asking, can we find ones in which we can restore the synthesis of the correct processing of the Stathman RNA, even though TDP-43 is still lost? And what you can see here is, we found We found the blue one suppresses the cryptic splicing and polyadenylation in a dose-dependent manner. So does the red one. But even more importantly, we found the ones that restore the synthesis of the full-length product in both the blue case and the red case. And so we argue from this that this really is a good target if, in fact, it can really help the neurons. And to do that, we've tested using those induced pluripotent stem cell-derived neurons that Jeanne introduced earlier. You can grow them. They have their cell bodies here. They grow their axons across this ruled array where the cell bodies are too big to get across. And they have their axons and their distal processes in the distal compartment. We're going to just injure them, remove them, mechanically remove them, and ask, can they recover? And so the answer is, well, first answer is, oh, Stathman is a prominent product and all along the full length of these axons. And it actually accumulates at the terminal of these neurons as well. And now, if you do that now in the actual experiment, we're going to look and see we cut off those distal parts of those neurons, let them regrow for a day. Oh, and if you add a non-targeting antisense oligonucleotide, yep, they regrow beautifully and they restore and Stathman is present. If you lower TDP-43 in those neurons, which is what happens in the ALS patients, oh, they don't recover at all. I mean, they don't recover. But if you then add yet another antisense oligonucleotide to restore, you're still suppressing TDP-43, but you're restoring Stathman. Ooh, they regrow almost as well as the normal ones. So we argue that yes, this can really affect neuronal ability of neurons to survive another insult. And the other insult, what's that? Oh, it's the other gene products that are lost or misregulated because you lost TDP-43. Okay, so we argue that now this really does set the stage for an ASO therapy for sporadic ALS, the bulk of ALS. And indeed, the scorecard on this, there was a pair of teams, mine and Kevin Egan's, then at Harvard, who discovered Stathman as the potential target in 2019. And we achieved restoration in the mammalian nervous system by humanizing the Stathman gene in mice. We achieved that last year. And the Egan team went to trial in 2023 with an ASO that they had developed, and we're hoping that Ionis will go to trial. I am not a representative of Ionis. I can make no statements about what they're going to do. But we're hoping that Ionis will go to trial this year. Okay, so here's the final scorecard. So where we were from 2004, the first demonstration that could change disease course, 2006. We know we can use these antisense oligonucleotides and can deliver them broadly throughout the nervous system of rodents, non-human primates, neurons, non-neurons, all the cells of the nervous system that have been targeted, that have been tested, take up these ASOs in an effective way. You can use them to degrade an RNA or to alter the splicing of it. And I forgot to mention one of the amazing things about these ASOs is that when you take an aspirin, you get 3 or 4 hours of relief. And here we were going to use continuous flow pumps until we've completely failed in a negotiation with Medtronic, which has almost a monopoly on the market for human continuous flow pumps. And then I got the big team together, 3 of us. And one of us said, well, why don't we just inject them in and see how long it lasts? And because we just couldn't imagine that we actually, instead of getting 3 or 4 hours, instead of getting 3 or 4 days, instead of getting 3 or 4 weeks, we get 3 or 4 months of efficacy from single injections. And so then we realized we don't need pumps. All right, so and there have been about 10 trials that Ionis has taken forward, and in almost every case they've been declared to be safe. There's one exception. And the founding approval in spinal muscular atrophy, there are 5 indications that went forward in ALS. And actually, Timothy, you mentioned one of them, and the biggest genetic cause of ALS is a nucleotide expansion in the C9orf72 gene. That trial failed. We lowered the product, no clinical benefit. And there remains an intra-team disagreement about why did it fail. All right, but and there's efforts in Huntington's disease. That too, its initial trial failed, but there's a second one with treating patients much earlier in disease, and Roche is running that. We're hopeful. I mean, the scorecard overall, we're 2 for 4 so far, which in the pharma world is actually pretty good. But there are failures, and we don't quite understand them. There's the tau lowering trial that I mentioned in Alzheimer's disease, and there are the two in Parkinson's disease. Okay, so, and just really to close, here's the founding team. The use of an ASO in the nervous system is Tim Miller, who was then a postdoc with me, now has co-run the clinical trials for SOD1 in ALS. Richard Smith, a local neurologist, who was absolutely adamant that we attempt this. My mouse colony coordinator, a surgeon who was responsible for almost all the surgical things that in rodents, Melissa Macalonis, and Frank Bennett, the chief scientific officer of Ionis. And then finally, the Staffman team for this final therapy that I'm proposing could be broadly used in ALS. Oh, that's Yoni Lopez-Oraskin, who's right back there. She and another postdoc, Zivik Melamed, now in faculty in Israeli University. My 2 then graduate students, now postdoc, Michael Bond and Melinda Bakari. That's the team, and I should a big call out to follow Tim Yu about Neil Schneider in developing the strategy for FUS. Those ASOs were actually designed because Sandrine DeCruz and my team had insisted that Ionis do it. So those are actually Ionis designed ASOs that were used to target FUS. Okay, well, thank you very much for your attention.
Thank you, Dr. Freeman. I was also, a couple of weeks ago, my team sent me some new publication about Desmond Kill. I saw, I know it. But it's amazing to see how we can really translate those early discovery into the therapeutics, right? So let's continue. Our next speaker, Jeanne Yu. He's supposed to be here, but he has some health concerns, so he will present his work remotely. Hi, Jeanne.
Can you hear me? Can you hear me?
Yeah, we can hear you.
Okay.
So Jeanne Yu is the professor of cellular molecular medicine at UCSD, serving as the director for the UCSD Center for RNA Technology and Therapeutics. Chief Scientific Advisor for Sanford Laboratory for Innovative Medicine and founding member of the Institute for Genomic Medicine. His pioneering research focuses on RNA processing regulation and the role of RNA binding proteins in developmental processes and diseases. Dr. Yu has over 250 peer-reviewed publications, including impactful contributions to neurodegeneration, computational biology, and stem cells models. Dr. Yu's impact tends to therapeutics with significant contribution to RNA binding therapy for neurologic disorders and cancer. And he has developed stem cell-based models for disorders such as myotonic dystrophy, Huntington's disease, ALS, and FTD. So, Jeanne, the stage is yours now.
Okay, great. Anyway, thanks for the introduction. I'm sorry I couldn't be there. I caught a bad bug on the way back from UPenn. So today, what I thought I'll do is it's going to be hard to follow Don's amazing talk, but I'm going to try to be a bit somewhat controversial since the workshop is really about iPS-derived models for disease correction, and so here's how I want to think about this, right? So, but just a bit of intro. So my lab works on primarily RNA biology, And just to remind everybody, including folks that don't think about RNA biology, that RNA is never naked. This is work from one of my students who draws for the New Yorker, where the RNA molecule there is always chaperoned by RNA binding proteins, including the RNA binding protein TDP-43. And so we are very intrigued about RNA biology as the basis of, or dysregulation as the basis for many diseases. And so in my group, we really study how proteins affect RNA regulation and how the dysregulation of RNA binding proteins give rise to human diseases, right? But we spent quite a bit of time developing genomics technologies and also disease modeling, leveraging iPS technologies and other technologies to give rise to the novel therapeutic paradigms. So what I thought I'll do though is, since I knew I was following on Don's talk, right, is to talk about how we've been modeling ALS and using iPS lines, and then how we're thinking maybe that's one way of doing that, but there might be other ways of modeling the disease. And so as Don already mentioned, the majority of ALS patients have cytoplasmic, right, inclusions of this RNA binding protein TDP-43. Normally, TDP-43 is in the nucleus of healthy neurons, is the idea, and that it mislocalizes over time in the conditions in ALS backgrounds, right, or sporadic ALS, right, as well. And so we've been very interested in thinking about how we model the disease. And of course, Don had already mentioned his work and Kevin Egan's work showing that that when nuclear TDP-43 mislocalizes to the cytoplasm, you have the loss of TDP-43 in the nucleus from its nuclear binding targets, and that leads to de-repression of cryptic exon statement 2, right? And so we'll come back to this as I think a good marker for loss of nuclear TDP-43. Of course, there are now many other exons that depend on TDP-43, including many non-cryptic exons. And so that makes, as Don may have not pointed out, but that makes knocking down TDP-43 a terrible therapeutic strategy. And so therefore other strategies like statement 2 correction is a viable one. And so in our group, we've been very interested in understanding the interplay between the environment, stress, genetics, and aging. And so in the past, we've been, for more than a decade now, leveraging iPS-derived motor neurons, right, to study ALS, right? And so the unperturbed, non-modified form of iPS-derived motor neurons only have nuclear levels of TDP-43. And so that's sort of true for wild type, but also true for the ALS, TDP-43 mutant or sporadic ALS samples. And so we've been thinking about how do you then model what you hopefully see in patients, right, with different factors. So I'll talk about two factors today. One is the environmental factors, and the other one is aging. And so, so the first one is, can we add environmental factors to iPS-derived motor neurons to try to mimic some of the features of ALS dependent cellular perturbations, right? And so one of these environmental stimuli is very simple. You stress the cells, right? And so one way of stressing cells is to use reagents that affect translation, mRNA translation, or ER stress, or affect the integrated stress response, right? And so the consequence of many of these stressors at the end is ERF2 alpha phosphorylation change. And that change leads to the formation of these protein RNA aggregates in the cell. And there are many protein RNA aggregates. There are ones which are GGBP1, which is another RBP dependent. And there are ones which are also TDP-43 dependent. And there are other ones which are, related to many other RNA binding proteins. So this is a cartoon from Roy Parker's lab showing that if you stress cells with arsenite, you form these puncta in the cytoplasm. There are GGBP1 positive, normally it's diffuse, but when you stress the cells, they react by aggregating these things. And this is a dynamic reversible process. And you do see that mutations in many genes can lead to increase or decreases of clearance of these different RNA granules. And so we propose a model where these granules may be a useful system where we can study the effect of environmental insults to iPS neurons, normally we expect that this protein RNA aggregates will be transient and dissipate over time in a wild-type condition. But in ALS mutations, as we know, that may lead to aberrant aggregation. And some of them are GGBP1 dependent and some are not. And so again, just to remind everybody, including TDP-43, many other RNA binding proteins are mutated in various forms of ALS. And many of these mutations occur in the intrinsically disordered regions of the protein in green that changes the stickiness of the protein to itself or to other RNA binding proteins. And so a couple of years ago, Sebastian, Mark Miller, and my group tried to leverage iPS mononeurons and stress conditions to see whether we can recapitulate disease-relevant phenotypes, right? So again, if you take a wild type and an ALS iPS-derived mononeuron, you don't really see differences. TDP-43 is in the nucleus, and you don't, see the loss of stathmin 2 gene, a protein, and all these other features, right? Which makes sense because ALS is because iPS lines are a good model for embryonic development, but patients kids don't get ALS when they're young, they get to a certain age and then they get ALS, right? So the stress conditions here, a low dose pyromycin stress, or the translational stress, seem to lead to. Sorry, is there an issue with the sound on the slides, or okay?
Not good. Okay, good.
Oh, good. Okay. So pyromycin stress leads to TDP-43 to accumulate in the cytoplasm when it's treated in the cell over time. And also the prolonged stress leads to altered TDP-43 binding to the stathmin 2 cryptic exon, right? Because TDP-43 is in the cytoplasm. It leads to a lack of recovery of RNA localization across many thousands of RNAs in the cell. And when you add stresses to ALS lines, you get exacerbated cell death. But the same stress added to a wild-type line, healthy line, you get some cell death, but it's definitely not at the same degree, right? So we think that it amplifies the genetic vulnerabilities is amplified by the stressor to lead to delayed onset cell death, but in an increased manner. And so then we got very interested in saying, well, since this paradigm is helpful in modeling the disease, perhaps we can try to identify other targets around these granules that may stabilize or change the RNA metabolism of the cell as novel ways to modify disease, right? And so to begin to do that, the first, again, very basic experiment is what is in these RNA granules upon stress. And so what Sebastian did was use APEX labeling, which is a proximity labeling protocol, where you treat cells with hydrogen peroxide and biotin phenol. And in the cell, you have a knock-in line with using CRISPR-Cas9 technology to knock in the APEX2 enzyme, which is a proximity labeler. And in the presence of the hydrogen peroxide, you get activated biotin phenol that activates about a 1 to 10 nanometer radius around the protein that you've tagged. So in this case, GGBP1, this is a stress granule protein. And others have now done this in other labs with TDP-43 and FUS and other ALS-related, more relevant proteins. And what we can see here is that you get labeling of all the proteins in close proximity. That means that the proteins doesn't have to be directly bound to the protein of interest to come down, right? As long as they're nearby. You don't have post-lysis reassociation. You don't get proteins that falsely sediment by centrifugation. You recover these proteins using streptavidin, followed by MESPEC. And so we found actually that we discovered the human compendium of proteins for the first time, right, in these RNA granules. And we found that many of these protein constituents are already pre-assembled on the mRNA and are just amplified in its aggregation upon stress, which makes sense because ribosomes run off. And for the first time in the cytoplasm, the RNAs are more naked, but then RBPs can bind and cover these RNA molecules, right? So we found that many of these RNA binding proteins, we've discovered that if you deplete some of these stress granule proteins, which are also RNA binding proteins, you can reduce this formation of granules in the cell. And so we've been trying to narrow down or filter down candidates, right, by this approach. So we took the list for about 300 genes, and we've been working with folks initially at Biogen, now at Christy Wharton at Brown University, to leverage fly models to study how knockdown of some of these granule-associated proteins can recover ALS relevant neurodegenerative phenotypes, right? So I won't cover some of that work that Christy and her student Anchi Zhao has done, but we at the end led to phenotypic rescue. That means if you knock down these 4 genes in these fly models of ALS where you overexpress TDP-43 that is mutated, or other proteins, FUS or C9orf72, which is another ALS, like the G4C2 repeat gene, you seem to phenotypically rescue these neurodegenerative diseases, right? And just to point out, one of them we're very excited about, because this one, when we knock down O2D4, again, don't have the time to go into what the gene is doing, but when you knock this gene down, stathmin-2 levels go up, right? And so that is, we've discovered, well, I'll show you in the next few slides, the mechanism behind that to some extent. So as I showed you, if you knock down O2D4, you have increases in stathmin-2, and is this coming from the protein level or the RNA level? And it turns out that it is from a proper repression, right, of the cryptic exon. So you here on the left, you see full-length stathmin-2 mRNA. If you, in these iPS-derived mononeurons, knock down O2D4, you get recovery of the full length. It's because you get a reduction in the truncated or the cryptic exon form of stathmin-2. And so one way, an artificial way, so this is a non-stressor way, but just to get around sometimes the arguments about you've stressed the cell, right? Well, maybe another way to do this is you force TDP-43 itself into the cytoplasm by expressing a lentivirus carrying mutant nuclear localization signal operating frame of TDP-43. As you can see, that forces TDP-43 in the cytoplasm, and so that should get you loss of nuclear TDP-43, and therefore you get depression of cryptic exon there, and then in stathmin-2, and then you get a truncated form. In the context where you do this with the mutant NLS, you see increases in the cryptic exon, right? Because TDP-43 is forced in the cytoplasm. And this is with just scrambled knockdowns. But with O2D4 knockdown, you actually restore stathmin-2 splicing, even in the context of overexpressing or cytoplasmically localized TDP-43. So we're excited. There are a couple of different mechanisms that we're trying to explore. Maybe O2D4 plays a role in stabilizing the aggregated form of TDP-43 in the cytoplasm, maybe depleting that recovers that. We have obviously another possible model is where it maybe directly controls splicing of stathmin-2 by enhancing it. We actually see this for many TDP-43 targets. So maybe there's a more global role that O2D4 can rescue more TDP-43 targets than just stathmin-2. So this is one approach in trying to get to novel therapeutic targets in neurodegeneration. We are working with Denali Therapeutics, John Ravitz at UCSD, and Eric Bennett to develop blood-brain penetrant oligos for O2D4 that you can deliver in the blood to get into the brain to hopefully have impact here. So this is one way we're using iPS-derived systems where we stress the cells to elicit disease phenotypes to get us to novel targets. But the other way is to say, well, maybe the iPS system isn't the right system to model age-dependent disease, right? And so is there a way to add the aging component here? And Kevin Reina, a postdoc in the lab, has been evaluating a different strategy. And so the strategy here is to leverage the idea that we know that perhaps in aging, you have many other RNA biology defects that happen. And so maybe the cells are much more poised to synergize with genetic mutations that therefore lead to neurodegeneration at some age, right? And so he leveraged the transdifferentiation system. So he took fibroblasts from patients, wild type in this case for now, also about ALS patients I don't have time to talk about, and you just transdifferentiate these fibroblasts directly into neurons, right? And same for you obviously can do the same. You reprogram, like I already talked about, iPS lines, and then you normally differentiate these into iPS-derived neurons. So let's call this an old, in quotes, and fetal, in quotes, neuronal system. You get relevant, similar neuronal cell types, electrophysiologically active ones. And as a reminder, when you reprogram cells from fibroblasts, you wipe out the epigenetic state of the genome, so the cells are now back to a say a embryonic-like stage, right? And so on the left, you see an epigenetic clock from bisulfite or methylation analyses across the genome, and that's absolutely true. The iPS-derived neurons are basically babies, right? But when you transdifferentiate the neuron from fibroblasts, they retain the epigenetic marks, which is the age represented by the patient that has those fibroblasts, right? And indeed, these transdifferentiated neurons are already stressed, or they have markers of senescence. They have apoptotic markers different from the iPS-derived neurons. And just, for our interests, right? Turns out that there's a global change in RNA metabolism, right? In the transdifferentiated neurons, you see that the spliceosome is very different, right? Using proteomics efforts. In fact, they're much more enriched. And then you see sort of differences in the downregulated, and then you see that many of these normally are higher in the iPS-derived neurons from the transdifferentiated neurons. And TDP-43 is enriched in the cytoplasm, not in the nucleus anymore, right? So this is quite exciting because we've had spent a decade trying to see whether stressing iPS-derived neurons can get you TDP-43 in the cytoplasm. Here, we think that the epigenetic landscape of aging gets you TDP-43 in the cytoplasm kind of for free, but including many of these spliceosomal proteins. So these are RNA binding proteins. And so basically RNA splicing and RNA probably polynylation, because polynylation is related to splicing as well. They're already messed up, right, in the transdifferentiated neurons. And we know that TDP-43 binds to GU-rich motif. These are things that we've done. We had looked at in mouse very early on in 2011, right, 2012, right? We see that the motifs are not different between the transdifferentiated neurons or iPS-derived neurons, but the binding patterns are very different. So in the iPS-derived neurons, you see TDP-43 in the nucleus, right? So you see a lot of binding in intronic regions. Here, for example, you can see this blow up here, beautifully completely covering AGRIN1. But the transdifferentiated neurons, by clip analysis, you don't see these binding patterns anymore because now it's in the cytoplasm. So it's pretty exciting to see that we can try to model this. We think this is not just in this funny transdifferentiated state. We've done many lines, but also in old mice. So in 24-month-old mice, you see TDP-43 and other splicing factors already in the cytoplasm, but in young mice, it's in the nucleus. I have other sort of phenotypes here that I might skip over. I'll skip over this one. But we think that a lot of the RNA granular components are already pre-aggregating. So that means in the older neurons, in quotes, many of these proteins are already aggregating in the cytoplasm, which may be why if you have a genetic variant in TDP-43 or something else that doesn't clear TDP-43 from the cytoplasm, you get this acceleration that is age dependent, right? Like this was the other factor we've been missing. Okay, I'm gonna skip this one as well. So we think that this may be a different way to model age-driven RBP, RNA binding protein mislocalization or misregulation. And you have chronic stress that you can add to an iPS-derived cell line as a way to overcome it, or the epigenetic age itself is a kind of stressor already that that without any treatment in the cells, you get this age-driven defect. And so again, we are continuing to pursue this interplay. How do you model stresses with, you know, and age-related cofactors here with the genetics, right? And so I think having sort of the right ways to model these things could be very helpful in identifying novel therapeutics. And so I'm going to stop here. Thank you for the opportunity to tell you about our basic science, and the folks in my group have done all the work. I just speak about it, and I'll just stop now. Yeah, thank you.
Thank you very much, Jeanne. Great talk. So our last session, we invited 4 industry leaders. Some of them are entrepreneurs. Some of them are scientists and the company founders. So the first speaker, Winston Yan. Winston is a physician scientist and genome engineer motivated by using genetic medicine to treat patients, particularly those with a rare disease. He is the founding president of the N=1 Collaborative, co-founded by Julia and Tim, and co-founder and head of translational strategy at Arbor Biotechnologies, working in both biotech and academia nonprofit settings to enable new treatments. Winston completed his MD from Harvard Medical School and PhD from Harvard University, working with Feng Zhang at the Broad Institute on early CRISPR genome editing technology development. Without further ado, Winston, you can start sharing your screen.
Fantastic. All right, thanks so much. I honestly really wish I were in sunny, San Diego with you guys. My hands are freezing literally right now as I'm sitting at my desk presenting this to you. So I'm going to change gears a little bit from what we've talked about so far. And the gear change is that after all of the data that you've seen from Jeanne, Don, after the stories you've heard from Julia and Amber and Michelle, like it's really now thinking about how do we put it all together and how can we continue to scale this work to move into that long tail of ultra rare disease. So this is not a data-driven talk. This is more conceptual and more about thinking about how we can work collaboratively to make this all happen. So the title of my talk is Individualized Medicines Need Collective Learning. We heard a lot about these ASOs and what types of technologies they can, or what types of treatments at the both N of many as well as N of few level they can enable. But the key is now, as a platform technology, how do we translate that learning into growing the pie for everyone? So with that said, I'm going to shortcut this introduction because I think the left side of the slide needs no explanation to this audience. The right side, though, with this plot where on the y-axis you have the prevalence, or the number of patients in a given disease, and on the x-axis just schematically representing all the different disease, I think actually could use some explanation. And this is where my background is both in biotech as well as now in nonprofit comes together, where there's a keen demarcation around 1,000 patients of when a disease is commercially viable. Beyond that, there becomes very little motivation for a company to work on something, particularly as one of its first few programs. There are, of course, exceptions, but the reality is that if we continue with business as usual, with just building biotech companies, there's not going to be treatments for this long tail of ultra rare disease. So I'll give you some examples. I mean, all of those disorders that we talked about with C9orf, with ALS, with Batten disease, they're in this long tail right over here. There are more prevalent rare diseases like cystic fibrosis, sickle cell. I mean, those are the ones that will have treatments. When you actually add up that area under the curve, the AUC of all of these diseases, from 12 patients to 300 patients, that's a huge number of people who could potentially benefit from treatments, but just don't have a pathway towards it. So my thesis is that if we're going to address this long tail of ultra rare disease, we need to break the mold of how we do things. There fundamentally need to be more collaborations between patients, academia, and industry, and that's why I'm happy to kick off this section of the iXCells seminars today. So the key question when I was thinking about this is just why now, right? People have been talking about personalized medicines for a long time, yet it feels like there's a dam breaking at the moment. And I think this gets back to the two people that you've heard from earlier today. It's really showcasing the power of programmable medicine. So on the left, you have nusinersen, also known as Spinraza. That's an antisense oligo splice skipping ASO that was approved in 2016. And then you have milasen that was built on the same chemistry, but with, crucially, a reprogrammed different sequence that was designed, made, and dosed all within a single year. There were a significant number of people that helped in this that already seeded this model of the industry academic collaboration. But fundamentally, it showcased the power of these programmable medicines. And then, you have a single example starts to open up this trickle of more. So Tim published this in New England Journal. There was a critical path meeting with the FDA. Out came several draft guidances from the FDA about individualized ASO therapy. And then since then, about 10 to 15 treatments have been developed by many of treatments that many of you guys are familiar with in this room because you have actually contributed to its development. And so this is all based on one technology, that of antisense oligos. And what I hope I can convince you of is that these oligonucleotide programmable technologies, they're going to be the norm rather than the exception in the coming decades. There's going to be a third pillar of medicines after small molecules and monoclonals that really are genetic medicines that are based on targeting nucleic acids. So you'll have things from siRNAs, CRISPRs, in all this different flavors, whether it's nucleases, base editing, prime editing. There's RNA base editing by ADARs, and just every other week it seems like there's more publications about how we can manipulate this exquisite nucleic acid chemistry to target the very genetic source of genetic diseases. So the reason why I think they're so valuable to accelerate these programmable medicines are so valuable to accelerate drug development and the NFU is that you have this opportunity to really stand on each other's shoulders. This bottom of the pyramid is supposed to represent the huge amount of work that happens in industry, hundreds of millions of dollars of venture capital funding, things that are really driven by that desire to make the billion-dollar molecule. But what you can learn on that particular molecule are things like, biodistribution, your ADME, safety, immunogenicity, how to make the drug, what's the route of administration. All those things you can learn on an N of many, that then suddenly, when you look at an N of few, you can take all those learnings and accelerate the process of developing these customized, individualized medicines by reprogramming it. The way I'd like to think of it is you're turning a problem that was once hardware development, where you have to strip down the molecule and kind of build it up again, to something that's more software in its nature. So why do we need the learnings? Well, I've articulated that there's this base of the pyramid, and that's really the top part of this schematic I'm drawing, where biopharma really is, when you're building these marketed treatments, you have to learn how to make it, you have to take all that money and make state-of-the-art compounds, the CMC, the tox, just the experience of using those platforms is so valuable to bring to academic centers where the investigators may not be initially an ASO expert, but they can work together with the industry representatives to provide the best and most safe and efficacious treatments to these ultra-rare disease patients. But I also want to draw your attention to the bottom part of this, right? The very fact that it's a platform means that you can actually learn on N of fews things that you might not have tested or even evaluated in a otherwise commercial setting. So what this means is that you can have a company that's operating for about 10 years, and they might have 1-2 treatments just because that's how long it takes to make a new drug. Meanwhile, at academic medical centers, you're constantly varying up small pieces of the sequences. You're building new treatments, and you actually learn more from perturbing that different design space, if you will, with these different treatments, but then can take the learnings and feed them back into making better commercially viable drugs. So here's where I'm really articulating that there is that symbiotic relationship, and we should embrace it rather than kind of think about it as like charity work from one direction to the other. So another thing that I want to emphasize is why even between these N of fews, it's so essential to collaborate. It's not just one person doing this, and it's certainly not just one institution, although there are certainly some leaders here. The reality is that these academic or these current N of 1 efforts are often led by very motivated families or a dedicated physician scientists and PIs who are just willing to bend over backwards to make something happen. They also sometimes may not talk to each other simply because it's just, this is all so new, right? I think they need to. You need to have the collaboration between the N of fews for several reasons. First is that when you have safety, the concept of a platform is that as you're learning more about the safety parameters, a single safety event, say in trial here, could actually affect the ongoing trials that are here or subsequent trials that have yet to be designed. So that real-time sharing, or as real-time as possible, that sharing of data really protects patients who are just really donating their bodies to these treatments and understanding how they work. The second thing is just about efficiency, right? If we can share data openly for these patients where sometimes, that adage of time is neurons, right? Like this is true in neurodegeneration. Some of these children have rapidly progressive diseases where if we are constantly reinventing the wheel, we don't get treatments to them in the time that they need. And then lastly, it's the right thing to do, right? For us to transparently work on these and share learning that honors those patients and families where they literally are volunteering their lives, their time to advancing science. So we have this responsibility to learn all that we can from each patient. So we have Mila, you heard about her earlier. This is a proof of concept. We've seen that it's possible, but it's still in the experimental stage, and it's honestly incredibly expensive. Our goal should be, as a field, that we have children who have orphan genetic diseases, they can receive these bespoke, individualized treatments as the standard of care. So in industry, you learn about how do you work backwards from the goal and kind of be very deductive in your thinking. So if we want to find a world in which they're just, the standard of care is this type of individualized medicine, we need to figure out who's going to pay for it. We need to figure out how to scale it. And ultimately, that's also grounded in accessibility and equity. So it's not just the families raising several $1 million who have access to these treatments. Well, to do that, especially for the payment, typically that's gated upon a regulatory body saying, yes, we have evaluated this rigorously, and we have seen the benefit risk is there, and it's approved, or this process is approved, so it's worth paying for. And obviously working backwards a step before, it's how do you rigorously establish the safety and benefit risk? To do that in a heterogeneous field of not just one disease, but many different diseases across different platforms, we really need repeatability, standardization, and sharing of learning. I pose to you that we are still here. We are early in this, and we have an opportunity to do it right and to change the ecosystem together so that we can build towards this future most effectively versus kind of having to reconstruct things in the future. So I mentioned on this last slide, like where we need this repeatability, standard, and learning. And we've thought about the different areas that are, I would say, like self-reinforcing, right? One is thinking through sharing best practices. How do you, and we don't want it to be the Wild West, and there's certain best practices that really are now starting to emerge. How do you share, align, and then ultimately gather good data that's driven by these shared best practices? So if we achieve this, that allows us to reinforce data collection standards that gives you high quality data sets that can inform regulatory or payer thinking. And then here, those data sets could also allow us to improve, right? We can analyze them to say, can we design this better? Could we attract those clinical outcomes better? And we need a space in which we can talk, discuss, and learn across a huge number of different programs. So then we update these best practices based on peer review. Where does this space live, and who's responsible for it? Well, we thought, as being one of the early hubs of individualized medicine, that the N of 1 Collaborative could really serve that purpose for the field, so we can help be this neutral party and an independent body to grow this capability, if you will, for the good of everyone. And this is where I'm just going to briefly talk about what we do. Our mission is to make individualized medicine safe, effectively, and widely available. And the key is as a routine treatment option, not just like, last line. It really becomes this routine option for rare genetic disease. We do this by convening and discussing. We share our learnings. We make sure we're openly sharing our best practices. We're building a database on which we can deposit both preclinical and clinical data to learn and iterate. And then we just simply put, there's value in connecting people, much like you are doing today in that room and online. We think it's so important to be able to do that routinely via us as a hub. And above all of our work is based on rigor, independence, and transparency and inclusiveness. So in the short time that we've been around, we've really had this opportunity to first of all, genetic disease is worldwide, right? So we have participants from around the world. We have over 500 now as our membership. In our specific work groups, we only have 2 full-time employees. Everyone else is a volunteer. And so they're participating with their excess capacity, just like myself, their spare time, because they care so much about this. So we have 65 volunteers spread across these 7 work groups in the bottom. We have 9 signed data sharing agreements. We've published 3 things and more to come. And we have 1 preclinical data partner and 1 prototype clinical database. And again, we do this all to help grow the entire field in individualized medicine. So that's it about the N1C. It's a very small snippet. As I mentioned, this is really a concept talk. And I want to share with you in the last couple of slides where I think this can all go. In particular, just advocating for what I see as a really, it's a new horizon. It's a new specialty called interventional genetics. And why do we choose the name interventional genetics versus, gene therapies or genetic medicine? It's that there's really a precedent for it, and the name matters. So if you look on the left, we can start off with interventional anatomy, right? Like this is once upon a time people just observing. And then finally, with that knowledge, they're saying, now we can do something about it. Let's actually perform surgery. More recently, you had radiology and other associated internal medicine fields starting off with just observing, but now they have tools that can go in through blood vessels and actually start opening up vessels that are clogged or retrieving clots. That is the intervention that they're doing with what they're able to see. And I pose to you that there's an opportunity in our very near future that, looking at pedigrees, looking at exomes, we're now identifying, just observing the causative mutations. But with all of the technologies that I mentioned earlier, we have a chance to perturb and intervene. So hence the name interventional genetics. And I think this is actually an important thing because this procedural model provides a couple of important precedents. I'll walk through this and I'll share with you why I think that there's important parallels to be drawn for the sustainability of this ecosystem. Take the interventional radiology approach. There's already a set workflow in which a physician determines the exact approach. An industry collaborator, or really a vendor, they create the tool. Sometimes you can even have very custom 3D printed N of few tools that are broadly approved by the FDA, but they're still just made by the manufacturers. And then finally, the MD in these interventional cases, they do the intervention and they monitor the follow-up. Every patient is different. Their natural history is different, their disease course, their anatomy is different. So it's like the N of one, but in anatomical form. So meanwhile, you have on the bottom, the proposed interventional genetics, where similarly an MD, which I propose in the future will be an interventional geneticist, they look at their genetics and their genome. They might be assisted by preclinical design tools. They can choose, is an ASO appropriate? Is a base edit, a prime edit, you name it. And then similarly, an industry partner would create the tool, and they would work with, say, a unique partner like iXCells to maybe screen these in a standardized set of assays to establish what are the criteria for here is a genetic medicine that is safe to go into patients. And then finally, the MD, they may not have to understand any of these things that the industry does. They deliver the therapy, they track the outcomes, and they follow up. So it's important to have these precedents because here are some very different things about doing this as a procedure versus doing this as like a clinical trial. First of all, this practice of interventional radiology and procedural work, it's governed by a professional society, something like the N of one collaborative, place for interventional genetics, hopefully, as well as institutional boards themselves. They're locally and professionally regulated versus by, the FDA. It's very much about treatment with iterative learnings. It's not about doing a clinical trial. It's also not about compassionate use Hail Marys. They're saying, let's try something at the end of a patient's life just because we don't have anything else. You're treating someone, but it's important that you're iterating and those learnings get captured to help the next treatment become better and better. That's how surgeons learn for decades and if not centuries. And then furthermore, the business model exists, right? Industry gets to provide the tools. They might not be making $1 billion in any single given molecule, but what they're doing is getting small amounts of each treatment that's being made. And similarly, the academic medical centers, they're able to bill for all of their practices as well. So I think this is a model that we can build towards if we start from where we are now of saying, let's actually start embracing the ecosystem approach of how can we, how can you today say, let's build this ecosystem. Let's embrace pre-competitive sharing. Can we reset the drug development paradigm, particularly for these NFUs where otherwise we're not going to get there? Can we intentionally have people talk about bias towards action, right? Well, what about a bias towards openness? The default can be sharing, and we kind of radiate that outward so that we encourage others to do the same. And then, as I mentioned, the N of one collaborative, we have room, right? There's iXCells is already an incredible core sponsor with your early vision. Many thanks to Jeanne Loring. But all of you who are listening online, come to our website, email us, and see how you can contribute your expertise, your time, or just pure dollars and cents to help enable individualized medicines. So I leave it here just saying, none of this is impossible today. I think the science has some work to be done, but fundamentally, it's not a gravity problem, as I put it. Many parts are hard. We're going to need to work together, and we're going to need resources. But it's just, to me, when you see the possibility of the tools we have, and you see these patients who need them, we owe it to them to connect this together and build this field for these patients who need us. So with that, there's many people to thank, but first and foremost, just wanted to say, Jeanne Loring, you and your team at iXCells have been a believer in this from the beginning, and just so wholeheartedly throwing yourselves behind this. So thank you, and Yeah, I'll leave it there.
Thank you, Winston. So I saw I was attending those weekly seminar hosted by N-of-1. That's great talk. A lot of patients starting to share their experience. I think that's exactly what we are trying to do here in order to tackle those long tail problems, right? So we do need to create, last time we discussed a little bit about how to create a kind of like marketplace that people can start depositing the data and contributing their knowledge. So I think N-of-1 is an equal strong collaborative can be one of the potential platforms. Keep up the good work. Our next speaker is Jeanne Tao. Jeanne is a director of in vitro pharmacology at iXCells. Her team focuses on supporting rare disease patients through antisense oligos, siRNA, and small molecule screening, contributing significantly to advance the N-of-1 therapies with multiple patient foundations here. With a PhD from New York University and postdoc training at Harvard University, Jeanne brings academia excellence and over 11 years of experience from roles at Amgen and WuXi AppTec in drug discovery and CRO services. Jeanne, the stage is yours.
All right. Good afternoon, everyone, and welcome to iXCells. So my presentation today has 3 parts. First, I will give a very brief overview of our company. And then the second part is the disease-relevant cell models. And number 3 is the drug screening platforms. So first, I would like to give you a very brief overview of our company. iXCell Technologies was founded in 2014, headquartered in San Diego, California. And over the past 10 years, the company has undergone remarkable transformation by recruiting experienced commercial staff and executive members, as well as the talented scientists, building the infrastructure and expanding our pipeline, product pipelines, and also CRO services. So today, as you all know, we are celebrating the grand opening of this beautiful new facility, which is definitely a milestone for us moving forward to the next phase of growth. So our expertise is to provide innovative solutions for preclinical drug discovery. And we offer both products, cellular products, as well as preclinical CRO services. So I will share some details with you in the later part of my presentation. So moving to the second part, which is the disease-relevant cell models. So this slide is a summary to show you our business model. We offer 2 major categories of cellular products. One is primary cells. The other is iPSC and iPS-derived derivatives. So in addition to the ready-to-use cryopreserved cells, we can also perform customized primary cell isolation, as well as iPS reprogramming. So we can utilize the cutting-edge technology CRISPR to manipulate the iPS, either by inducing a mutation or by correcting a mutation. So the iPS cells can be targeted, differentiated into different cell types, including different types of neurons and other cell types like hepatocytes. And those cells can serve as cellular models for different stages of studies in drug discovery pipeline, including target identification, drug screening, functional assays, as well as toxicity assessment. Primary cells are valuable tools for preclinical studies. So we have a diverse collection of primary cells, including multiple cell types as listed here. and epithelial cells, endothelial cells, muscle and neurons, etc., isolated from a variety of tissues as listed here. And those cells can be from animals, different species, rat, mouse, etc. And also we have a large collection of human adult donor primary cells, including individual, healthy individuals as well as disease patients. So, and we can also conduct multiple cells isolation, primary cells from the same donor, which is a very powerful approach to study different cells from the same donor. And in terms of delivery, we have different delivery methods and platforms. The primary cells can be shipped to our customers either in the frozen vials or in the fresh culture in the culture dishes, just depending on our customers' needs. We also have a very stringent QC process to ensure the quality of our primary products. Induced pluripotent stem cell, or so-called iPSC, is another expertise areas at iXCells. So we helped many pharmaceutical companies as well as rare disease patient families to generate iPS lines from either PBMCs or skin fibroblasts. So we have a collection of both healthy donors iPSC lines as well as disease iPS lines. And as I mentioned earlier, we are capable to use CRISPR technology to manipulate gene expression in the iPS lines by either introducing a mutant or correcting a mutant. All the iPS lines generated in-house, we perform a comprehensive characterization, including cell variability test and the alkaline phosphate staining and iPS marker staining, as well as cryotyping. And again, all the cell lines has to pass our QC, make sure it's mycoplasma free and pathogen free. So all the iPS cells can be differentiated into multiple cell types, as you all know, which is a revolutionary approach. So since our focus is neuronal diseases, we are able to produce multiple types of neurons, and those are our catalog products, including a cortical neuron, motor neuron, and the GABA neuron, and dopaminergic neurons. And all of these different types of neurons have been used in our in-house CRO services, as well as cellular products that you can purchase. So we can also differentiate the different other cell types from the iPS, including muscle cells and hepatocytes. So our team is working really hard to keep this list going longer and longer. Moving to the third part, as I mentioned, so we are not only offering cellular products, we are also providing preclinical CRO services. So drug screening platform is one of our services. And one of our focus right now is to develop personalized therapy to treat rare diseases. As you can see here, I listed several cellular models, and I use neuronal diseases as an example, which we can utilize either both immortalized cell lines or iPS-derived neurons, as well as primary cells. So later in my presentation, I'm going to show you a summary table to compare the differences and our recommendations for these different cellular models. And those cellular models in our hands empowered us to work on multiple drug modalities, including, but definitely not limited to, RNA-based therapies, including ASO, antisense oligo, siRNA, and we can also work on virus-based gene therapies to deliver mRNA, CRISPR, and base editor. we are able to evaluate small molecules as well, either by screening a large commercial libraries or a smaller customized collections. And the last item is so-called drug repurposing, which means we try to uncover a new indication for FDA-approved drugs to use for a new disease. So we have a standard protocol, a procedure for drug screening. Here, I'm just showing you an example of this flow chart is a typical ASO screening performed in patient cells. So the very first stage is always a discussion with our customers. And most of the time, those are private foundations and the rare disease families or research labs. So we try to understand the biology and understand the needs of our customers. And the next step would be to generate patient-derived iPS cells, and then differentiate those cells into the disease-relevant cell type. And then we need to design the ASO, synthesize the ASO, and perform assay development, followed by high-throughput screening. Of course, the hits we identified needs to be conformed and characterized. So to help the rare disease patients, and especially the young kids, we understand we are not only fighting with the diseases, and we are also most of the time fighting against the time. So one of the most common questions we always receive from the patient's parents is, how long does it take to develop an ASO treatment? So here I'm showing you a timeline estimation. And this estimate is based on the development of Millicent. And you heard a story from Julia earlier this morning, which is Mila's mom. And you also heard the presentation from Dr. Tim Yu, who led the efforts to develop this personalized ASO treatment. So as shown here, it is possible within 10 months to accomplish all the stages listed here. And as you may notice, some of the activities happened at the same time in parallel. For example, generating patient's iPS cells and also produce the iPS-derived neuron takes up to 6 months. But while this effort is ongoing, we can start ASO design and synthesize. While we're waiting for the ASO to be synthesized, we can start the assay development in immortal cell lines. And also we can perform the primary screening in the cell lines. So when we identify the good hits from the primary screening, hopefully the iPS cell at that point is ready. Then we can further conform, validate, and characterize, optimize those hits in the primary cell lines, in the iPS-derived lines. So we have been using this strategy in multiple projects, using this in parallel, different arms in parallel strategy, which worked really well. It dramatically shortened the timeline for the ASO drug development process. But I have to say, each case is unique. So the timeline may vary for each individual patient and each individual case. And another common question we always receive from the parents is, which cell system to be used? So I'm showing you here is a comparison table listing 4 different cellular models. So on the very left side is the immortalized cell line. So the advantage of using the immortalized cell line is it's very easy to access and very low cost and almost unlimited sources. Of course, we all know the disease relevance, the score is a little bit lower compared with iPS-derived cells, but I think it's still a very useful tool or cellular system to be used, especially at the early stage of the ASO drug development process. For example, for assay development or primary screening. And on the right-hand side, for the iPS neurons, of course, nobody would argue. The eye neuron from the patient is the most biological relevant system. However, we all understand it takes longer time, so the cost will be higher. And something in the middle would be a wild type or a normal iPS cells, iPS neurons. And since we have a collection of wild type iPS, different iPS lines and different types of neurons, so that will save us some time and also some cost. And in some cases, we can utilize the wild type line, eye neurons to perform the screening. So moving forward, this is a very simplified chart to summarize how ASO works. So we can utilize this very small oligo, a short oligo nucleotide antisense to either increase or decrease the target gene expression. So for downregulation, two well-known mechanisms are RNase H mediated mRNA degradation, and the other one is so-called steric blocking to stop translation. And for upregulation, one mechanism is to inhibit naturally existing mRNA, microRNAs or antisense, or a so-called splicing modulation, either by exon skipping or exon inclusion. We have been working on multiple ASO screening projects. So I'm going to show you some examples now, which utilize a different mechanism of action for the ASOs. So this project, we collaborated with PCH1 Foundation. And you heard the story from Michelle earlier this morning. So the goal here is try to identify knockdown ASO in immortalized cell lines. So we utilized a cell line called, which is called SHSY5Y cell. It's a neuroblastoma cell line. So we screened a relatively small collection of ASO, only 30 ASOs, but we are very, very lucky to identify 2 of them give us inhibition effect on the target gene. And then we moved on to the next stage, hit confirmation. As you can see that both of these hits give us very nice dose-dependent gene knockdown effect. So the third stage will be hit validation in eye neuron, which is ongoing just next door in our lab. So hopefully in early next week, we will get the results. Case study number 2, collaborated with India Accelerator, as well as Boston Children's Hospital and Crayon. So the goal here for this project is to identify allele-specific knockdown ASO in patient iPSC-derived neurons. As you can tell here, we are measuring or monitoring both the expression of the wild-type allele represented by the blue dots, or the mutant allele expression represented by the red dots. And higher concentration of ASO is the darker color. And we have 3 concentrations for each ASO. And for this particular project, we screened a very large ASO library, about, I would say, over 800 ASOs total, because it's not easy to identify an allele-specific ASO, knockdown ASO. But as you can tell, at least in this orange box I highlighted here, these 3 ASOs give us dose-dependent downregulation of the mutant allele, but has a very minimal or small effect on the wild-type allele. Case study number 3. So this is a collaboration between iXCells and the Quido Chat Foundation, as well as Meiji. And the goal here is to restore a target expression by targeting microRNA. There are microRNAs naturally exist, which can inhibit gene expression. So the first stage for this project, we evaluated 11 microRNAs in immortalized cell line. And we are very lucky to identify 3 microRNAs showed inhibitory effect on the target gene. Stage 2, we wanted to select which cell type, which cell system we are going to use for ASO screening. And we evaluated the 3 identified microRNAs, both in the immortalized cell line, as well as 2 NPC, neuron progenitor cell lines. And we found that neural progenitor cell has similar or even higher levels, endogenous levels of the microRNAs. So now we have the candidate microRNA, and we have the identified cell model. The next stage would be to perform ASO screening, try to identify the ASOs which can inhibit those naturally exist microRNA. And we are currently in the discussion phase for the ASO screening. So hopefully we can initiate the screening very soon. Okay, so those 3 examples are just some cases among all the ASO projects we have been doing so far. And as you can tell, we are collaborating with multiple patient families, private foundations, as well as research labs. So, and we worked on different diseases and using different design strategies and so on. So definitely this is a very educational process for our team as well. We are learning a lot, gaining experience. So hopefully we can serve our patients better. And here is a list of small molecule projects for rare disease. So for rare diseases, we collaborated with Rarebase and to evaluate small molecules. And some of these projects are so-called drug repurposing, trying to identify new indications for the FDA-approved drugs. So we work closely and collaboratively with all our partners within the rare disease community. As you can tell from here that the ecosystem is built and with the contributions from biofarms, from research labs, as well as patient families and foundations. So I think we need to work together really hard, support each other, try our best to save and help those lovely kids. Finally, I want to say thank you to all our collaborators. The list here is definitely not exclusive. Those are just some organizations whose projects were referenced in my presentation today. I also want to thank the whole iXCells team. So today's presentation is definitely a joint effort, a team effort. And thank you very much for your all.
So we have 2 more speakers. Those are all our great fans of iXCells. So the next speaker, Christopher Hart. Chris is the co-founder and CEO of Crayon Bio. He led Ionis Pharmaceuticals Genomics Department, pioneering company-wide initiatives in rare and common diseases. With a PhD from Caltech and postdoc training at Yale, he blends research and teaching in bioinformatics. As a visionary leader, he advised the White House on diverse topics before shaping the future of biology and technology at Crayon. So,
thanks everyone for sticking around, and thanks for the kind introduction and opportunity to talk. So I think I'm gonna, where I'm gonna come in is I'm gonna start talking about what we're building at Crayon. I think it connects very nicely from all the stories we've heard today. And as we work forward, I think one of the things that probably highlights why we're all here today is really this notion of what modern biology really gives to us. And I think Francis Collins nailed it about a decade after he sequenced the genome with the public genome project, where he says perhaps the most profound consequence of the genome revolution in the long run will be the development of targeted therapeutics based on a detailed molecular understanding of pathogenesis. This is actually why we're all here really today. It's because we can, in fact, understand at a foundational level what's driving pathogenesis, both for common diseases often, but also for rare diseases, And I have a very similar slide to Winston's slide where we're looking at this long tail of rare diseases. And I think this is actually the product of exactly what we see with modern biology. So we really can understand the genetic basis of disease, and it drives to us understanding that there's a reason for the rare disease crisis, and maybe there's hope that we could actually start to address some of these underlying pathologies. Francis also highlighted really what's holding it back, though, which is it's not our lack of desire or hope or want to sort of build therapies in this place. It's really that it's hard. And he coined it here very, very eloquently, where he says, this is also the goal most challenged by long time lines, high failure rates and exorbitant costs. So when we think about how do we start to make inroads to developing therapies across this long tail of rare diseases, obviously we're thrilled to be part of an ecosystem here in San Diego and more generally globally, where people want to collaborate, where there is a lot of interactions to go forward. But I think we need to also think very deeply about what are the foundational challenges that are blocking us from making strong process here. And I think there's an elephant in the room, and it's the same elephant in the room for common diseases as well, which is that it takes too long and costs too much to make too few drugs. And if we extrapolate that out, and I think it was said by multiple people here today, if we just extrapolate out the timelines it takes to make a drug, and we look across that long tail, well, none of us want to face a millennia while we wait. And nobody wants to spend the GDP of the planet to make these drugs. So we really have to think, how do we foundationally lower those costs? How do we make it more efficient? And how do we make it so that we don't have to raise millions of dollars to build these drugs? And you know, we've heard many talks about the promises of oligos, so I won't dwell on it, but the real value of oligos is that they do have this capacity to intervene at the foundational processes that drive molecular biology. We know that we can engage directly to modify the expression of genes, either at pre-mRNA level to mRNA level. We can change how they splice. We can edit genomes. We can introduce new things. We can even change how the ribosome engages. We can change how proteins interact with molecules. What's the commonality here, though? We heard this notion of a programmable medicine. I think that's a fantastic term. What makes them programmable, though, is that they're actually just polymers. They're polymeric molecules. They're not small molecules. And as polymeric molecules, there's a thing that makes them really interesting as drugs, which is that the base sequence that we use to interact with the targets of interest, most often that happens to be a nucleic acid like an RNA, that's actually separable from the chemical modifications that we put on these molecules to make them into good drugs. That separability is actually what makes them programmable. That's what makes them possible to be put forward. And the thesis that Crayon's put forward is that you can think about it as programming, but I think it's more generally, and these are hardware. This is an engineering problem. How do we actually solve this as an engineering problem? How do we make these polymers engineerable? And when we think about that, then we have to think about what is the thing that we need to engineer most importantly? What is a sort of an invariant property of drugs that we always want to make better? I don't think anyone's going to ask you to make a drug less safe, with exception of maybe some chemotherapeutics, but they want those safe as well. They just want them targeted. And that's what's really been, I think, limiting the field, is that we've seen amazing success stories, and I think we all can stand on the shoulders of our history and the past and say that there is huge potential with this modality, but we can't look at it and not say that there's an issue, which is not only when you change the base sequence, it may be toxic. It may not be active, but it may be toxic, and you don't see that until you actually put it in animals, because those toxicities emerge actually as organ level failures. And this is something that's plagued the field and really held back our capacity to push these drugs forward. So how do we get to a place where we can solve that problem? And when we started Crayon, it was really about seeing this as the opportunity, which was we should be able to engineer these compounds, but we don't know how to engineer them, and we don't know how to engineer them because we haven't collected a sufficient amount of data or the right data to actually address the things that actually hold back the field. And I'll sort of give you an idea of why it's been so hard to collect this data. And it's because the space is ridiculously large. And for those who have thought about designing oligos before, you've probably already stumbled upon these numbers. For those who have just started to think about this process, Most oligos are somewhere between 16 and some odd 20 some odd bases long, but they're short. But they're composed of 4 bases, A, C, Ts, and Gs. Even if you go in the shorter end, 16, that's 4 billion possible combinations. That's a big number. But then we can't just put a DNA and RNA molecule into the cell. The cell doesn't like it, has a lot of adaptations to both get rid of it, or worse, will think it's foreign DNA and have an immune response to it. So we chemically modify them. And we chemically modify them in different places. And I show here a molecular structure. For the scientists among us, we've all thought about it before. For those who are new in the space, we have bases over here. These are the things that allow us to make Watson-Crick-Franklin hybridizations to the target genes of interest. We typically modify the sugar that the base hangs off of, and we typically modify the backbone. And sometimes we even modify the base. Every time we make these modifications, the key thing that we need to recognize is that they're actually not very far away from each other. And if you were an electron hanging out in this sugar, you're going to be impacted by whatever this base is. And likewise, if you're an electron hanging out in this backbone, whatever the sugar is going to change properties. And what we know is that the electrostatic properties, or where electrons like to be on these molecules, impact what proteins they like to bind to, what proteins they don't bind to, how they aggregate. And those interactions are the things that drive the pharmacology, whether it's active, whether it's safe, whether it migrates to the cell. Those are not independent now. Now we have this collusion, this deep collusion between the sequences you use, which we know we have to modify because that's how you're going to target your gene, with the chemistries you use to improve the pharmacology. The field's been hoping that maybe there's one sugar or one backbone that'll make it safe across all sequences. We've yet to see that to be the case. In fact, it hasn't been the case. It probably never will be the case because they are in fact entangled. They're stuck together. And just to follow up on my combinatorial problem, that means that you have to understand how when you make these modifications, they're going to interact together. And that means that you're dealing with, like, even with simple things, 10 to the 24 possible molecules to construct. Now, if you're thinking that doesn't seem so big. It's huge. It's like the number of stars in the galaxy. It's a big number, which means that if you're thinking about what can I use, what data can I use to learn my way around this space, you're going to be screening for the rest of time before you even come close to approaching a place where you have enough information to actually extrapolate. So we have to think deeply about how we find this. And that was one of the first things we recognized at Cran, is maybe there's a way to survey this space in a way that was a little bit more efficient, but it required us to think a little differently. And I think this is one of the critical things that we've thought about, which is a lot of times, I've heard it multiple times today, there's no business model in this space. The long tail means that, these patient populations are too small. And it's because people haven't thought about how do we actually integrate across that tail such that we can combine that revenue. And that's where you get a business model. But the way you do that means you have to be able to actually distribute your costs across that long tail of diseases and distribute your workload across there. Sometimes you'd hope that the same drug might work for multiple diseases or work for a common disease. But as we've heard, sometimes you need a real selective drug. Sometimes these modifications or these variants are exceptionally rare. Maybe one, maybe a few. That means the same drug is not going to work. It's not going to extrapolate. So you have to think about how do you integrate these efforts across multiple programs so you can then share that workload, so you can distribute the cost but still integrate the revenue. And that's really where we are at Crown, is we're thinking about how we do that. And we do it by changing our perspective. We do it by thinking about how do we solve this engineering problem? How do we change making oligos from a discovery effort to one in which you're actually engineering compounds that have favorable pharmacology? And just to sort of highlight the point and why it's so important, and this is something that I think is critical for anyone walking into the space to understand, which is that the sequencing chemistries do in fact collude quite a bit. I have a little data slide here. It's 96 oligos targeting a liver-expressed gene in mice. None of the details all really matter, except that they're all made with the exact same chemical motif. And for those in the space, this isn't all that surprising, but what should be highlighted all the time, which isn't always recognized, is that these compounds along the x-axis is knockdown in vivo in livers, and then on the y-axis is a measure of ALT, which is toxicity. If you go above 100, it's considered to be pretty bad for the mice. Once you get up to, and that'd be a log 10 of 2. And you can see very, very clearly what comes out is that there are compounds, same chemistry again, just different sequences, that look terrible. I'm going to tell you, this isn't because they're making some off-target binding to some other gene and knocking down some essential gene. It's just because they're binding to something in the cell that makes the cell unhappy. I don't know what it is. I just know it makes the cells really bad. I want to avoid those. And then I can tell you there are other genes, other oligos, same chemistry, different sequence that look like saline, except they knock down this gene. I want more of those. I don't know why they're so safe. I don't know how they get in there. But what I want to understand is what are the chemical properties that have been imparted into those molecules when I put new chemistries on with some aspect of the sequence that produce this biophysical property that allows it to sneak into the cell, do what it's supposed to do and nothing else. So that's what we need to figure out how to do. But the key thing here is the sequencing chemistry collude. And they collude here when we're sort of pulling chemistry constant, but they also collude if we do the opposite, which is say I have a sequence and I just put on different chemistries. Sure enough, we can have the exact same problem. So in this particular case, I took a drug that was targeting a gene, and I put it into a mouse. This is a 3-10-3 lock nucleic acid. For those who are in the space for a while, these were historically thought to be pretty toxic in the liver due to some publications that came out at some point. It turns out there's just a collusion that was not appreciated. They're not all toxic. It's just some of them are toxic. But when you put them in the brain, you can have things that look actually pretty clear. But one of the things we had to think about is how do we assess toxicity in the brain and be very careful about assessing for toxicity in the brain, because last thing you want is have neurons dying, and you have to be very sensitive about how you see those. So when we do that, we actually look at tunnel staining, where we can look at apoptosis in the brain at a very high dose. And we see these compounds are exceptionally well tolerated. Generally, if you choose them well and you design them well, you engineer them to have properties that make them well tolerated. But if you take that same sequence and make it a 5-10-5 mode, which is, there's lots of safe 5-10-5 modes out there, but they're not all safe. In fact, this one actually causes quite a bit of neuronal death at half the dose of the LNA compound. That's concerning if you're not paying attention to the fact that the sequence and the chemistry collude. You can't just assume you're going to change the zip code and magic's going to happen. These are foundationally different molecules every time you change either a base sequence or a chemistry. And you have to understand what you're doing by figuring out those design rules, those engineering principles. And that's really what we've been building. The sort of the foundational idea here, though, is how do we think about taking this notion of a polymeric molecule and turn it into truly an engineering platform. So we've been spending a lot of time creating these really bespoke data sets where we can learn. These data sets are not meant to teach us to make a drug. These data sets are really meant to inform models that then allow us to connect the biophysical properties, really from a quantum first principle, to the pharmacology that we observe by putting these molecules into mice and observing with very detailed assays. And these are like IND-like studies, but at the end of the day, what we got to is this place where our models allow us to improve the random hit rate of getting a safe oligo by at least 30x. That has immediate benefits in how fast you can go from I have a target of interest, a allele I want to target, to a drug that I might be able to put into a patient. And I'll show you a story where we were able to make a drug using the platform in about 13 months. We've done that more than once. We also use that same platform. This is where I think we get to, how do you think about the business model? Well, this same platform works for making common disease drugs as well, because we want to modify common diseases just like we do for rare diseases. They're just different genes. We want to be able to program modifications of genes that have pathology. And we can do that with the same sort of basis if we understand how to engineer these compounds. And there we've handed over some compounds for a neurodegenerative disease for a partner, and then we have new compounds coming up for a common disease. And we've repeated this process. It takes about 4 months to go from a start to a couple of leads. The other aspect, which I'm not going to talk today, is about these compounds are amazing. They can get into cells and do great things to modulate gene expression, but they only can do that if they get into the cells. So we've also been working on how do we make access to different tissues and cells also programmable. And our core competency is really about how do we engineer polymeric nucleic acids. So we made aptamers. Aptamers are just slightly longer aptameric extensions. We sometimes think about them. These are exactly folded sections of an oligo that you put at the end, and they fold up into things that can have high binding affinity to receptors on cells in a very specific way, so they can get pulled in to the right cells at the right time. Ideally, what we're aiming to do is really unlock both common and rare diseases by being able to sort of engineer these compounds in a way that is differential from the way that you might think about discovery. The sort of one layer deeper into terms of how the platform works, I sort of alluded to this, is that we spent a lot of time, and this is another thing that I was actually a little surprised, but when we started thinking deeply about what drives the properties of these molecules, we realized it was in fact the electrostatic properties of these compounds. Electrostatic properties are where electrons live. And then you go and you talk to your computational chemist and like, that's cool. That's a quantum problem. You got to understand the quantum nature of these molecules. But then you realize the challenge there, of course, is that you can't really simulate a whole polymer at that sort of level. So we had to spend a fair amount of time building up ways of stitching these molecules together. So we could take a quantum resolved model and stitch it together into a larger molecule that would still project out biophysical properties that were meaningful in terms of the pharmacology of these types of medicines. And that's an interesting academic thing we did, but it was only the point in which we were able to actually connect it then to hard-earned pharmacology data sets that we created with a very purposeful intent to inform models. So here we did thousands of IND-like rodent studies where injected compounds that were pretty, they're pretty useless by themselves, but in aggregate, they built this platform that allows us to engineer these compounds. One of our investors, when I first showed him the set of data that we created, we'd done our first set of, I think, a couple hundred compounds, put them into mice, did this ridiculous experiment, really bet the company on it, showed it to our investor, and he came down and he was like, that's great. Chris, you just did the largest mouse study I've ever seen, and you still don't have a drug. And I was like, yep, that is true. I don't, but I have something better. I have a platform, and I can engineer drugs. I'm biasing ourselves to making drugs, and part of their mission is to invest in the Star Trek future rather than the Star Wars future. So we were on a path to making a radical abundance of gene-centered medicines, in the words of Matt Aka, the founder of TCBC. So we put that forward, and we couple, of course, this huge amount of data from rodent studies with, rodents are great to understand mammalian toxicities, but we do worry about human things. So we also put these oligos into in vitro systems where you can look at things in tissue culture, so we can understand human cell culture experience. But then the real crux that you have to worry about a little bit sometimes is that immune systems are different across all species. So we also spend a lot of time doing ex vivo experiments with PBMC assays to understand what the immune impact of these compounds would. And you can't do that in a monkey, you can't do that in a dog or pig or mouse. You have to find something that's humanized. So that's why we have these ex vivo experiments. Plus, when you're in vitro, you can do dose response and time courses that you can't afford or think about doing in animal studies. But this data together is what ends up aggregating, and then we use very, very purpose-built machine learning tools to solve what I think is really a problem that's perfect built for machine learning, which is it's an engineering problem, which is how do I connect a feature vector of biophysical properties on these molecules to pharmacology that I observe in vivo, so I can then understand what properties I want to impart on my molecules to have the favorable pharmacology that we want on our drugs. And by doing that, we now can say, for any given compound, here's the sequence I need. What would be the right chemistries to impart a safe drug? And with that, we could start to make drugs. So I'll tell you one story of making sort of a allele selective drug for a rare disease. It's a story that's too familiar, but is also a story of hope, I think, which is this family was able to get diagnosed about 1 month of age. They enrolled in a rapid whole genome sequencing study. They got sequenced, and they were identified to have this single nucleotide variant, exceptionally rare. There's one other individual in the world that had been identified with this particular variant, and it was a nuclear transporter. And that nuclear transporter kept things in or out of the nucleus, and it was particularly interesting because it was effectively causing things to be willy-nilly moving in and out of the nucleus. But he had one good copy, one bad copy. The therapeutic hypothesis is pretty straightforward. Let's knock down the bad copy and hope that the good copy provides sufficient transport in and out of the nucleus to be helpful. The patient was presenting with seizures and sort of having real trouble. They found us about 7 months after their diagnosis, this is never a fortunate encounter in that sense, but it was a counter that provided from synergies, which is to say we were in a place where we could say, yes, we'd like to try to help. So we tried to help, and in about 5 months after we fired up the platform, and this is a place where I think collaboration needs to be smoothed out. We waited for a month, maybe a little longer, to get some cells out of some academics, to make sure we could do the experiments that we needed to work with. And then we were able to push forward and start doing the experiments. We ran in parallel, of course, the activity study and the safety pharmacology studies. And we were able to run it with a very small number of compounds because we had high prior that these compounds should be safe based on our models. at the end of the day, the compounds work pretty well. What I show here on the left of the left graph is the relative expression measured through an NGS assay. And you can see we have a control oligo, doesn't target anything really, and you get, it's pretty flat line. It tells you two stories that one, an oligo doesn't do anything to impart the sort of allele expression of this gene if it's not designed to that particular oligo. And the patient fibroblasts here express the gene at about 55%, the reference copy or a good copy of this gene. When we dosed with our lead compound, we're able to drive that up to about 90 to 95%. Good copy, 10%, 5% bad copy. That's exactly what we want to do. It's doing what it's supposed to do at a molecular level. But of course, we had to make sure we didn't get rid of the good copy. This just tells you the relative expression. So we also normalize expression. This is measured with qPCR, then overlaid with the NGS data to sort of impart the relative expression of each allele, where the pathogenic allele is colored in red and the reference or good copies are colored in green. Apologies for the colorblind. That's the bad copies on the bottom, good copies on the top. And what you can see is that as we increase dose, we get a very nice knockdown of the gene to about 50% control. We even see the cell doing what we hope it do, which is it realizes it doesn't have a jock opposite and starts to upregulate. But what's really telling is, of course, that we knock down mostly the pathogenic allele. That loss of expression is almost entirely accounted for by loss of pathogenic allele. And then, of course, we are thrilled that it cleared through all of our tox studies. One thing I'll say is that when we do these tox studies, we often think about the tox studies as a the mice are really sort of, and we think about inject them into mice. This is going to be an intrathecal administered drug, but we're worried about systemic toxicities. Why? We worry about systemic toxicities because we think that the organ systems, when you expose them to an oligo, are kind of like canaries in the minefield. It's hard to see things go wrong in the brain because the brain's complicated. It's got a lot of things going on. You don't have a lot of bioavailable markers. But if you put it systemically and you see ALTs go up, you know you're hurting hepatocytes. Will it hurt neurons? Maybe not. But if it hurts hepatocytes, it's probably not the best drug I want in my brain. So we think we use every one of these sort of biomarkers as a systematic way of being sort of canaries to see where there may be warts in an in vivo setting for a drug that could be going into a patient to ensure that we get as much safety as we can on each one of these compounds. And the sort of happy ending at the end of this is that in about 5 months we went from whiteboard to compounds where we were fighting about which compound to advance forward because they all look pretty good. We then scaled up for GMP and GLP studies. This is required, of course, to get something into a patient. We then filed an IND after we got the data back, managed to get that IND approved to dose, and then we were able to dose the patient. I'll tell you, in this process, we had hoped that we'd be doing this in a way that would allow us to have a little extensibility beyond an N of 1, if you will, and we'd hoped to be able to sponsor this, but the patients, the interactions and the patient's symptoms were getting worse, and the regulatory path was not emerging fast enough to do something different. So we fell forward and we used the N of 1 guidance that allowed us to get this into patients as fast as possible. And I'll say, and we'll have more to share on this, I'm sure, in the future, but insofar that patient's been administered for about 6 months and that drug has been exceptionally well tolerated. We have no safety signals, and we are seeing significant benefit as we look at the various different markers of that clinical trial. And I think that highlights where we're aiming, which is, and I think we all see this, right? We see exactly Francis Collins's notion, which is we can understand the molecular pathology of disease now. And we for rare diseases, it's often super clear because it's monogenic and we can drop them down. It comes out of a single sequencing event. For common diseases, this is coming forward as well. We get to a place very rapidly now where we have actionable understanding of this disease biology. What we want to do is we want to connect that dot with a platform that allows us to engineer these compounds in a way that we can get treatments to patients in a way that's efficient enough to make the sort of commercial model about whether you're treating 1 patient, 1,000 patients, 100 patients, or 1 million patients, sort of an irrelevant conversation. It should be about what's possible. If it's possible and you can have benefit, we should be able to make a drug. And then it has benefit, payers should be able to reimburse and we should move forward. And I'm looking forward, and I know this session was a little bit about how do we bring an ecosystem together. And I think part of this is making sure that everyone in this space is able to work together and find places where we can have synergies and be helpful in terms of making sure that as we find paths, we can build off on each other's successes. So with that, I'll sign off.
Thank you, Grace. So last speaker, Jeff Milton. He is the founder and CEO at La Jolla Labs, a company focusing on RNA therapeutics technology. Jeff is the former head of data sciences at Arcturus Therapeutics, contributing to rare disease programs. He was also the director of functional genomics and drug discovery at Ionis Pharmaceuticals. Yeah. Thank you for sticking around. So when I was at Ionis, we used to have this thing called a program review, and it was an annual, it was an annual thing that we used to have to present to the entire company. It was a much bigger room. It was full of people. Stan Crook would sit right in front of you, and all the senior leadership would sit right in front of you, and your boss would sit pretty close too. Sort of feels like that right now because there's my old boss right there, Chris Hart. So. And Tamar was also in our group as well. She's my boss now, but I'm going to take us out a little bit. I have a little bit of a different take on the space of basically a combination of money and how do we create a competitive landscape around rare disease and the interaction with patients. And I'm going to use Lafora disease as a case study for this because I think it's the canary in the coal mine, to use Chris's parlance, for something that's coming that we need to be aware of. And so La Jolla Labs. And so what I'm going to start off with is actually a story. When I was young, I lived in Berlin, and back in those days, it was a divided city, and it had a wall around it. And it took about 2 hours to drive from, so actually the West Germany there was British sector, French sector, and American sector. The big part, that's West Germany. It took 2 hours to drive through the Soviet sector to get to Berlin. So it was a walled city of freedom inside the Soviet sector. And inside Berlin was actually divided into several sectors as well. So there's the French and the British and the American sector. And I lived down on the bottom of the US sector. My father was in military intelligence. So we had a house actually in Potsdam, so in East Berlin, and or in East Germany, sorry, Potsdam in East Germany. So we used to cross this bridge called the Locanabio. And this is that bridge. And the one thing that was very compelling to me when I, as a young child crossing this bridge, and you can see it in this picture, there's a nice green section, and right at the exact middle, it turns black. And that's the border between West Berlin and East Germany. And the difference was that obviously there was care and there was civilization that progressed, and there was a civilization that didn't progress. And the way, as I grew older and understood the economy and how government worked a little bit, I understood the concept of private ownership versus common ownership. And in 1985, this is the Glienicke Bridge. The Soviet Union is on the other side. Now, the reason it was black, first of all, was because back in those days in 85, they used to heat their homes with coal. So you'd have a coal furnace in your basement. And then they would dump the ashes out in the dumpsters, and then they put those out in the street. So everything was black. And everything was just dark and covered. But in any case, what I'm trying to get to here is that in 1985, in the West Berlin side, you could have a BMW M6, 256 horsepower. And then in East Germany, it was a Trabant, 25 horsepower, and you were lucky to get it. It would take years to apply for this, and you only had a couple of colors you could choose from. So in rare disease, we need precision, we need high technology, we need an economy that breeds this type of technology. And so really in rare disease, we need something like the 1985 308 GT. And okay, some people might want to take Tom Selleck with it, but in any case, we need something that's very high with a lot of high precision. And so it's, you know, it's great to be in an environment that actually promotes profitability, competition. These are things that breed innovation. So we have to keep that in mind. And so I come from working many years at this company. Chris and Tamar were there with me. And this is a product of innovation and a competitive environment, is the platform that they developed. They started off with $5.2 million in VC funding. There were 5 other companies that started off in the same field, and they just duked it out. And that's the way it should be, right? So the one who wins is the one who moves forward. And now they're at a point where 16 RNA targeting drugs approved, the first blockbuster drug, Spinraza. They're expecting another approval this year from iPSC-3 GalNAc conjugate. All routes are enabled, broad distribution after systemic administration, targeted delivery to liver, beta cells, and pancreas. And what's really amazing is that now with this technology, over 250,000 people have been dosed, and 20,000, mostly kids, have been dosed intrathecally. So, and obviously there's a lot more that's going on. So what is this technology? And I have this up and sort of showing 2 different pathways here. The one on the left is the Ionis sort of what they focused on, what they developed is essentially this RNase-mediated knockdown. It's a single-stranded ASO, binds lots of quick base pairing to a target RNA. RNase-H being there, essentially located at the point of transcription, cleaves this RNA, and then you have degradation of the RNA, so you don't create that protein. So that's the mechanism of action of this drug. So it's exactly what Chris was saying before. It's a matter of programming the sequences and understanding that. Well, siRNA is the same, somewhat the same approach. It uses different enzymes. It's a duplex of an antisense strand and a passenger strand, and it uses a RISC-mediated knockdown approach. And that's been the 2 main companies that kind of paved the way for that, is Ionis is the ASO, or the single-stranded ASO, and then the Alnylam is the one you think of with the siRNA approach. So now I'm going to jump into a disease that is fundamental to La Jolla Labs, because we actually started our company because of this disease. And it's a disease called Lafora, and there's a drug that was created for it. And it's a progressive neurodegeneration. This is Chelsea Gerber. There's a foundation called Chelsea's Hope. And this is a disease that once you are diagnosed with this, it is a steady decline into a vegetative state and then death. So first, to understand the disease, you have to understand a little bit about how glycogen, the metabolism of glycogen. So glucose enters the cell, glycogen synthase polymerizes or catalyzes the polymerization of this into long carbohydrate chains, single strand chains, and then a branching enzyme takes that glycosidic bond and moves it down and compacts it a little bit and keeps it into a very tight but soluble carbohydrate bundle. And then on the degradation or the breakdown side, glycogen phosphorylase pulls the, or debranching enzyme along with laforin pulls this off, and then it goes back into glucose and it's used for energy. So during the fed state, you create this glycogen polymer and then you consume it as you need it. It's a cache. Over on the right-hand side, you'll see that there's a diagram that shows glycogen in the context of, really, it's been linked to a lot of very fundamental biology. And that's kind of the roadmap that I like to look at coming from the point of view of rare disease, because that means that there's actually a lot of other avenues that you can pursue as far as a therapeutic. So when we talk about this long tail, I like to think that there's a long tail, but there are hidden spikes in that tail. And that's where you can have these rare diseases that have potential broad applications. And that's some of the stuff that we're doing at La Jolla Labs. So in any case, going down the path here to the therapy, this is just a look at wild type periodic acid shift. So it stains the actual glycogen deposits. Laforin knockout. The mutation can actually happen in two different genes, laforin and malin. And as a result, you get these LaFora bodies deposited in the cytoplasm. And that's the pink or the magenta speckles. So you have the laforin knockout and the malin knockout, and you can see that there. So the idea is that's the pathogenic part, is this the accumulation of these LaFora bodies. So if you can essentially reduce the synthesis of this in a way that prevents the accumulation of these glycogen LaFora bodies, then you could potentially keep it in solution and prevent these LaFora bodies from forming. And it turns out you only need to knock down the synthetic enzyme or the synthesis, the enzyme responsible for synthesis, about 50%. So that's the rate limiting step in the process. So I'll skip to this, and this is just kind of a schematic that highlights how this drug works. So there's a healthy state, and that is that you're producing the glycogen at the same rate that you're consuming it. The LaFora disease state is that it's obviously accumulating, and you have aberrant LaFora bodies. And then the therapy and the therapeutic approach is to use a GABAR ASO that you can knock down the RNA of glycogen synthase 1 in a way that slows down the synthetic process of the polymer in a way that, and prevent the LaFora bodies from accumulating. And this is actually a drug that's so this sort of highlights how that mechanism works. So obviously looking at the central dogma, you have the DNA creates RNA, RNA creates protein, the protein goes off and does a function. In the middle there, you'll see the actual diagram. The Pac-Man is essentially the RNA-CH enzyme. It cleaves in the gap of this gapmer ASO, and then the enzyme or the RNA is degraded, protein isn't created. It turns out you only need 50% knockdown to make this work. And so I'll skip through some of this, but basically these are some of the experiments that were done to show that this actually does work in mice. And you can see that with, you can see kind of, actually this image isn't that great as far as the coloring, but you can see that the deposits are much less in the GAIS1 ASO. So like I said, it's a progressive neurodegeneration. And this is, I think she was diagnosed in 2020, and this is her last year. And it's absolutely devastating disease. This drug was created by Ionis, and it really happens at the screening process. So they do the design and they screen, and then they patent it. And it's actually created by Tamara Grossman, who's sitting right over there. So I'm trying to speak for all the work that she did. But so she, so it's gotten to, it went all the way through to preclinical IND or pre-IND phase. And they got to a point where it wasn't financially viable as far as how they were going to move the asset forward. And this is a reality across the entire space. We all have to face this. And again, this is a company that's for profit, and they've got to make the tough decision. And, I think we were there when they made that decision. And so they tabled it. So normally in a pharmaceutical company, you have executive oversight over a particular program, and you have executive decisions and thought and strategy on where that program is going. Once you table that structure, once you table that asset, it goes into a separate bin. And that bin is a bin that's basically business development. It's transactional. It's, okay, we're going to liquidate, we're going to find a way to move this forward. And that's, at that point, you don't have a strategy other than liquidation and moving it forward. And you don't have executive thought or leadership over that necessarily. And that's all pharmaceutical companies right now, especially big pharmaceutical companies. That's not isolated in any way. So what you have is you have a drug that's gone all the way through to preclinical setting. It's at IND, it's ready to be dosed in the clinic. It's a very expensive clinical trial. They have to make a tough decision. And actually, to tell you the truth, we kind of agreed with that decision as far as a financial model. So the reason that La Jolla Labs was created was really to find that way to go past that point. But when you look at the landscape of where LaFora is right now, this is what you look at. And so this is a very difficult place for a small rare disease. So I should say the rare disease is about 200 to 300 patients. So it's in that long tail, and it's a very difficult space for any company. So what you'll notice is actually there are two companies that I've shaded them in blue. These are companies that actually have engaged with the patients, either through natural history or they participated in compassionate use programs. And what that does essentially for a small rare disease population is it essentially sucks the financial motivation out of the investors. In a sense, it's basically what happens is when you do rare disease therapeutics, the funding, the due diligence is a very high priority. They scrutinize that population. And the first thing they look for is who's already there. So when you start a rare disease program, if the NIH decides they're going to come out with a clinical trial and you've got a preclinical asset, you're kind of in trouble because it's all about getting to the patients. And if you have a company that has, or a couple companies in this case, that have essentially completely encapsulated those patients, you can have a good compound, but you're not going to get anywhere. So, and this was actually reflected by, it was actually a whole talk I went to yesterday. I was at UCSF Neuro department, and Dr. James Levine presented the 6 guiding principles that drive innovation in rare disease. And that was one of his principal topics was the patients, getting to the patients in these ultra rare disease programs are a rate limiting step to any of these development programs. So like I said, La Jolla Labs was founded really to try to find a way to do discovery and to bring and deliver these things to the patients. And so what we decided was, there's this concept of essentially a boutique therapeutic. And that is, we do discovery. That's what we do. We create. People come to us. They say, I have a target, and we generate a compound for them, a lead compound for them. And we don't necessarily take it too far down the road. But what we found out is actually that people want to come. They have targets. They want to do a collaboration. They'll fund it. And a lot of times, as we've seen in this room, there's a personal connections. And that's changing the whole dynamic of rare disease, because traditionally there is the big pharma, they sit in the buildings in their cubicles, they don't interact with the patients that much. There isn't a lot of energy, but I'll tell you, I speak to patients almost every day. I speak to patients and families every day. And there's so much more energy in them. And there's so much more information out there. And they're high functioning. They want to figure this stuff out. They have responsibility and they have needs instead of wants. And that's a very different driver. And that's why I think that from the point of view of a boutique therapeutic, you can have this personal connection that's really driving this whole thing. So what is it? It's basically a patient-driven, it's for-profit. I think that's by design. It has to be for-profit, modality agnostic. There has to be a succession plan, and I'll get to that in a second. There's low overhead, meaning it's a very targeted team. And there are built-in de-risking strategies. And I'll talk about that later. And this is very attractive to a whole investment group out there called impact investors. And impact investors are nonprofits. The funds that they get go back into the nonprofit. So they invest with the idea of making money, but those funds go back into the nonprofit. And so, what we're trying to do is basically promote that ecosystem where we can spin off, we can have these companies that come to us, say, we want to develop a therapeutic. It's for, there may be an emotional connection to it, may not, but we find strategies for de-risking those assets in a way that's, okay, here's your target, and then here's how we can de-risk that by saying there are other targets that have broader indications. So we do this, I'll just highlight this very quickly, do a little bit of a deep dive, a little into the machine learning side of things, but we have a splicing model that helps us to do some of the design. We're focused on splicing. So this is an example of where Spinraza sits in our splicing models. We have attributes associated with the exclusion of exons and then the inclusion of exons. And it turns out Spinraza sits in this nice little pocket. It's almost like rational drug design. It just sits right in there. It's very nice. But taking that strategy or taking something like a platform that helps us do discovery and package that up into a team. And I like to think of this, if you've seen my N1C talk, I talk about it. I look at everything from the point of view of a competition where there's an offense, there's a defense, and there's a coach. The offense is, how do I increase demand? The defense is, how do I decrease costs? And the coach is just there to raise money. And so taking something like glycogen synthase 1, there are lots of other indications we can go after. So Lafora is our target audience or target patient population. But it turns out, and as Tamar published here in this paper, it's actually can be a drug that can be used for Pompe. But as you saw that map before, it can go well beyond that. It can go into, it's just a matter of good, smart, scientific minds sitting down and saying, what other areas can we go? What's the phenotype that we're looking for? Can that be, are there other mechanisms that we can go after that can de-risk this asset? And so getting back to our AI models for this particular target, we decided to kind of use this to generate our own glycogen synthase 1 inhibitor, but we're doing a different mechanism. We're going to actually slice out an exon and induce downstream stop codons. And this causes nonsense-mediated decay, and we get 50% knockdown with inducing nonsense-mediated decay using a splicing ASO. And it actually tiles, and we tile these over, it almost represents like a dose response to as you tile into the attributes that are inclusionary, it's cutting that out. We still have to validate this, so this is something we're working on. But in any case, that's the type of asset that we can kind of build into our system. And of course, my slides were a little out of order. I don't know what happened there, but the defense side of this is also to keep in mind that the FDA is a responsive institution. They're not going to proactively do things to enable rare disease. You have to approach them. And they have this as their formula: analysis of the condition, current treatment options, potential benefit, and risk. And when you're talking about something like Lafora, these kids are going to die. We know that. There's no real treatment that's out there for them. The potential benefit is they could survive. And what's the risk? It's minimal when you're considering the devastation of that disease. And of course, the coach is there to explore different programs. So the idea here is that instead of putting a drug into a business development bin, you keep it going with an executive team who can make executive decisions, and you move it forward. when it comes to communicating with the patient population, we actually have a program we're spinning out right now. It's targeted, it's allele-specific knockdown. It was recognized by very expert teams that I didn't even came out of left field. They recognized this particular mutation as having broad indications. So they licensed it. And I had a problem, and that is that it was built for a very small community, and they licensed that. So the succession plan is built into this, and that we put a trust in place such that if that program is deprioritized or fails, it goes into that trust to ensure that that original patient population is protected, because I've already connected with them. I've already been working with them. So this is something that's new. We're kind of figuring our way through this whole thing. And so it's really important. I think that this is something that we're going to all kind of bump up against as we do genetic interventions on very rare populations. Thanks, Winston, for that term. I think that's perfect. The last thing I want to mention is, and this is basically getting to the end, but there are a lot of things that are changing in regulatory space. And there's also, I'm not sure how this will impact us, but it's something we all need to pay attention to, and that's the Promising Pathways Act. This is a law that was a bill that was proposed to this, introduced to the Senate recently by Senator Mike Braun from Nebraska, I think. In any case, I talked to his office. I got kind of the scoop on what the idea is, but basically it's a provisional approval for a rare disease drug, which basically before you even at the time of dosing, it's really kind of pretty far out there, but it's got a lot of support, and it's something you should read and understand if you're in this community and you understand the impact of what this is going to do. I can already tell you that NORD is not a fan of it, but I've looked at the list of who are supporting this, and it's a lot of nonprofits. So it's something you should definitely look at.
Why NORD? That is so.
I don't know. That's why I'm hesitant. I don't know. I would love to have somebody read it. I just read an article that they were not a fan.
How about global genes?
I don't know. It's Charlene, right?
Yeah.
I saw her yesterday. No, I didn't talk to her about it. Anyway, what we are is we are a discovery platform. We're trying to promote these things. And the one thing I'll end with is, companies like iXCells and Oil Labs, we're opening the doors of big pharmaceutical companies. I mean, basically we're trying to promote these small targeted therapeutic avenues. And to the extent that we can support you through preclinical development, that's what we do for very targeted small diseases, small indications. And I have the names in the notes, but anyway, I just wanted to end with, I forgot, let's see. Anyway, recently diagnosed, this is her this year. And then Kyle, I believe his name's Kyle, was diagnosed in 2020, and he passed away last October. And, this is the difference. And the key here to remember is that when you stop a drug from being engaged in the strategy, the disease doesn't stop, it's progressing. So we have to always keep that in mind. And so that's, I think, our responsibility to keep that going. It's all in there. Thank you so much. Yeah. This is end of our presentation, but we will have a closing remark by Amber. And at the same time, we are going to arrange a chair. All the speakers today in our facility, please sit in the front, and we will have very quick Q&A section. We don't have too much time, but after the Q&A, you are more than welcome to stay, hang around, and mingle with your friends here. So Amber.
I wrote down some thoughts after being here for this great day. And the quote that came to mind is my favorite Steve Jobs quote. And it's that if you really look closely, most overnight successes take a long time. And it's so true. When you're at the end of the journey, it feels like it was quick. But when you're in the middle of it, it doesn't feel that way. Before we conclude, I want to thank iXCells for this extraordinary day and my heartfelt gratitude to everyone for attending. Today has been a celebration of collaboration, urgency, and a lot of unwavering passion to make a large difference in the lives of those facing rare disease. And I think that everyone here today has really woven a tapestry of shared values with a commitment to treating every patient with the dignity they deserve, while harnessing cutting-edge treatment for the betterment of all. And we're all united by this common purpose that transcends patient populations to really, or excuse me, I lost my place, that transcends patient populations and embraces the diversity of all of these challenges to witness the collective drive that no one is left behind, no matter if they're an N of 3, an N of 300, or an N of 3 million. And I think that this spirit of collaboration, urgency, and passion is what is going to guide our efforts going forward and how we're going to make progress. It's meetings like this of putting people in the same room with like minds that drive things forward. Thank you so much for contributing to this meaningful day. And I really look forward to seeing how much impact we can have together as we move these rare diseases forward. I can't wait for happy hour and just to give out hugs by how inspired I am for everybody today. And I'm leaving this workshop feeling more invigoration and hope for my little Maxwell. I can go give him a hug tomorrow and say so many people are working to help you. Thank you all.
And for the attendees online, joining remotely, you're welcome to type in any questions. We're happy to deliver to our speakers. Any questions from the audience?
For Dr. Loring, I have two questions. The first one is, I would say Parkinson is an aging-related disease, right? So do you think that the decreased blood circulation or the chronic inflammation contributes a lot to this disease?
I'm sure, we don't know what causes it, honestly, but I suspect that there's some kind of insult that starts, and then inflammation either mediated by astrocytes or microglia, just continues. So one of the interesting things about these dopamine neurons is they have more connections than any other neurons in the brain. They're really metabolically active. And so that makes them more susceptible to insults that might not affect other types of neurons.
So then the second thing is, if you, before transplantation of the iPSC-derived dopamine generation cells, if you mix the differentiated endocellular cell with it, then do the transplantation together, do you think?
Well, yeah, I know what you're asking. The question is whether you can integrate blood vessels into that. And in this case, we don't need them because it's really not very many cells. But there are cases, especially with cardiac transplants of probably just cardiac, so I can think of right now, in which you really, the inside of the transplant will die because there'll be no blood vessels. But for these, there aren't that many, we're not putting in that many cells. So there really isn't a mass that requires blood to be circulating through it. And then I said, I think I told you earlier, we can't really make this too complicated. We have to go step by step with the FDA. And the FDA is already having, I mean, believe me, this was really, they were at their edge approving this because it has to be as simple as possible. And it's not because they don't understand, it's because they want to make sure that you test every component separately and together to make sure that none of those causes any problems for safety, for example.
Thank you.
So it's a lot more challenging.
I also have a question to Dr. Melton. So the disease you talked is glycogen abnormal disease, right? So do you think that diabetes patients, the symptoms would be much severe?
So I don't know if Tamara, you want to respond to that? You're the expert in that space, but I don't think that's true.
No, it's not really. This is a neurological disease, so it's mostly in the brain. So it's systemic kind of effects that happen in these patients because it's a brain specific phenomenon in the patient.
Yeah, it's brain. It's a neurological disease, but yeah.
Yeah, I know
But
because
it's
The patient won't get insulin into the brain. Insulin, does insulin get into the brain? So could there be an effect of the diabetes on brain diseases? Just curious. That's a question.
But I think the overall glucose metabolism doesn't.
They have to do, right?
Right.
Yeah.
It's small.
Yes.
It's small.
yeah, and the neuron only takes glucose as energy. So that's why I'm curious. Then another question is, this is a neuron-related disease. I see you shoot the data used the MCF7 cell and
Yes.
what is the benefit of another one?
Yeah. Right.
So why didn't you use the relevant stuff?
Yeah, so that's part of the de-risking strategies is that we can find there's glycogen has been linked to many Ewing sarcoma is one of them. There's potential so it's upregulated in leptomeningeal disease, glycogen synthase is upregulated in leptomeningeal, definitely used for that. So it's these proof of concept pathways that we go down and just kind of see if there's something there.
Sorry, I forgot to introduce Mallory. You guys see a new face here. Her name is Mallory Hart. From the name, she's representing Trail. If you have any question for Chris, then you can ask Mallory.
You know, I'm gonna ask. So I think that I actually have two questions. First of all, thank every presenter for sharing all the wonderful presentation and also great experiences, especially from the patient advocacy perspective as well. And I think that no matter whether we come from the translational application or research drug screening perspective application for the iPSCs, or even going back to the embryonic stem cell lines, and then we all can acknowledge that there are line to line or individual to individual heterogeneity, or even sort of like from the clone to clone heterogeneity, right? So I think that I'm just wondering that can you actually comment on that, how you should actually take the cellular heterogeneity into the consideration when you actually try to develop the therapeutic application, or even probably relevant to like the drug screening application. So maybe Jeanne can also kind of address this perspective as well. And then I think for relevant to Jeanne's interesting space project, I think that when the things actually got into the space, it's not only just affected by the loss of the gravity. And one of the very important aspects is also that, for example, inside the space, then you can have a lot of the space radiation things, and then that can possibly affecting the cell, obviously. So then I'm just wondering that when you send the cells into the space, do you actually protect them from the space radiation?
Yeah, I'm going to answer your last question first.
Sure.
Because the answer is no. They are protected from radiation. We've actually had radiation monitors in the incubator, the little incubator that the cells go in. And the radiation is really minor. In fact, the whole space station is really protected, because there are people in there. So we've decided, and there also is people who have looked at upregulation of certain genes when you irradiate cells, and we don't see any signs of that. So I think we've eliminated that, even though it's a really good question. And the heterogeneity. So I have a colleague who worked for the FDA for a very long time, and she was in charge of cell therapies and a lot of gene therapies for deciding whether or not something should pass the FDA regulations. And what I learned from her was to embrace heterogeneity. That's what she said. You cannot avoid heterogeneity. In developing cells, there'll be some that develop faster than others. And so the idea would be to have in that population effective cells. You might have some cells that will not be effective, and hopefully you have some cells that don't become something else that you don't want. So that's where the timing of the gene expression, it's really important to have the entire culture using RNA-seq, not single cell RNA-seq, because we want to find out what the balance is of all those cell populations. And that's really the key to an effective transplant.
So,
did you have any more? Sorry, yes.
Yeah, so then from let's say the drug screening perspective, or actually disease modeling perspective, because actually my lab is also working on the disease modeling as well, and particularly in the rare genetic disorder area. So then we also see that there are a lot of, for example, potential clone-to-clone variation, or even sort of like the somatic cell source reprogramming. Somatic cell source may possibly affecting the differentiation derivative outcome. So then from that perspective, then is there any sort of like particular interesting strategy that taken place in iXCells that you guys are looking into to kind of like mitigate this type of variation, or how do you take this part of the factor into the consideration when you interpret it?
Yeah, actually, I think you raised a very common question. Yeah, heterogeneity, thank you. It's not something unique or special to treating rare diseases. I have worked in a pharmaceutical company for about 11 years. So that applies to all our drug development programs, regardless whether it's a rare or common disease. So that's my comment number one. And number two, how we are going to deal with it. There are several layers. So for me, I think some cases we need to, or the concern is more than the others. For example, it depends on the strategy we're taking. If, for example, we are regulating the gene, knocking down the gene expression, regardless is mutant or wild type, then our strategy at the very beginning, I think heterogeneity definitely is not our top concern. We need to identify the drug candidates first. Yeah, but if we are targeting allele specific, a mutant specific, of course, the patient-derived cell would be the best, or probably the only cell modular system model we can be used. So, or in other words, regardless what kind of candidates we have identified, the final test would have to be in the patient-derived cells to conform. But your concern is valid because the microenvironment, the whole gene expression profiling in each individuals, and also within the same individual, different cells might be different. That plays a role. Yeah.
I'm just going to continue to comment. There is a sweet spot. And there, there is you can reach that with any cell line. We hope. And if we can't, then we can't treat that patient. I mean, honestly, that's what it comes down to.
I just have a quick question. Actually, two. Do you have a situation of there we go.
Oh, you're just talking about this.
We have a situation where we differentiated the cells and we were doing a low selective knockdown and we lost the allele. What's the chances of that?
Well, in your case, 100%.
We haven't verified that.
There's a lot of that. I
Okay.
mean, there's a lot of structural changes when you target a gene,
Yeah.
and most of them are unknown. You just have to look for them.
Yeah,
But
yeah.
it's not going to happen every time, I promise.
Okay. Good. And then the last thing I wanted to ask was, so why were the cells mature?
He wants to know why they mature faster in space, and honestly, I'm supposed to be thinking of industrial applications for putting cells in space, because that's what NASA wants. And I think, oh, if they mature faster, that would be useful, right? But, we're a long way from that. And I don't know why. I have a lot more experiments I have to do, Jack.
Can I make a comment on that?
Yes,
That
please.
is probably also relevant to the question that I brought up, because there are so many environmental factors, differences, right? And I think that, okay, we put let's say the space radiation issue aside, because they are protected.
You have to set it aside.
Sure, I totally agree with that, but I think another thing is that during the launching process, there's a drastic gravity changes as well, right? And I think that that's certainly very different from the cells only staying on the Earth. So then, of course, that period of time, the strong gravity changes may actually just stay very short period of time, but that could cause a lot of stress on the cells.
Yeah,
And
so
stress can actually stimulate the aging or like the differentiation.
It's not really aging, let's be careful, yeah.
Sure.
But yeah, so we had a motion detector in the box, right? And the centrifuging cells in your lab, it's a lot stronger than anything they encounter. But also we discovered that the worst time the cells had, the most jostling they got, the highest gravitational pull they had, was when they fell off the FedEx truck.
I have a question. Why did they fall off the FedEx? It's more a question on some of the data that you shared with the rats, and that's implication also for broader, diseases in the neurologic space using the stem cells, which is because you've seen changes in the implementation and the neurites and such, and I'm wondering if that has to do with, and if you looked at the changes with age, like with neuroplasticity in younger, rats or later, because that could potentially shed light on therapies for children, if you want to go.
Yeah, no, I understand. And children still have neuronal precursors in their brains, so that makes a difference. I agree. But in this case, I think it's a maturation program that neurons have, and that enables them to make synapses with whatever environment they happen to be in. And that doesn't necessarily mean it's the right environment. There's a lot of mistakes are made, but those get pruned back later, so we fix everything, right? So I think that probably means that you could treat childhood diseases differently. I just can't think of an example in which that would be a really clear-cut way to do that.
I mean, if you try your models for the hemisphere in younger and older.
Oh, we don't, yeah. So our iPS cells are all, like, they start out 0. So whether we keep them a week longer or not, they're still real close to 0.
The animal one.
Oh, yeah, we have not done that. Yeah. Somebody may have, and I hate to say this because there's so much work that's been going on like the last 40 years. It wouldn't surprise me at all if somebody had done that, and I just don't know about it, or I knew and I forgot.
Okay, thank you.
I want to just quickly comment on the issues that Jeff brought up. I think that for the earlier laws, that's actually just echoing to what Jeanne said. It's very commonly seen because giving a very vivid example, like obviously people working on the Down syndrome disease modeling using iPSCs, and then this has already been found and published that for the iPSC coming from the Down syndrome with the trisomy 21 and during the culture or even differentiation, a lot of them actually fall back onto the becoming the disomy. So then this sort of like
that's another one of my papers.
Yeah, and I'm fully aware of that, right? So I think that this is also kind of like going back to, I believe that the cellular heterogeneity is definitely important. I think that along the in process, no matter whether we are developing the potential therapeutic modality or trying to model the disease, I think that the trying to in process monitor the whole things to a certain degree will definitely be very important thing to do, because otherwise you wouldn't be able to know that what you are actually looking.
Right, exactly.
Can I ask a question?
Sure.
Everybody, anybody? So I know that Down syndrome, for example, is a mosaic disease, and the severity of it is really dependent on exactly during embryonic development which cells became what. Are these other diseases, these rare diseases, mosaic?
Yes.
Okay.
Rett syndrome is
Yeah.
the same known as X-linked. It's actually good nice.
It would depend on when the mutation occurred.
Is that?
Yeah.
Yeah, mosaicism. Oh, I think it was in, I can't remember the rare disease, but there was a very noticeable contribution in mosaicism and potentially also contributing to, the same variant resulting in different phenotypes.
Yeah, let me make a cautionary note here. So there have been examples, and I unfortunately had one of these experiences, that when people make stem cell or iPS cell banks from mutant cells, if they are in fact mosaic, and you don't check your cells at the end, you could have wild type cells. And that happened to me once. So yes. Right. So yeah. We worked for a year on this. We discovered that they were wild type. And that it still pains me. Yeah, so you can't assume that the cell, just because you made iPS cells from somebody with a mutation, that you're going to end up with a mutation in the iPS cells.
I think that going back to the mosaicism question, because the mosaicism issue, I think that for the disease that my lab works on, it also, I think that based on the reported data, it appears to be that there could be some low degree of mosaicism as well, because it's not like the every single patient, once they actually have the genetic mutation, is completely abolishing the protein expression. So there's definitely one potential possibility that some of the cells existing in the patient body actually has the intact gene expression.
Absolutely. Yeah, and that may be necessary for survival.
It's possible, yeah. Because this is also relevant to, because we are working on the enzyme deficiency, and then the disease expressivity is actually very distinct, even for the two patients with the same genetic mutation, actually. So, and then it can be different in terms of how long the patient can actually live.
We're not trying to have just a lab meeting here. We're just like questions for other people.
I have a question.
Yeah.
Jeff, your comments on where do programs go to? That would be great. This timely. I just thought, someone just called me yesterday from CLN2 saying, what can we do with this? Right. That's not. I guess this is to the whole panel. And I just was fielding some questions the other day also. And I have this from council programs for my customers, gene therapy. Where can the data go to be used?
Yeah.
What do you guys think? Do you have the perfect world, or do you know of something? What do you think? Amber, yeah.
Well, is it a
I'd
failed
like your
company,
input.
or is it a company that failed, or is it a program that failed? Those are very different approaches.
Yeah, both, yes.
Yeah, both. Yeah, I mean, companies, they go to liquidation, they go to the trustee.
Yeah.
That's appointed. But that's, yeah.
I would
But
suggest
like a central repository where
Oh,
stuff.
I see.
stuff can go.
But they
this is
still,
actually stuff.
Yeah, I
physical
mean,
things.
it's gonna sit in, if it's gonna sit in a drawer somewhere,
Yeah.
well,
why
it
not
depends.
make it a drawer that someone else can open?
It depends on the value. It depends if it's a valuable asset. Basically,
right,
these trustees sit down and they have to try to figure out the value of those assets. And they spend months doing that. And then they take bids and they try to, sell it off to the lowest bidder. And that's what happened to Valerian or Heisman, sorry, not the lowest, yeah. Sorry. Thank you. I need to get oriented here. Up, okay. valerian is a good example of that, which is a Lafora drug. It's a fusion enzyme antibody fusion. It's a very large molecule. It's been approved for Pompe disease. It was picked up by the CSO of Valerian, and has now been repurposed in a new company, but it's not progressing. It's basically, there's no trial. So they're dosing and they're charging for this, $50,000 a patient to dose these patients for Lafora. Not really, I don't think he's advertising for Lafora. He's basically hoping it gets across the blood-brain barrier. This is a 150 kilodalton protein. Is it ever going to get, I mean, that's not going to work. But there's no effort to actually figure out whether it's in the CSF anyway. So I question that approach, as far as, I think there needs to be a prospective kind of, this is going to start to accumulate. People are going to start picking up assets that die because companies can't survive, and they're going to start, they're dealing off of wants versus a patient population that needs that drug. So they're willing to take it. So we have to approach this very carefully and be responsible at the beginning. That's my thought on this, but yeah. I mean, we're just experiencing this now. We're at the tip of the tip of the spear or whatever, because this is all new. I mean, all this technology has just happened, but it's going to start to accumulate. And I look at Lafora as a field of a mess because of all these halted programs, because the patient population isn't there. And there isn't a lot of strategy. There's some, but not a lot that's going into making it work. So I don't know.
I have a question. So I have a lot of questions actually, but I can approach to each one of you after this. But because the focus is rare disease today, so I want to ask a question to Amber. So what do you, because you've been working in this field for years, right? 3 years, I don't know. So what do you really want to see from this ecosystem, and how do you expect from this kind of joint force with the collaboration with the academia, the industry partners? So a lot of people ask, what define a success business model or success platform, right? So what will be the immediate needs from the patient advocates? Hear from your side.
It's a great question. What would patients like to see from industries, scientists, and doctors? And I think that we're evolving toward the correct model. It's just taking time. All 4 of those people need to have an equal seat at the table. A large focus of our patient symposium this year was that so many clinical trials fail because they don't talk to the patients, and especially with rare epilepsies, it's very measurable to count seizures, but seizure burden isn't necessarily what ruins everybody's day. It's behavior, it's cognition, it's movement. It may not be exact seizures, but it's seizure-like activity. And the more you can involve parents, we may not count every seizure perfectly, but we'll count whatever we see perfectly. And so the more we involve patients from the beginning in drug development, I think that the more drugs will become successful. And also allowing patients to be more involved. We're allowed to do things that industry is not, just flat out not allowed to do, and that scientists and doctors probably don't want to do, like go to the FDA and be angry, or go to their politicians and say, this is exactly what needs to change and why. We may not be able to explain everything perfectly, but we know who's important and who to pull into certain meetings. So I would like to see more patients involved.
I've been to a couple of patient-focused FDA meetings with patients. And it's not a good idea to get angry. I mean, it won't do any good because they're used to having people be angry. They're not used to people being really good at coming up with an argument that makes sense to them. So I don't know. I just think that the FDA is a bunch of people, not like a, they're not uniform. And if you can get somebody within the FDA to be a champion, then you're going to be much better.
I always like to think of rare diseases, and this is going to sound, I may strangely describe it because it's the end of the day, but you guys will understand the spirit once I'm done. Like in the 80s and 90s, it was all innovation in HIV and AIDS. And people with HIV and AIDS could march on Washington and they had very visible signs of being ill, but they still had their strength and they could march and show the unmet need. And cancer has been the story of the 2000s and 2010s. And now I really feel like it's the time for rare disease. The innovation is there. The patient populations are there, not in isolation, but they are as a totality. The differentiating factor, and I don't know how to address this, is that it is a lot of pediatrics and super, super sick people who can't go and march on Washington. And a lot, people with HIV and AIDS laid in body bags across Capitol Hill. And some days I would put my twins in there and just walk away for a hot minute, but that's frowned upon. So we probably should not do that with kids. How do we form a loud voice and receive the attention that we all need? It started with the patients and moved on to gathering greater scientific momentum, and industry was then empowered to go on and create the drugs. That's exactly what we need. And I'm not sure how to go about that.
Yeah, I've often used that myself as a way of actually changing things. But it worked really well. I mean, I know people who are in ACT UP, and I know that they, I knew them then. So I think with this, what you really need is to use the power of the press, because that's ultimately what people pay attention to. And I know that Peter Marks reads newspapers, so if there's lots and lots and lots of press on a particular subject, like pediatric rare disease, and why can't we fix it? Then I think that would be a good way to start. That's all I suggest. And I know you already have had some press, but I think you need something that's more coordinated among all the different groups.
Yeah, today we have a mayor here, we have a press here. So I hope we deliver the message to them. That's a beginning. Okay, I have another question for maybe Maureen, maybe. So I'm really curious. I'm so excited to see the data shared by Facebook. Maybe this is my first time really looking at your detail of your technology. So I'm curious, what is the major difference between what you're doing in Braille versus Ionis? Because Ionis also have data, a big data group, right?
So I'm going to answer this question by starting off by saying I'm the Director of Patient Advocacy and Policy. My PhD is in Science and Technology Policy. Okay. So, but I do think that the key difference that Crayon is trying to innovate on is, as Chris said, really thinking about how to engineer these molecules, how to move away. These are, as he said, these are polymeric molecules. We can think about them as repeating units. That lends a lot of opportunity to think about how to engineer them. We have been coming at drug development for, the last 150 years, thinking about trial and error. We find something that sort of works, seems to have some benefit if you, that we kind of acknowledge helps with pain or something like that. And then we started to, experiment with other things and see how that would affect pain. And then as the chemistry industry really started to evolve out of the dye industry, people started to be able to understand, well, these are chemicals, let's take them, let's tweak them a little bit, let's try them again. And then high throughput screening came through. And we've been going along this path. And it's one of the things that has worked for as best we can for small molecules, but we have an opportunity to think about doing something different. And that's where companies like Crayon, and there are others in the ecosystem kind of looking at engineering proteins and things like that, to think about how do we learn these design rules? How do we understand what makes these molecules safe so that we can then engineer them? We can say, okay, we need to target this part. We need to target this particular part of the genome. So we have this sequence. Let's think about what chemistries we need to make sure that this is a safe and active drug.
Yeah, it's so sorry.
Yeah, I have a related question. So Chris presented today about the different modifications, chemical modifications. So we all know for antisense oligos, the sequence matters, no doubt. But now more and more people realize the modification also matters, especially from the safety point of view, and also probably from the efficacy point of view. So I'm wondering if you have like a very brief summary, or do you have any profiling in terms of different modifications or ranking of different modifications?
I think the short answer there is that it's all of it is going to be interdependent. The sequence is going to be very interdependent with the chemistry when you're looking at safety and activity. And so there is, I don't think there's thinking about ranking the different modifications is quite the way that a lot of the models that Crayon has built is showing. It's really about how to understand, given what we need to target, what modifications can we employ to make this a safe and active drug? And in the future, because there's been lots of chemistries and modifications that have come forward, but they're not done. There's chemistries and different modifications that are being developed and published on frequently. And so that's, understanding how to incorporate those into thinking about how to develop safe and effective molecules is going to be really important as well.
Yeah, I'm just thinking that, and I know I shouldn't bring up AI, but the really fundamental basis of machine learning is that you have a thing and then you have an outcome. And you don't try to do anything biased. You don't say, I think this thing should have this outcome. Instead, you have a thing and you test it for an outcome, and you have another thing, and another thing, and another thing, and you get different outcomes. And eventually, what comes out is what works. I mean, it's really pretty astounding. You really need a lot of data, but start soon. That's what I would recommend. Not when you have like thousands of data points. Start with 10 data points and try to get a machine learning thing started.
Absolutely. And think about the right data as well.
Right, exactly.
Yeah, I mean, in the past, the way you think about chemistry and sequences, there were rules that we would kind of follow. Oh yeah, somebody told me this works this way, and basically I think what Crayon's doing is, and to some degree what we're doing is, we're taking it to the next step beyond the rules, and we're actually putting up an algorithm behind it that actually gives us much more precision on whether or not those rules are correct, or whether it's the context is different. Some chemistry may be toxic to some sequence, and that's something that's applied to it goes beyond our capacity to remember rules. Yeah,
So
exactly.
A lot of that challenge, though, due to the rate of infection. So you've got this balance with safety
Yeah.
regulatory bodies, and they're expecting to look back.
Yeah.
on what was said. in
Yeah,
the
yeah.
past.
But
And now we're in a space
the-
where we can design, we can test, and we can build things that we can dream of a year ago.
Yeah, you still have to go through the regulatory process of testing it, but if you can prospectively design in software a safe ASO, given a sequence and a chemistry, then you're going to minimize the cost downstream when you have to go through those tests.
But then that's kind of like playing middle ground, and trying to keep the rule book, but also.
Yeah, but the rule book,
Will go away.
he's saying the rule book is in the FDA, but
Yeah.
you know, we don't have to use two species anymore for safety. The length of time that you do a safety study is malleable with the FDA. If it makes sense, they'll say it's okay. And I don't see I see rats and mice being used in the future just to find out whether the thing is toxic. Something terrible happens to them, right? But other than that, that's all they really care about. They don't even care if it's not efficacious. That's your problem.
I believe the other one is really effective.
Yeah.
No, absolutely, and I think what I was trying to get to is it's this combination with the long tail. The way that you link discovery in the long tail back to kind of like larger patient populations. So yeah, the mindset's got to change in how do you link the data that you're seeing and the new models that you're using to give you indications of how it related to the past to say X is equivalent to Y 5 years.
The key is to go after tolerability first. You have to be able to predict tolerability.
So you could get a model in iPSCells in which you could figure out what toxicity is, what toxicity is. Yeah, tox.
Right.
Yeah, a tox in a dish.
Well,
I like
I also, I'll say.
It's yours.
You know, looking at that one graph that Chris showed up, those were predictions actually, and against true toxicity. So that's one of the things that CRAD is really building, is to be able to understand how we can predict toxicity. And there was a slide he didn't share, but the models are getting up to roughly about 80% at this point, 80% across all different types of toxicities, hepatotoxicity, kidney toxicity, neurotoxicity.
So I suspect, just from what I know about the brain, is that you don't want to isolate a single type of neuron. You want lots of types of neurons. And you want the immune system as well. So you need to add microglia. I know that's hard, but you need to add microglia. So a system like that, in which the toxicity could come through the microglia, then I think that'll inform a lot better than a single cell type. So that's what I would recommend if you're going to build an in vitro model, that you make it more complex than you think it should be.
You can play on top because no matter how complex it is, it will be.
Yeah.
Absolutely.
Yeah. I don't choose the most part.
I just have a general question for probably everyone, because today we're talking about rare diseases. But I think it's more related to like personalized medicine. So to all of you, what do you think, how we can really develop personalized medicine? Because in traditional way, when we are developing a drug, there are clinical trials, you actually can learn from their failures. But for personal medicines, each individual will be so unique. So even one method doesn't work for this patient, it might work for the other patient. So, or the other way around, it might work for this patient, but the same strategies don't work for the other patient. So is there a way we can learn from personalized medicines previous any experience that can apply for later personalized medicine development? Yeah, it's a very general question.
Yeah, actually, there's a lot of discussion around that. So there's a big initiatives. Actually, there's several nonprofit organizations already doing, which is data, right? We need to share data. So each patient is so unique, but by sharing all the methodology, preclinical and clinical data,
and you guys just talk about the tox. We probably cannot really learn the efficacy from individuals, but toxicology definitely we can learn something, right? The NYC actually is doing something. They are trying to build something upon that. And I told Julia, I told Winston that, instead of trying to build a big building out of that, maybe we can start from just create a marketplace or data sharing platform, right? We're generating tons of data. We have data from Crayon. Actually, I'm hoping we have very good hits. Hopefully, this is the second big round, actually. The first round, Rohan screened 850 ASOs. Very good hit, beautiful knockdown, but toxicity. So this time they are using your technology. Hopefully, the toxicology can be improved. So think about this: if we deposit all these data in one place, right? People can see is a sequence or it's a combination of your modification or it's a combination of both, right? So this will all and I like AI a lot. You need to use the AI. It's the era of AI. You need to use the machine to tell us, right? I'm biased, right? You don't want to predict. You want machine to tell you, right? So I think that's a way eventually we can address that. Again, it's one of the very, very challenging thing for efficacy is really endpoint, right? So we have a big topic around in the SLC61. I remember people talking about endpoint. So each patient is so unique. Even SLC61, I remember there's a section, everybody going up to the stage telling their kids' symptom, right? They're all carrying the same mutation, same gene, but their symptom is so different, right? So it will make the endpoint and also efficacy is very, very challenging to set up this, right? The benefit and risk. But I'm supporting Amber saying, that's why it's because this is so difficult. That's why the patient's voice is so important. So your view and I remember there's a lot of topic talking about different metrics, right? So what will be the most important value or metrics for the life improvement? One is whether the kids can go to the bathroom independently, right? So if your patient didn't give out their voice, nobody gonna think about this. They will think about a seizure. They will think about the brain development or cognitive things. It's very hard to measure, right? But we need to start from some very, very basic measurable thing.
Yeah, as part of the analysis for Parkinson's patients is the diary that they keep. And in fact, in this trial that I told you about, the best results were with the diary. The unbiased results are not as impressive.
Yeah.
But that's all that really matters, doesn't it? And if it is a placebo effect, then good for placebo effects because they feel better.
If we can predict placebo, that would be good.
That'd be great. Absolutely.
So correct me if I'm wrong, but people may know the SMA story better than I do, but from what I remember, it was the patient feedback on patients who had multiple copies of SMN2 that had less severe disease that led to understanding that the paralog was an approach to therapy. So it's really important to have, when you see spectrum like that, to have those patients sequenced and potentially have a therapeutic approach that's not necessarily the target itself.
I think we can wrap up here, and thank you so much, everybody. I really, really appreciate it.